Nobody is there.
Once again, a very good morning. I request all of you to kindly switch off your mobiles for ICR song, please.
जय जय कृषि परिषद भारत की, सुखद प्रतीक हरित भारत की। कृषि धन पशुधन मानव जीवन, दुग्ध मत्स्य फल यंत्र सुवर्धन। वैज्ञानिक विधि नव तकनीकी, पारिस्थितिकी का संरक्षण। शस्य श्यामला छवि भारत की, जय जय कृषि परिषद भारत की। हिम प्रदेश से सागर तट तक, मरुधरती से पूर्वोत्तर तक। हर पथ पर है मित्र कृषक की, शिक्षा शोध प्रसार सकल तक। आशा स्वावलंबित भारत की, जय जय कृषि परिषद भारत की। जय जय कृषि परिषद भारत की।
Thank you. Respected dignitaries on the dais, respected teachers, scientists, scholars, and students, a very warm welcome to one and all for the 2 days Student Symposium on Genomics-based Next Generation Crop Improvement Approaches, organized by the Division of Genetics, ICAR-Indian Agricultural Research Institute, New Delhi, in collaboration with DuPont Pioneer, in commemorating 150 years of Mendel's loss of inheritance. On March 8, 1865, Gregor Johann Mendel presented his findings entitled Experiments in Plant Hybridization at the Natural History Society of Brno. His experiments in pea plants allowed him to elucidate the laws of genetic inheritance, which predated the molecular era. The symposium is thus organized to commemorate 150 years of Mendel's loss of inheritance, with an intention to provide opportunity to postgraduate students and young research faculties to interact with both public and private sector experts who will be delivering presentations on different advanced tools and techniques of genomics for crop improvement applications. Let us start this auspicious program with our Saraswati Vandana. I request The PG students choir to render the Saraswati Vandana, please.
जय जय हे भगवती सुरभारती तव चरणों प्रणमाम्यहम्। जय जय हे भगवती सुरभारती तव चरणों प्रणमाम्यहम्। नाद ब्रह्ममयि जय वागेश्वरी नाद ब्रह्ममयि जय वागेश्वरी शरणं ते गच्छाम्यहम्। जय जय हे भगवती सुरभारती तव चरणों प्रणमाम्यहम्। त्वमसि शरण्या त्रिभुवनधन्या त्वमसि शरण्या त्रिभुवनधन्या सुरमुनिवन्दितचरणा नवरसमधुरा कविता मुखरा नवरसमधुरा कविता मुखरा स्मितरुचिरुचिराभरणा। जय जय हे भगवती सुरभारती तव चरणों प्रणमाम्यहम्। आसीना भव मानसहंसे आसीना भव मानसहंसे कुञ्जतुहिया शशिधवाले हर जगता गुरु बुद्धि विकासम हर जगता गुरु बुद्धि विकासम शीत पंकज तनु विमले। जय जय हे भगवती सुरभारती तव चरणों प्रणमाम्यहम्। जय जय हे भगवती सुरभारती तव चरणों प्रणमाम्यहम्। प्रणमाम्यहम् 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 प्रणमाम्यहम्।
Thank you, friends. I request the dignitaries on the dais to kindly light the lamp and pay floral tribute to Father of Genetics, please. Thank you, dignitaries. I now request Dr. R.K. Jain, Dean and Joint Director Education, IARI, for the welcome note, please.
Very good morning to all of you. Chief guest of today's function, Professor M.S. Swaminathan, an icon in the field of agriculture. Dr. Ravindra Kaur, Director IARI. Dr. A.K. Singh, Head Division of Genetics. Dr. Raman Babu from DuPont Pioneer. Invited guests, Professor Anand Singh, Professor Balram Sharma, Professor Sarkar, Professor V.P. Singh, it will be difficult to name everyone, directors of sister institutes, heads and professors of the different divisions, and my dear distinguished scholars. At the outset, on behalf of the institute and on my own behalf, I welcome you all to the Student Symposium, which has been organized to commemorate 150 years of Mendel's laws of inheritance on genomics-based next-generation crop improvement approaches. As already been mentioned, this symposium has been organized in conjunction with DuPont Pioneer, which is aiming at to reach the next generation of plant scientists since 2008. Prior to IARI, DuPont has organized a similar kind of symposium in PAU Ludhiana and subsequently at Hyderabad actually. And this is the third in the series as far as Indian scenario is concerned actually. We are fortunate to have Professor M.S. Swaminathan, who symbolizes the dreams and desires, ambitions and aspirations of both the farming and the scientific community within the country and offshore actually. He has been associated with this institute since 1947. Can you imagine 70 years of association actually in various capacities. Actually. Besides Green Revolution, all of you will agree with me, he will be always remembered for creating ARS service. Actually. In fact, we are the beneficiaries of the service which he has created, and I think the coming generation too will be benefited by this service actually. On behalf of the director, the faculty, the staff of the students, and on my behalf, I once again profusely welcome Professor M.S. Swaminathan, and I would request Dr. Director IARI to welcome Professor Swaminathan with a bouquet of flowers, please. Before I request the Director IARI to give her address, I will briefly present before you what is IARI and the PG school actually. IARI, as you all know, has a rich history of more than 110 years actually. This institute, if you recall, was first established in 1905 at a place which is known as Pusa, Bihar, and the building which you see is the called Nolakha building, which was destroyed when there was a severe earthquake in 1935. And in 1905, we started with 5 major sections, including cattle breeding. So you can imagine, in 1905, we have the animal component too. Of course, now we are mono-faculty. We do not have veterinary or animal component. Let us look at how the vertical expansion has taken place as far as this institute is concerned. And let me make a mention. Our seniors were very cautious while going for vertical expansion to see that excellence is maintained actually. In 1905 to 1950, we only added 1 more divisions actually. So until 1950, we had only 6 divisions. Subsequently, in next 20 years, the divisions rose to 11. And then between 1970 to 85, we had only 14 divisions. And now between 1985 to 2016, the number rose to 19. So you can imagine we have been very cautious when it comes to vertical expansion. As you know that IARI has a multi-crop mandate and it has to address the problems which are there throughout the country. And to address those problems, we have 8 regional stations. And then besides regional station, we do have the off-season nurseries. Then of course, besides, we have the outreach programs. We have outreach program in Bangalore for the field of horticulture. We have the outreach program in Bhopal for the field of agriculture engineering. And soon we are adding IARI-like institution in Jharkhand and another institution in Assam. IARI knowingly or unknowingly has been following the meta-university complex. We were trying to utilize the strength or synergy of the various institutions which are available on the campus. For example, we are trying to utilize the strength of ISRI, that is Indian Agricultural Statistics Research Institute in the field of agricultural statistics, computer application and bioinformatics, National Bureau of Plant Genetic Resources for plant genetic resources, National Research Center for Plant Biotechnology for molecular biology and biotechnology, and then Indian Institute of Horticultural Research, and so on and so forth. Actually, let us know IARI by numbers. Actually, IARI at any given point in time is a micro university with multi-crop mandate. At any given point of time, our strength is roughly around 4 figures, that is 1,000 something. It's a deemed university with mono faculty and multi-crop mandate. We offer MSc and the PhD program in 26 disciplines. This is our 59th year of human resource development, and you will be happy to note that 20% of the human resource which goes to ARS scientists comes from IARI. We have at present 647 faculty members, including 24 adjunct professors. We have 159 faculty members from other ICR institutes outside Delhi. This year we had admitted 357 students, including 15 foreign students. So far, 9,395 students have graduated from this institute. And then, of course, 25% of the NAS fellows are IARI alumni. And every year, at least, we have 1 to 2 Jawaharlal Nehru Awards for their best theses. And of course, then we have 5 centers of advanced faculty training. This is just to give you an idea for the benefit of my younger students that who have been leading this institute and have provided the leadership since 1950. These are the living legends from Dr. M.S. Swaminathan, Dr. H.K. Jain, and so on. And then lastly, we had the Dr. Mahapatra, who is presently now the DGICR. Actually, besides the directors, you can have the various deans since 1950 who were looking after the PG school. And these are the living legends: Dr. N.N. Goswami, Professor S.L. Mehta, Professor Anupam Verma, Dr. Pramatma Singh, and Dr. H.S. Gaur. IARI has provided leaders to groom the next generation. Actually, take the example of 3 World Food Prize winners: Dr. M.S. Swaminathan, Dr. Raja Ram, and the Dr. Vassal, whose figure is not there. So we have the 3 World Food Prize winners. They are again IARI alumnus. And then, of course, we have Dr. V.L. Chopra, Dr. Paroda, and so on. So they have groomed the younger generation for future agricultural research and education. The question arises, where do we stand today? As far as Indian scenario is concerned, when we compare Indian agriculture universities, IARI is the number 1, and according to Career 360 University ranking, consistently we have been ranked as 4A plus actually. But when it comes to top 100 Indian universities, including pure science and other universities, IARI is at 39th position, whereas Indian Institute of Science, which got the deemed university status at the same time, that is 1958, is now number 1. So our challenge and our aspiration is, so can we be at a par with at least Indian Institute of Science Bangalore? However, of course, we do not figure when it comes to top 1,000 global universities, and that is another challenge, that how can we be at least in top 1,000 global universities. This is just to give you an idea that which are the disciplines where we are offering MSc and the PhD program at IARI Delhi. As I said earlier, at IHR Bangalore, where we have the outreach programs, we are offering PhD program in 4 disciplines. When it comes to CIEA Bhopal, we have the PhD program in the field of agriculture engineering. This is just to give you an idea that how does the PG school functions. Director IARI, who is the Vice Chancellor of this university, is the Chairman of Academic Council, and Director is being assisted by 4 Joint Directors, Joint Director Research, Extension, Administration, and Academics. And as we know that IARI works in a school mode, we have a School of Crop Improvement, School of Natural Resource Management, School of Crop Protection, School of Basic Sciences, School of Horticulture Sciences, and then School of Social Sciences. IRI has always been the most sought destination for the students. If you look at the statistics of the last 6 to 7 years, you will find that for each seat, there has been 10 to 15 applications, and consistently that has been the case actually. IRI has always been the most sought destination for international students also. This is just to give you an idea that what has been the figure during the last 10 years. Presently we have 49 students from representing 13 countries. This is just to give you an idea that how many students have graduated from this institute. And convocation has always been the most important activity of this institute actually. And this year we had none other than our president as the convocation chief guest. When you look at the strength of this institute, the strength of the institute completely depends upon its faculty. And when we analyze the age profile of my faculty, you will find that on an average it ranges from 40 onward actually. So that's a very healthy sign. So that means they have at least another 20 years to serve this institution. But if you look at the number of the faculty and the age in which range, we have both young and energetic faculty, and we also have experienced faculty. So that's a very positive sign as far as this institute is concerned. Strength of the faculty can always be determined by I-10 index and H index. This is just to share with you some of the people for which H index and I index has been projected. As far as the number of publications emerging from students' research as well as faculty research is concerned, it's a very healthy sign that the 50% of the share comes from student research. If you look at the number of publications which are come during the last 3 years, you will find that these are the number of the publications with NAS rating more than 6 actually. And of course, there are some publications which are even NAS rating with more than 10 and above actually. This is just to give you an idea that discipline-wise that what are the number of publications coming with their NAS rating. And you can see here there are some of the publications. There are 10 publications which are coming in molecular biology and biotechnology with NAS rating more than 10. So that is what is required, that students, while they are doing their outline of research work, they must plan in such a way that they aim at publishing in a journal with NAS rating more than 6 and above. This is just to share with you some of the publications with NAS rating more than 10. And the number 1 publication is in Nature Biotechnology with NAS rating 20 actually. This is just to again share with you that some of the publications with highly cited, which are highly cited actually. Strength of the faculty is also determined that by some of the national awards they got, and these are the figures over the years that the faculty has been receiving Borlaug Award, ICR awards, and NAS awards, and Prime Minister Fellowship, and so on. This is just to show you that what are the courses available with PG school in our portfolio. Maximum number of courses we have in Natural Resource Management, followed by School of Social Sciences, then of course in School of Crop Protection, School of Crop Improvement, and then School of Basic Sciences. And these are some of the new courses which have been added to our portfolio, like Climate Smart Agriculture, then International Agriculture Issues and Challenges, Introduction to Green Economics, and then of course Introduction to Emerging Agriculture Technologies, and lastly Advances in Instructional Technologies. Besides structured MSc and the PhD program, this institute does offer short-term and the long-term training programs. This is just to give you an idea that the number of the training programs being organized, and also how many faculty from SAUs and so on, they get benefited out of this short-term and the long-term training programs. Besides, we do organize certain special lectures such as Lal Bahadur Shastri Memorial Lecture. Then we also organize BP Pal Memorial Lecture. Then also more recently in September we organized the Teacher Day Lecture, which was delivered by our DDG Education, that is Dr. N.S. Rathore. Having realized the importance of IARI in terms of human resource development or the capacity building, the Government of India, Ministry of External Affairs, has assigned the responsibility to IARI in establishing the first agriculture university in Afghanistan, that is Anastu Khandar. and I am happy to share with you both for program in Afghanistan as well as Myanmar. Professor M.S. Swaminathan is the chairman of the task force committee. It is under his leadership that both these programs have been designed actually. This is just to share with you that we organized 6 months training program for 25 Afghan experts representing different disciplines of agriculture and veterinary sciences. Subsequently, we conducted 3 months training program for their technicians as well as faculty. Then we hosted first batch of 21 students of SCM agronomy, and this is just to share with you the picture of the graduation ceremony we conducted at IARI. At present, we have second batch of 20 students of SCM agronomy who are undergoing the coursework at IARI from Anastu, that is in Kandar. This is also to share with you that we have the responsibility of establishing Advanced Center for Agricultural Research and Education, popularly known as ICAR, in Myanmar. More recently, we conducted a short-term training program for their faculty, and let me share with you for Anastu's program, we have Professor Anupam Verma as consultant, and for this program, we have Professor S.L. Mehta, who shall joining Myanmar as a resident advisor. Then these are some of the initiatives which we have to undertake for quality education. There has to be a continuous building of the faculty competence because it has to be a continuous process. Then of course there has to be a regular updation of course curriculum. Then we have established a working group on massive open online courses, what is known as MOOC. Then of course we are thinking of initiating an integrated MSc and the PhD program for quality research. Then of course initiation of online teaching. For the first time we did initiate online teaching for the students of Anastu at Kandar actually. And then of course we are trying to address plagiarism by using a software that is called Turnitin. All the MSc and PhD theses which are submitted to the PG school, they are passed through this software and a similarity report is generated. Unless that similarity report is being examined, we do not accept an MSc and a PhD thesis. These are the three major challenges before IARI. IARI is known for its green revolution. In my opinion, in the next 10 years, IARI should be known as seat of innovation. Then of course we need to generate job providers rather than job seekers. Then of course we need to ensure excellence with the relevance actually. Any education system, for that matter, higher agricultural education system also entirely depends on 3E, that is expansion, excellence and equity. When it comes to equity, that is beyond our control, but nevertheless, expansion and excellence is within our hand, and I am sure we will try to see that we have excellence with relevance without expanding too much. Thank you so much for your patient hearing, and with this, I would request the Director IARI to give her address, please.
Thank you very much, Dr. Jain. A very good morning from my side also. At the outside, on behalf of all staff and students of IARI and my own self, I would like to extend a very, very warm welcome to our very beloved, very honorable Professor M.S. Swaminathan ji, who, for the sake of the students, I am repeating, was ex-director of Indian Agriculture Research Institute and also ex-director general. So basically, he is the creator of both IARI, Indian Agriculture Research Institute, and ICAR. So I would like to request the students to give a big hand of applause to our most revered leader, Dr. M.S. Swaminathan, sir. Thank you so much, sir, for joining us. And even at, with this kind of a health, you're looking very young, sir, though, but still you are amongst us. Thank you so much, sir. I'd like to also extend a very warm welcome to Asia Pacific, DuPont pioneer, and my colleagues on the dais, Joint Director Education, Dr. R.K. Jain, Genetics Head, Dr. A.K. Singh. In fact, it is his baby today. And many dignitaries of the dais, many stalwarts from the Division of Genetics and Plant Breeding. I very humbly welcome all of you, sir. Thank you for sparing your time. My Joint Director colleagues, Dr. Prabhu, Dr. J.P. Sharma ji, heads and professors of various institutions, directors of sister organizations, all principal and senior and junior, all scientists, and most of all the students from various state agriculture universities. I very humbly I welcome one and all of you. I'm sure this is going to be a very memorable experience, especially for the students. As we know that this is kind of a traveling seminar which is being organized by DuPont Pioneer, particularly I think to showcase and disseminate the high-end breeding analytics, I mean the tools and techniques which are used for analyzing high-ended breeding data. And I think it's a very good venture, I must appreciate. And I think you have been carrying out these seminars throughout the globe, and this is third in the series in India. And we are very grateful to you for choosing Indian Agriculture Research Institute. Thank you very much. And colleagues, simultaneously, I understand that we are celebrating 150 years of Mendel's law of inheritance. I remember when I was a biology student, I also used to read all these things, and I'm very happy that today I'm a part of this function, in which I have got a chance to commemorate such a worthy leader in the genetics and plant breeding. Well, we all know that there is no dearth of data globally. We have a flood of data in the globe, and the need of the hour is how to transform this flood of data to the rivers of information and ultimately transform it to the drop of knowledge. And this whole symposia is basically to expose the students that how this high-ended breeding data, the big data, can be analyzed by means of appropriate genomic and phenomic tools. And as I understand that this would basically educate the students to not only appreciate the genetics and its worth in crop breeding and in development of the novel germplasm, but also simultaneously to understand that what exists globally and where we are and what we are lacking and how to bridge this gap. And Dr. Rekha Singh was telling me this is going to be all live webcasted, which is a very good venture in fact, because as I see, not many students are here with us today. So webcasting would enable and help the students across the globe also, and not only globe but to most of the state agriculture university students who have not been able to come today, or even to the research scholars working with certain private organizations. I think this web link has been passed by Dr. A.K. Singh to all of them, and it has been very widely disseminated. With this, I would like to not come in between you and our leader, Dr. M.S. Swaminathanji, who is going to give us an address on this particular subject. And before I leave, I'd like to once again welcome all of you, especially the students for whom this 2-day student symposium has been designed. Thank you very much.
Thank you, Madam, for the remarks. I now request Dr. Raman Babu, Technology Deployment Lead, South Asia, DuPont Pioneer, for introduction to DuPont Pioneer Plant Science Symposia Series and 2016 Symposium on Genomics-based Next Generation Crop Improvement Approaches. So please.
Our respected and most beloved professor. When we have to address our professor, we really run short of adjectives. So I will keep it at our most beloved professor, Director, IARI, Dean, IARI, Joint Directors, IARI, and Head Division of Genetics, and honorable faculty members, and from IARI as well as state agriculture universities, and most importantly, the major target audience for this symposium, the students from IARI as well as from the state agriculture universities. A very good morning to everyone. And being an alumnus of IARI, I just can't tell you how incredibly happy I am today that finally the symposium is happening, and we are so very specially thankful to Dr. A.K. Singh for making this happen and for making Professor Swaminathan being here. I think that's one of the greatest accomplishments for this symposium, and we are extremely thankful to you, sir, for your precious and invaluable time. I think your presence itself is an amazing inspiration for all the students, for the faculty, for everyone who is even remotely associated with agriculture. I think there is no bigger joy than listening to Professor. So I think I'm incredibly happy about that. Today I am here representing DuPont Pioneer. DuPont Pioneer is a legendary agricultural genetics research company. It was started way back in the 1920s by another very legendary man called Henry Wallace, and I am sure you must be knowing. Henry Wallace went on to become the Vice President of the United States in the 1940s. And incidentally, I recently learned that it was Henry Wallace who persuaded the then Rockefeller Foundation to fund a small organization called the Office of Special Studies out of the United States in collaboration with the Mexican government, which hired Dr. Norman Borlaug, who was a researcher with the DuPont company at that time. And then this institution went on to become the International Wheat and Maize Improvement Center later on, which is called CIMMYT. So I'm incredibly happy that I'm working for CIMMYT. I used to work for CIMMYT for close to 8 years, and I didn't know this piece of history. So I recently learned this from the president of Pioneer, Paul Schickler, in his address to CIMMYT on the 50th year of celebration. So, and who knows history better than our professor? So we will be hearing from him later. So I would not like to stand between you and the professor, but I just want to very briefly touch upon this Plant Science Symposium series, which was initiated by DuPont, and DuPont is the parent company of Pioneer. So I just wanted to very briefly go through a few slides. And this is one of the significant flagship initiatives of Pioneer, just to enhance the capacity, the student research capacity in the countries across the world. And from 2008 until 2016, almost like 65 events in 5 continents have been held, and almost in all the major universities, as you can see, UC Davis, UC Berkeley, CIMMYT, Minnesota, and also on the Asian side, a number of leading institutions, the symposiums have been held, and they were very, very well received and very much appreciated by the young faculty as well as students. So I think one of the very important aspects of this symposium is that it's fully student driven. So that's why I kept insisting, Dr. A.K. Singh as well as others, that whenever we have to introduce, we have to speakers, and whatever we do here, it all should be driven by the students to give them that organizational experience. Even some of the topics were picked up by the students, and in fact we requested them to also identify some of the faculty for those topics. So it's fully student driven, and it's most importantly free participation. And in India we have had 2 symposiums until now, one in 2014 that was held in Punjab Agriculture University, and one in 2015 that was in the Professor Jayasankar State Agriculture University, Telangana. Right, very briefly about this symposium, 2016 symposium IARI. I am from IARI, and I have greatly benefited from the faculty here in IARI. We have today with us Professor Sarkar, from whom we learned the very basics of haploid, and some of his work on stock 6 has huge commercial implications. We have Professor Balram Sharma here, who used to teach us development of gene concept, and we have Dr. N. Singh, who is my Guruji, and he introduced me to the world of QPM, and we have Dr. K.V. Prabhu here, who used to teach some of the finest quantitative genetics courses. Sir, we use even today, every day in our job, the concepts that you taught us. We have Dr. A.K. Singh, who is very fundamentals of plant breeding, heterosis breeding, innovative approaches in plant breeding. These are tremendous courses that we have learned from you, sir. Dr. Mahapatra, you used to run eukaryotic molecular genetics, I think one of the most popular courses. And Dr. B.M. Prasanna, so he used to run elements of genetics, the developmental genetics and innovative approaches. I think we have greatly benefited, and I always felt that we have had the most illustrious set of teachers, and it's so nice to see that you keep continuing that job, and you keep producing a lot of students who benefit from this. And so this is the topic for this symposium we selected in consultation with Dr. A.K. Singh. This is basically, in keeping in mind that there is an unprecedented explosion of genomic information happening, and the volume, the variety, the velocity with which data is pouring into the breeding programs is something unprecedented. But the most important thing is, how do we convert all this voluminous data into a meaningful selection units that breeders can act upon? So this will be the common underlying theme for all the technical discussions that we are going to have in the symposium. And that's how we picked up this topic, and we have very wide range of topics, from pedigree breeding to whole genome prediction to genome editing, and everything will be covered by experts who do these things day in and day out in their labs. And Pioneer has also been supporting endowed chairs in universities, and it's mostly happening in universities like Iowa State University and other universities in the United States. Pioneer has been supporting, also DuPont has been supporting the Young Professor program. I think some of the brilliant minds are recognized for their hard research talent, and this has been going on since 1968, and over 700 young professors have been awarded until now. These are some of the very successful brilliant minds that we have been able to identify, and one of them is an Indian researcher who has done some cutting edge plant genetics research. I'll be more than happy if an IARI faculty gets this award. That would be my day. So, and also Pioneer has been supporting graduate scholarships across universities, and this is sort of summary of how the way in which Pioneer has been engaging the academic institutions all over the world. And I understand that most of these activities are mostly concentrated in the United States currently, but then the company is expanding very significantly in the Asia Pacific region, and I foresee tremendous opportunities for intensive engagement with the academia. And I don't want to stand between you and the professor, and we are all very keen to listen from him. Just one final message. Dr. Hans Bharadwaj is our Senior Research Director for Asia Pacific region, and he was to be here today, but because of a minor health issue, he couldn't make it. So he conveys his good wishes for the successful conduct of the symposium. And once again, we are extremely thankful to IARI, and this has been an incredible place, and I gained a lot of knowledge, and also I gained my better off here. So it's a very special place for me, and thank you very much. Thanks a lot.
Thank you, sir, for briefing about Plant Science Symposia series of DuPont Pioneer. Now it's the most awaited event. I feel very fortunate to invite the renowned world scientist of rare distinction, a great visionary, father of Green Revolution in India, Padma Vibhushan, Professor M.S. Swaminathanji, to deliver the inaugural address on Food for All in the Anthropocene Era. Sir, please.
Director Dr. Ravinder Kaur, Dr. R.K. Jain, Dr. A.K. Singh, Dr. Raman Babu, distinguished faculty and staff who are here. At the same time, the most important, those who are going to shape our future of genetics, the young students. I want to compliment DuPont Pioneer for this initiative. I think it's a wonderful idea of bringing students from different parts of India together, exchange ideas, and also discuss issues of importance to the future. For example, this very auditorium where you are sitting now was first used in 1964. It was constructed in 1963. The very first meeting was on how to prove the prophets of doom wrong. People like Ehrlich, people like Paul and William Paddock, the very first symposium we had here. And many young students participated. They were enthusiastic that we must prove the prophets of doom wrong. But you can't just say we should prove the prophets are doing wrong. You have to act, you have to do something about it. What is the technology, what is the approach, what is the... So, it's a very historic hall. Also in the name of Dr. B.P. Paul, about whom you mentioned. I want to say a few words about how can you contribute to achieving food security for all. Food, not only food security in the larger sense of nutrition security for everyone, because our country has made enormous progress in science, technology, in many places. But what is still shameful is the fact that a very large number of people are not... Shall I use this alone? Can you hear me at the end? There's a problem, huh? It's all right. I use this one alone. Can you hear me at the end, towards the end? All right. I'm happy. I'm going to share a few thoughts with you because, as I said earlier, our country is an enigma. An enigma, on one hand, we have made tremendous progress in science, technology, in every field of human endeavor. At the same time, which one do you... If you use both, you'll have a problem. At the same time, there are a very large number of children, women and men getting to bed hungry in our country. In fact, one of the countries where malnutrition is very high. How do we reconcile the two? Progress in science, progress in agricultural science, progress in production of various commodities, at the same time prevalence of both undernutrition and malnutrition. I want to address this issue briefly because the students who have gathered here are going to shape the future of our country. You are going to shape the future of our agricultural research, education, and thereby the future of food security in our country. Before doing that, I want to congratulate DuPont Pioneer for this initiative. I have known Pioneer for a very long time, from Henry Wallace. Great people are associated with Pioneer. In fact, if you use the word Green Revolution, if you see the meaning of Green Revolution is increasing production through productivity improvement, not through area expansion, but through yield enhancement. That is the correct definition of the Green Revolution. You see the Green Revolution as productivity improvement. The very first Green Revolution was initiated by Pioneer in hybrid corn. Hybrid corn was the first one where you had a quantum jump in production. And therefore I would congratulate Pioneer and DuPont for what you are doing. I saw some literature here. I think more such companies are needed. The food for all in the Anthropocene era, I do not know how many of you have heard the term Anthropocene. This is the new era which has begun. At the International Geological Congress held in August 2016 in Cape Town, the assembled scientists came to the conclusion that a new geological epoch, the Anthropocene, needs to be recognized as the dawn of human-influenced age. The difference between the earlier ages and the present one is much of the change is being brought about by human action. Human action or human inaction. If we don't do, or if you do, what we do. That is why it is important for the young people that you can shape this era, either for good or for bad. And the Anthropocene map of genetic diversity has appeared in Science magazine recently. Their conclusion in that particular analysis was that the Anthropocene is witnessing a loss of biodiversity with well-documented declines in the diversity of ecosystems and species. Biodiversity again is the very backbone of food security, nutrition security, livelihood security. Only recently we had that Agrobiodiversity Congress, the first Agrobiodiversity Congress, which was held here earlier this week. And one thing which came out very clear there, the loss of agrobiodiversity, the loss of every gene and species will limit our options for the future. In an era, Anthropocene era, as well as climate change era, the loss of every gene will be a loss which may have a great impact. That is why conservation of genetic resources has to be part of the agenda for use of genetics. Genetic erosion has been going on ever since agriculture started, because food and health security dependent on the past on several hundred plant species. There are books by National Academy of Sciences of the United States, The Lost Crops of the Inca civilization, The Lost Crops of Africa, which point out how in the past we all depended upon a very large number of plant species, both for our food security and for our health security, for medicinal plants. But unfortunately, slowly, with market economy, the number of plants have shrunk. For example, in our own country now, until recently, the public distribution system dealt with only wheat and rice. In the past, there were a large number of millets, large minor millets, major millets, sorghum, bajra, even the high yielding varieties program, initiated in 1968, had 5 crops, not only wheat and rice, but bajra was a very important one, maize and jowar. But gradually our food basket has shrunk, and that also is a cause for malnutrition, not undernutrition, but malnutrition. I had the great good fortune of being in the Mendel's garden in 1965. I was then head of the botany, what was then called the botany division, or genetics division of IARI, and I was invited to give a speech on Mendel's work in relation to what is happening in India. I had gone there. The meeting was held in the monastery where Mendel worked, Bruno, and they had redone what he was the piece, 3:1, very exact ratios, 3:1, 9:3:1. They were all showing there, but it was clear they had revived Mendel in the eastern part of Europe. Lysenkoism had taken roots officially because official support from particularly the Russian government, but gradually Mendelism again came back. One interesting thing with Mendel was we visited the village where he was born, Hensike, which is not very far from Brno, and there they had revived repainted his house and renovated his house. And I asked large number of people who were gathered there from the village, what did Mendel do for you? What is genetics? What is Mendel? He said that Mendel is a good man. He established a fire station in our village. In other words, they remember him for the fire station. One of the indications that human beings many times remember only the things which touch their life. Very important for scientists to know that the people recognize those which have touched their life. If you have done really something to eradicate hunger, you will always be remembered. But it was a good experience. People from different parts of the world had come. So I am happy that you are now celebrating the 150th anniversary of Gregor Mendel. Mendel did not know at that time of Darwin. If Darwin's work was known, some improvements would have been made even at that time. But Darwin also did not know about Mendel when he published his work. Mendel, after Mendel, you know very well, you are all students of genetics, I need hardly mention. After the rediscovery of Mendel's laws in 1900, one by one, very quickly, every decade has very important developments in genetics. The first decade, Muller and Stadler, mutations were born. First was ionizing radiations. Later on, during World War II, Auerbach and Rappaport started chemical mutagens like methane sulfate, all ethyl methane sulfate and so on. But they were important. So you had both radiations and chemical mutagens. Meanwhile, we didn't know the constitution of the substance of heredity, namely the DNA. That is where Watson, Crick, and Maurice Wilkins, these three were given Nobel Prize, but there is also a very important Franklin, the lady who did the X-ray diffraction. In fact, she did some of the most important work. Since I had taken her course, I know what she did. It was a remarkable thing, but when the Nobel Foundation was asked why did you omit her from, it should have been really 4, not 3, they said that she had just died, she was no more. Apparently, according to the Nobel Peace Prize, the person should be living and coming to. But that does not mean that we should forget her original contribution, because the whole X-ray diffraction data was provided by her. Well, before our independence, the most significant event was the Bengal famine, and the undivided Bengal, where several million people died out of hunger. This is from the cover of the Hindu paper of that time. Hindu is a very old journal, and inequity, Dr. Amartya Sen said the reason for the Bengal famine was not due to lack of food, although Myanmar, which was a major supplier of rice to India, it had been occupied by Japan. It's World War II, and the supply lines were cut. But nevertheless, Amartya Sen feels there was enough food, but there was not proper distribution, equitable distribution. People who had money started cornering the food. That always happens whenever there's a famine. Those who are well-to-do buy more and keep it and so on. But gradually this it's been changing. So from Bengal famine to right to food, because our legislation of 2013 provides a legal right to food, which you know very well, and it had a number of important clauses. The most important ones are when you deal with nutrition, you should take a life cycle approach from birth to death, from the child which is born to her old age. The nutritional requirements are somewhat different. Secondly, the law recognizes the critical role of women both in agriculture and in household food security, so that the entitlements under the Act are given, the passbook, whatever you want, will be given to the senior most woman in the family. The third very important element is the enlargement of the food basket. I told you how gradually, over a long period of time, the food basket has been shrinking. Today there are 20 or 25 crops of commercial value all over the world, soybean and so on. But we try to include the PDS and also in the public procurement, minimum support price, MSP. For example, one of the problems of pulses and so on is due to the lack of market price, and even if government announces a price, there must be a mechanism to implement it. And that's why the enlargement of the food basket by including Nutri-Millets and other orphan crops in the public distribution system. The other day at the Agrobiodiversity Conference, it was decided to recommend that 1 year of this decade should be Agrobiodiversity year with special reference to millets. This year is the year of pulses. Last year was the year of family farming. Before that was quinoa. Every year designated for some purpose. The entitlement, which you know very well, for each one 5 kilograms. One of the important facts emphasized by the Royal Commission, called Lithgow Commission, was the need for research, science. That is why I congratulate DuPont and Pioneer for your emphasis on the latest in science. Today's symposium topic itself is showing what is happening today. I will come to it in a moment, and where do we go. What Lithgow said was, however efficient the organization which is built up for demonstration and propaganda be, unless that organization is based on the solid foundation provided by research, it will be merely a house built on sand. Very strong statement of the need for supporting science and research. This led to the birth of the Indian Council of Agricultural Research in 1928. One of the recommendations of the Royal Commission was to increase the opportunities for science, and the government set up the ICAR, the birth of ICAR. Already Dr. Jain has mentioned the history of IARI, its birth in Pusa, Bihar, and its shift to Delhi as a result of the earthquake. And among the directors who has played a very major role in terms of particularly genetics, breeding, and also what we are now going to discuss, the new genomics and so on, a whole series of new developments. Dr. Paul, when I was here, instituted the Center for Advanced Genetics in the Botany Division. That was a very important one. That helped us to enlarge our work to cover a whole series of other basic genetics, apart from applied genetics and breeding, basic genetics and breeding. This is a stamp which was issued by the Government of India on his birth centenary. A 5 rupee stamp is available showing the two crops in which he worked most, wheat and roses. The roses which you find here are all products. His stamp is there. I mentioned earlier one of the first seminars we had in this hall was in relation to prophets of doom. One of them was Paul and William Paddock, and he said that there will be extensive famine in 1974. The reason is the demand-supply gap will be very high. The demand will be very much, supply will be less, and it will lead to famines. Ehrlich went even further. He is a very famous man in the population area. Sometime between 1970 and 1985, the world will undergo vast famines. Hundreds of millions of people are going to starve to death. That is, they will starve to death unless plague, thermonuclear war, or some other agent kills them first. The United States should announce that it will no longer ship food to countries such as India, where dispassionate analysis indicates that the imbalance between food and population is hopeless. This was their analysis. A very respectable person, the population bomb, that India probably will not be able to feed itself. But the government responded in very, very many ways. IADP was the first one, launched in 1960-61. This was for maximizing the benefits of irrigation water and mineral fertilizer. It was clear that unless plant was also fed with more nutrients and with more water, the yield productivity will not go up. That is why we had a low productivity for a long time. Unfortunately, the impact on yield improvement was not up to expectation. This was also called a package program, package of improved practices. But that package had one important missing element, namely a genetic strain which can respond to the rest of the package. A genetic strain which can respond to fertilizer and to water was missing in that package. These were Jawaharlal Nehru, who was fond of large dams. He went to the United States and saw very large dams and structures. We started the Bakra Nangal Dam, multi-purpose irrigation projects, fertilizer companies, particularly the two in the cooperative sector. In the cooperative sector, the Indian Farmers Fertilizer Cooperative, IFCO and CRIPCO, they are some of the leading ones. They also provide a lot of support to farmers in the form of knowledge, in the form of training and so on. Being Indian, the basic principle is Indian soils are hungry and thirsty. You must overcome the soil hunger, you overcome soil thirst if you really want to increase productivity. And now, I said earlier that the IADP, the Intensive Agriculture District Program, lacked one very important ingredient, namely a genetic strain which can respond to the rest of the package. That was provided in the 60s and 60s really, late 50s and 60s, both in wheat and rice, to the semi-dwarf varieties. Their history is well known, I'm not going to repeat to you, but these varieties were characterized by 2 or 3 important characteristics, genetic characteristics. One is they were photoinsensitive. Secondly, they had a very high harvest index. Harvest index means translocation of photosynthase to grain formation. They were characterized by high harvest index. They were harvest. They could also not lodge because of the straw, semi-straw. So these 3 or 4 important characters: photoinsensitivity, high harvest index, and the ability to utilize more nitrogen because of the plant architecture. At that time, in the 60s, all of us became plant architects really. We used to discuss what kind of plant architecture we should have in order to enable that plant to utilize water and nutrients more efficiently. So plant architecture even today, in fact, people like Dr. Asana, who was the head of the physiology division here, very eminent physiologist, he mentioned that we should have a single tiller plant and more population per unit per square meter, 200 to 300 panicles per square meter. So he suggested a number of changes in the plant architecture. The young people, I would like you to keep it in mind that 3 principles which went into the plant architecture, the harvest index, and so on, which you will have to. Science and technology has always progressed fast. Ever since the rediscovery of Mendel's laws of inheritance, there was the mutation by Muller and Stadler, which won them the Nobel Prize, induced mutation. Then there was a polyploidy, colchicine, and aneuploidy, the various kinds of chromosome combinations. So you had continuous progress in genetics. The early days of Mendelian genetics were followed by basic and enlarging the base of the theory and its application. Then came the Green Revolution, which because many people use many definitions, but Green Revolution really means through the green plants, through photosynthesis, you increase the productivity of the plant and yield per hectare, not the area. The Gene Revolution Green Revolution gave way to Gene Revolution following the discovery of the double helix structure of the DNA, and also a whole series of developments which you are going to discuss here. We have to produce, according to government, 100 million tons of wheat minimum by 2020. That means an average yield of 4 tons per hectare from 25 million hectares. There is a large untapped yield reservoir in eastern, central, and western India. At the time when we started the so-called the Green Revolution kind of research, there was a large untapped yield reservoir in Punjab, Haryana, western UP. That has now been tapped to some extent, and still there is some to a large extent. But the fact remains that the whole of eastern India, eastern UP, West Bengal, Pachim Bangla, Assam, Orissa, there you have. When you develop a strategy for dealing with genetic material or genotypes, there are 3 principles. One is defend the gains already made. This means you will have to work hard on pests and diseases, particularly in the case of wheat, all the 3 rusts, the stem rust, the leaf rust, and the stripe rust. The other day in the agrobiodiversity conference, there was a feeling that still the stem rust problem, I myself mentioned it, with 1 to 2 degrees higher temperature, minimum temperature, and today's papers say the new President of the United States may not very much honor the Paris Climate Convention and so on, but that depends. I hope he will change his view. But as things stand today, minimum 2 degrees centigrade more will happen by the end of the century. That will create much more problem when you have a higher temperature. Then the stem rust in particular, which is more important in southern India today, stem rust will become more important in northern India also. Apart from leaf rust and stripe rust, you will have also the problem of stem rust. So defending the gains means largely working on disease resistance, disease and pest resistance. Extend the gains to new areas. I have already mentioned about eastern India, and make new gains. That's what you are going to do. The aim of this, Raman and Babu, I think, is to make new gains, to make new. Well, many of you have seen here, some of you have not, Dr. Norman Borlaug, who was a remarkable man who gave us the first semi-dwarf varieties, which started our dwarf wheat breeding program. And next to him was Glenn Anderson. Glenn Anderson was a Canadian scientist, a very excellent scientist. He died rather young. And next to him is H.K. Jain, a very famous geneticist who used to be director of IARI. Our main aim while developing the new varieties of wheat, high-yielding varieties, we knew conditions which are favorable to the crop are also conditions which are capable to the pathogen. Same conditions are favorable to the pathogen. Therefore, if you want to improve crop yield, you have to handle the pathogens, pest and pathogen. And that is why we had started a program of a genetic checkmating of pests, studied the Puccinia path and deployment of leaf rust resistant genes over North India. You checkmate, this is the problem, this is the race, this is the resistance gene, map it together so that you have a genetic checkmating of diseases. Innovations in the 1960s, synergy between technology and public policy. We had excellent political support, national demonstrations started by this institute first, National Tonnage Club of Farmers. We organized the farmers on the basis of their capacity to produce yield. I remember that meeting, very funny meeting. We had a large number of farmers who were discussing about what should be the minimum ton they should produce for eligibility to become a member. And one said 5 quintals, 2 quintals and so on. Finally they agreed upon 1 ton. 1 ton. You should produce minimum 1 ton to become eligible to become a member of the tonnage club. So the concept of yield has changed enormously. I'm trying to say among farmers themselves. Today it's changed. The high yielding seed villages, Jonti village, were famous at that time. Even today I find media go there and find out what happened and so on. The high yielding varieties program, the minimum support price, public procurement and public distribution. These are important elements. The most important was the farmer. The farmer is the one. It is, after all, we can provide technology, but it's only the farmers, very hard-working farmers, synergy between technology, services, public policies and farmers' enthusiasm. This is what we call Green Revolution Symphony. The 4 most important components of the symphony was technology, services, public policy. Services include seed production, seed distribution and so on. Public policies and farmers' enthusiasm. Indian farmers achieved as much progress in wheat production in 4 years as during the preceding 1,000 years. That was a very great achievement of the Indian farmers. As I said, political support. Prime Minister Shastri himself was a very farmer-centric. That is why he gave the slogan, Jai Jawan, Jai Kisan. The Kisan and the Jawan being the 2 legs of independent India, who will take us forward. And it was about, I think, midday in the month of end March or April, I forget it now. Very hot day, but he just went to the field and went through the material. Sorry. The other 2 in those days of the Green Revolution, who were very important political leaders apart from Shastri, was Indira Gandhi and C. Subramaniam, who were known to you. What I am trying to say is, why are the Americans very concerned today? Because Mr. Trump had made many statements, including he will scrap the Paris Agreement on climate. People are afraid, unless political support is there, whatever scientists can do. They can do something, but not their work can be enlarged, impact of the work, if political support is there. Even at that time, I had warned that if you overexploit your natural resources, the Anthropogene era, overexploit your natural resources of soil, of water, of biodiversity, you will have great difficulties. You will have great difficulties in terms of sustainable agriculture. How do you sustain agriculture? This particular sentence from my address at the Science Congress in 1968 at Varanasi Science Congress, I made this statement warning that don't overexploit natural resources, you will have difficulty. So I coined the term evergreen revolution. In other words, green revolution was somewhat criticized by environmentalists. I said our green revolution increase in productivity in perpetuity. In perpetuity, that's very important, and that's what the evergreen revolution is. This book, published by the Academic Foundation, provides all the techniques which can be utilized for the purpose of improving productivity in perpetuity without ecological harm. Mention was made by Dr. Jain about IARI becoming a university. It was the first deemed university, along with the Indian Institute of Science. After the UGC Act of 1956, the very first two deemed universities were IARI and the Indian Institute of Science. Dr. C.D. Deshmukh, who also established the India International Center, he referred in his speech that these two institutions, IARI and Indian Institute of Science, should be referral points, should be the bottom line in terms of how to compare, how to enlarge the deemed universities. Of course, today we have hundreds of deemed universities in the country, but in those days, the deemed university was to also not sacrifice quality. I mentioned earlier also the South Asian enigma, extraordinary economic growth, population largely dependent on agriculture, yet 2 out of 5 children are stunted. Women and children suffer from under and malnutrition. If you want to attack hunger in your own scientific work, you have to contribute at 3 levels. One is to calorie production, yield per hectare, higher yield, higher productivity. Then the whole question of protein content, protein deficiency. Because generally, if there's higher protein, there's a calorie penalty. If there's higher calories, there's a protein penalty. So how do you really overcome these two, the two penalties? Because I said higher calories means protein penalty, higher proteins means calorie penalty. But how do you really reconcile? So the protein deficiency, the most important is the micronutrient deficiency. Micronutrient deficiency of various kinds. Today, the micronutrient deficiency like iron, iodine, zinc, vitamin A, vitamin B12 and so on, it's also called hidden hunger. Hidden hunger, and one method which is getting popular today among scientists and geneticists is biofortification. In fact, the latest World Food Prize was awarded to the biofortification work in sweet potato. There are 3 methods of biofortification. Naturally occurring biofortified plants like moringa. Moringa has now become exceedingly important in the nutrition area. There are now so many articles which are coming up. This is from Down to Earth, published from Delhi, Center for Science and Environment. Sweet potato is vitamin A rich, nutri-millets and fruits and vegetables, as well as milk, eggs and other forms of animal. So one is naturally occurring biofortified food material. The second is biofortified varieties selected by breeding and selection. Iron-rich pearl millet. There is one by pearl bajra, iron-rich bajra, which is put out by Nirmal Seeds. The original material came from Icosat. They have improved it and developed a variety. Public-private partnership is a good example in biofortification. Then you can have genetically biofortified crops like what Ingo Potricus produced in terms of vitamin A rich, iron rich, golden rice. Golden rice, Ashoka and his group are working, so I won't go into it. But the problem with golden rice and many others, all genetically modified crops, whether it is BT brinjal, has been a lack of agreement in the country on how safe is safe. The other day Peter Raven criticized us for fears which have not affected the Americans and so on. What fears? Are there genuine fears? Are there only doubts? But anyway, one thing is very clear: we must have regulatory mechanisms which are above board, which people have confidence. When you have a regulatory mechanism in which people are confident. So how do you marry nutrition and agriculture? Very important today. How do you make agriculture and nutrition? I developed a methodology called Farming System for Nutrition, mainstreaming nutrition in the design of the farming system. You can add pulses and so on. Then I would request some of you, including our DuPont pioneer, to put out genetic gardens of biofortified crops. Dr. Jain mentioned about IARI's work in Afghanistan. Apart from in the university campus at Kandahar, one of the important elements will be a 400-acre genetic garden of Afghanistan, because Afghanistan is very rich in all almonds. Many of the material you get in the markets, the dry fruits, they come from Afghanistan. Tagore wrote a beautiful poem, Kabuliwala. The Kabuliwala, what all they bring, that shows you the importance of Kabuliwala in terms of biodiversity. The genetic garden of biofortified plants and crops will contain valuable germplasm of naturally biofortified crops as well as through plant breeding. It will also serve as a center for conservation and nutrition education. For example, when you enter the garden, biofortified garden, one section will be plants which are rich in iron, another section will be plants which are rich in vitamin A, another will be plants which are rich in zinc, so that when you want genetic material for zinc, for iron, for vitamin A, you can know which plant which will give you. That's why it's education. It's an interesting garden, genetic garden. This year, the year of pulses, what we have done is, from my foundation in Chennai, we have persuaded several panchayats in Tamil Nadu to become pulses panchayats. They don't have enough irrigation water. They have to do with rainwater harvesting. Pulses are high value, low water requiring crops. So one of the first pulses panchayat was developed in Tamil Nadu. Then in the case of conservation, the role of women engendering all our programs becomes very important, which are applied programs. These are all tribal people groups, 9 different tribal groups in Odisha, in Koraput. Koraput has been declared by a few as a globally important agricultural heritage site, GIAHS, globally important agricultural heritage site. You see how much they are preserving, not only in cultural diversity, curative diversity, and culinary diversity. This group of people, Kanda, 124 medicinal plants, and they also know for what purpose they are preserving. This is why I call them value-added taxonomy. The tribal taxonomy is value-added taxonomy because they know exactly what they are doing. Last year, the Nobel Peace Prize went to this lady, Dr. Tu from China, for her work on Artemisia. The first time a medicinal plant has been recognized. Cure of a disease through medicinal plants. So it's very important. Let food be thy medicine. Hippocrates' very famous sentence, "Let food be thy medicine," was the background, was the focal theme of the Borlaug Symposium held recently, last month in Des Moines. It's a very interesting papers are presented. Let food be their medicine. In fact, my daughter, Dr. Soumya Swaminathan, she has been saying that food, nutrition is the best vaccine against TB. TB is spreading in the country enormously, and only a drug-based approach won't help. It has to be a food-cum-drug-based approach, which is important. Well, we can take pride in the fact that we have a legislation to protect our plant agrobiodiversity. Agrobiodiversity is fundamental to the work which we are going to discuss here. Unless you have variability, it is very difficult. Of course, we can create some variability by mutation. Ours is the only legislation which confers concurrent rights to breeders and farmers. I mentioned biofortification through breeding, high iron pearl millet. This was developed, and then pioneer hybrids also are very rich in iron. Nirmal seeds has produced. The first genetically modified crop was in Petunia in 1983. by transgenic methods. So we have come a long way from that time onwards. This is why one has to have a real policy in the country. What are we going to do? Are we going to permit genetically modified crops, or there are some restrictions and so on? For example, the Rubber Research Institute, Kottayam, has excellent rubber plants which are very tolerant to temperature, but they have a difficulty in testing it because the policy is not clear. So we require, at that time, we require a policy. I had myself chaired a committee where I suggested that we must, the bottom line of our national agriculture biotechnology policy should be the economic well-being of farm families. Is it good for the farm families or not? That's most important. Food security of the nation. When you evaluate each technology in biotechnology, it has to be done by case. Very generalized principles can be done, but most of it, with the BT brinjal or BT cotton, you have to evaluate them separately. But economic well-being of farm families, food security, health security, biosecurity, these are important, and also trade security. For example, today, if AK Singh produces basmati rice, which is genetically produced, you will have difficulty in exporting it. You can't export it to European Union and so on. So trade security is exceedingly important. There are now new approaches to bypass GM method, recombinant DNA method. One is marker-assisted selection. The Chinese use marker-assisted selection very extensively. In fact, many of their new varieties of rice and so on are developed by marker-assisted selection. You can also use the new CRISPR-Cas9. This has become suddenly very popular among geneticists, the latest technology. In fact, science called it the most important breakthrough of the year, the last year. I think we are going to discuss it here. Gene editing, it was one of my dreams, directed mutagenesis in the 1960s. It has now become true. It has become possible. Beside mutations intended specific gene. Since you are going to discuss this, I shall not go into it, but only to show that all the time science progresses and we should be. So to close the unfinished challenge, ending the alarming state of malnutrition in India, 39% of children are under 5 are stunted, 15% under 5 are wasted, 40% of women of reproductive age are underweight, 48% of women of reproductive age are anemic. So how are you going to, in your case, how are you going to bring together agriculture, nutrition and health? That's a great challenge. No time to relax, avoiding food losses and food waste. 1.3 billion tons, according to FAO, were wasted last year. Climate change, temperature, precipitation and sea level, shrinking per capita land and water resources. We have no option except to produce more and more from less and less land, expanding biotic and abiotic stresses, adverse cost risk return structure of farming. Market volatility is very important, and then above all, reluctance of youth to take to farming. I mentioned about Jonti village. When I went there 2 years ago and talked to the farmers whom I know, only the older people are there. The young people have all left. They all left for various places, for jobs. So how do you, in the National Commission on Farmers, we said, unless agriculture becomes technologically exciting, technologically interesting, and economically rewarding, it will be very difficult to attract educated youth in agriculture. It must be both intellectually satisfying, at the same time economically viable or rewarding. Well, we go back whenever there is a doubt to the father of our nation. Before independence at Novakali, he said, just before, he was there because there was a problem in that area. Novakali is in Bangladesh. There are people in the world so hungry that God cannot appear to them except in the form of bread. God as form of bread. That was, I think it was said in 1946. Now we have come a long way. We have got a Food Security Act, we should be proud of it. But as I said earlier, the Indian enigma, a large number of children, women still going to bed hungry is something which we should not do. And I hope this particular symposium, the participants here, the young people, will be on the vanguard of the fight to achieve this goal. The presence of the God of bread in every home and heart in our country. Thank you very much.
I request the audience to give a standing ovation for an ever-inspiring, enlightening talk by Professor M.S. Swaminathan. Thank you, sir. I request respected Director, IARI Madam Director, Dr. Ravinder Kaur Madam, to kindly present shawl and memento to Professor M.S. Swaminathan, the living legend, as a mark of respect, our love, affection, and regards, please. Thank you, sir. Thank you, madam. I now request Dr. A.K. Singh, Head Division of Genetics, IARI, to present memento to Director, madam. I request Dr. Raman Babu to present memento to respected Dean Dr. R.K. Jain, sir. Thank you, sir. I now request Dr. A.K. Singh, Head Division of Genetics, to present vote of thanks, please.
Most esteemed Professor Swaminathan, Dr. Kaur, Dr. Jain, Raman, distinguished participants, ladies and gentlemen. To hear about father of genetics from the father of Green Revolution, Professor M.S. Swaminathan, has truly been an enlightening experience for all of us, and we will cherish this for all times to come. On behalf of all the students and staff of Division of Genetics and the Indian Agriculture Research Institute, I feel greatly privileged and overwhelmed, but short of words, in expressing our gratitude to Professor Swaminathan for his blessings in commemorating 150th years of Mendel's Laws of Inheritance. As a matter of fact, the very idea of this commemoration was given to me by him when he came last in April for IARI Foundation Day lecture. Thank you very much, sir, for reminding us for commemorating this function. Thank you so much. Sir, we know that you have been extremely busy for last 5 days participating in the first International Biodiversity Congress, and yesterday you traveled to Mumbai early morning, returned Delhi late night to be with us this morning to bless this occasion. And this speaks volumes about your concern to the student community and also to the science of genetics, which you have nurtured for more than 6 decades. I am sure all the participants have thoroughly enjoyed your lecture on food for all in Anthropocene era and will cherish it forever. Your lecture reminds the agricultural scientific community in general and that of geneticists and plant breeders in particular of their important role in meeting the future challenges. I would also like to thank Dr. Ravinder Kaur for her constant support and facilitation in organizing this function. Thanks are also due to Dr. Jain, who has been looking after this program every minor detail on day-to-day basis to make it a success. Our co-organizer and sponsors, Pioneer DuPont India, represented by Raman, who is also an alumnus of IARI, and his team, deserves special thanks for coming up with their Plant Science Symposium series on genomics-based next-generation crop improvement approaches as a befitting tribute in commemorating 150 years of Mendel's Laws of Inheritance. Thanks, Raman. Thanks a lot. Friends, another unique opportunity has been that through this symposium, the scientific community in 73 agricultural universities, all the ICR institutes which are crop-based, and all the seed industry partners through National Seed Association of India, we passed on the information to them to access to IARI website. The entire program is being live webcasted, and the community in the entire country is watching this program live. My request to all the viewership is that please do not go away. We have very exciting scientific program for next days, for tomorrow and for today. Please be with us and watch the program. That will be very, very useful for you. Finally, I would like to thank all our guests, particularly the faculty members who have superannuated from the Division of Genetics. I can see Dr. N.N. Singh, my own teacher, Dr. Balram Sharma, Dr. Sarkar, Dr. V.P. Singh, Professor Tandon, Dr. Gulati, Mrs. Raman, Dr. Chaudhary, Dr. Kharkwal in the audience, and most importantly, I also see Professor Anupam Verma on the other side, sir. I wish you were here in the front for gracing this occasion. This has been a rare moment to get everybody together. My colleagues in the division, Prabhu, Firoz, Tara, Bharadwaj, Yadav, Shailendra, Vignesh, Ranjit, Ramaprasad, and many more. And all the students who have been deeply involved in organizing this function. I have one more person in the audience, Professor B.D. Singh, my teacher, and I request you, sir, to kindly stand up and turn on the back side, because for the community of genetics and plant breeding, Dr. B.D. Singh is a household name through his books on plant breeding, genetics, and biotechnology. When he came here, 4 of the faculty members from Pioneer DuPont, they were so delighted to see him. Now they are occupying the most advanced field in the respective areas, that they could not have done it without reading your book. So thank you for serving this community through your writing. Thank you so much, and thank you for being with us. I thank you all for being here and request you to continue in the same number to see the benefit of the remaining program. Thank you very much.
Thank you, sir. Now we come to the end of inaugural program. I request all the registered participants to kindly assemble in the lawn outside the auditorium for a group photo, and also we'll break for coffee. Thank you. Kindly assemble back after 20 minutes. We'll start the technical sessions. All the students, all the audience are requested to assemble back to start the session. Welcome back. We'll start the technical session 1 on next generation genomics approaches, where we have 4 lectures. Here, the speakers are reminded that there will be 40 minutes time for each speaker. Our first speaker, I feel very privileged to invite Professor B.D. Singh, Emeritus Professor, Banaras Hindu University, Varanasi, to deliver a lead lecture on Gregor Johann Mendel and 5 Dreams of a Plant Breeder. Sir, please.
My esteemed colleagues,
and 10 minutes is the introduction.
Okay.
That is it.
Okay. Esteemed colleagues and young friends, when I think of Gregor Johann Mendel, a slogan comes to my mind, slightly ahead of his time. This year, the world is celebrating the 150th anniversary of Mendel's publication. And on this occasion, Theoretical and Applied Genetics has brought out a special issue. The chief key contribution of Mendel is the emergence of the gene concept, gene being particulate, having alternative forms called alleles, governing the development of a character, and during inheritance, the alleles of a gene remain pure and segregate. The only exceptions known to this are plasma genes and paramutation, and non-alleles are inherited individually. Of course, there have been modifications to some of the things and advances in some of the areas as listed here. Mendel studied 7 genes, 4 of these genes have been isolated, characterized, and the basis of mutation also characterized. The seed shape encodes an enzyme that is starch branching enzyme, and the basis of mutation is insertion of a transposon. The cotyledon color gene encodes a protein that regulates chlorophyll degradation, and the mutation is because of 2 additional amino acids inserted into the gene. The other one encodes a transcription factor, the seed coat color, and the plant height gene encodes an enzyme involved in GA3, GA biosynthesis, gibberellin biosynthesis. And the mutations here are transitions from G to A, one affecting splicing and the other leading to a missense mutation, replacement of an amino acid. 3 of the other genes are less well characterized. In fact, for 2 of them, there are 2 candidate loci, and it's not very clear which one was studied by Mendel. But it is known that, pod color encodes a protein that is possibly involved in chlorophyll accumulation. And then secondary cell wall deposition is involved in pod shape, and flower position involves a signaling protein. The brilliance of Mendel can be seen from his paper. If you study the introduction part of it, you would see that he has incisively analyzed the previous work on the basis of he planned his experiments and worked out a perfect design that has not been modified in any significant way so far. He applied mathematics, but he was brilliant enough to recognize that deviation from 3 by 0.1 or something like that was essentially a random deviation, and the value represented 3, not mathematical difference. In addition, he was lucky. A couple of items that the 7 traits he studied showed single gene control, complete dominance. He was not confused by linkage, although there were opportunities that he could have run into linkage. A couple of genes could show that. And the problem cases that he encountered, they came to him later on. By that time, he had already developed his hypothesis. But anyway, luck cannot be counted for too long, and luck ran out on him. He chose a plant, Phaseolus. Traits selected by him were a mixed bag of qualitative and quantitative traits. And he selected an apomit, Hyraceum. He also worked with honeybee, that I did not list here. But most important of all, he ran into analysis of R.A. Fisher, and criticism from R.A. Fisher included many things, but 2 of the points that impress me or overwhelm me are questioning whether Mendel understood what he wrote, and also suggesting that he had falsified data. These are by other workers who followed Fisher, not by Fisher. By looking at these 2 criticisms, it seems that something more than science is at work here. And the criticisms still linger in the scientific community, despite defense by many, and analytical defense by many of Mendel. 2 of the most recent and most convincing rebuttals are by Fairbanks and colleagues, in form of 2 papers published in 2001 and 2007. Even those who appreciate Mendel somehow are not very fair to him, because they tend to limit Mendelian concepts and principles to oligogenic inheritance by using phrases like these. Most recent example, the volume that was brought out by TAG to commemorate the 150th anniversary has several papers that have this. I selected just one example, random example by Hori et al., which says that ratios 3:1 or 15:1 expected on the basis of Mendel's law, as if inheritance of quantitative characters is by any other law than Mendel's law. To top it all, the misconception about Mendel includes mistranslation of the title of his paper as experiments in plant hybridization, whereas it should be experiments on plant hybrids. As we run with Mendel from discovery to rediscovery to criticism and to the impact, the time frame goes on expanding, and the expand of Mendel continues even today. His research led to the establishment of the field of genetics and the myriads of developments in genetics and related fields, including the molecular plant breeding. Plant breeding, it is classically regarded as a game of numbers because of three basic regions. The genotype is determined by random events. The phenotype has a component of uncertainty because of non-additive gene action and environmental effect. And the mutations that generate all variation we observe in the biological world, they are low frequency random events. Therefore, a plant breeder would love to be able to select on the basis of genotype rather than on the basis of phenotype, and the development of molecular markers satisfies or caters to this need of the plant breeders. When we look at plant markers, all markers are random in their genomic location. Some of them are located in genes, and a small fraction of gene-based markers are functional markers in that the marker allele reflects the allelic function of the target gene. The functional markers are the most informative in terms of the gene you are selecting for and most reliable, but they are also the most demanding to develop. The markers that show association with such genes and QTLs that are significantly associated with the trait, they can be used for marker-assisted selection. But a very ambitious scheme of genomic selection has been proposed to select for all genomic regions that influence the trait in question. This scheme is effective, but it is demanding in terms of the resources as well as the technology. The situation with molecular markers is that in private industry it's routine, their use is routine. In public sector, largely their use is confined to molecular assisted or marker assisted backcrossing. The use of markers increases tremendously the amount of data that has to be handled by a breeder. The speed of decision at which the breeder has to take various decisions, there has to be the best use of molecular markers is when you have a reliable off-season nursery or greenhouse facility, and that involves several rounds of decision by the breeder. Overall, the cost of breeding program is increased by the use of molecular markers, and you need resources like statistics and bioinformatics to be able to use the full, make the full use of molecular markers. Plant breeders generally work with the primary gene pool because it is easily used and very little linkage drag is generally observed if the wild type is closer to the cultivated type, As the distance increases, the linkage drag increases. But plant breeders would like to have no limit on the gene access to the genes. So any gene anywhere should be available to plant breeders. To serve this need, the transgenic technology has been developed that can use DNA sequences from any biological entity, or even synthetic sequences can be used. But the snag with transgenic technology is that there is always a biosafety concern because of DNA sequences that are used, and also because of the site of integration in the genome. Since the sequences that we use, they are known, their products are known, they can be assayed relatively easy, but the assay of the effect of site of insertion of the transgene is really demanding. The biosafety concerns have a dampening effect on the development of transgenic technology and its application, because on an average it requires 5.5 years and $35 million to develop the dossier and get the approval. And at the end of the approval, and this Dr. Pentel would attest to, that even if you have the approval, it doesn't satisfy anyone, let alone the consumers. So it has been suggested that instead of producing transgenics, a section of transgenics should be produced. These are called cisgenics. Cisgenic plants are produced by using DNA sequences from the primary gene pool only in the native state, the gene with its own regulatory sequences. Therefore, the product would be comparable to a backcross product, and therefore the requirement for safety evaluation will be less rigid. They are less rigid. But the problem remains that the problem remains in this case that the DNA sequences are integrated at random sites, and that evaluation has to be done. There is another variation that is suggested in intragenic plants. In this case, all the sequences are from the primary gene pool only, but a gene could be combined with regulatory sequences from other genes. You could use RNAi technology or antisense RNA technology to achieve your end. Their regulatory requirement is more stringent than that of cisgenic plants. As I said, the site of integration of cis genes is random in the genome, therefore biosafety requirements are there. What would be ideal would be to have a cisgenic plant produced by integrating the cis gene into the genome by homologous recombination. In place of the gene it replaces. If that happens, this can be achieved by something like what is referred to as site-directed nuclease technology. If that happens, then you have the cleanest backcross product in form of a cisgenic with homologous recombination integration. To make transgenic plants safer, there is another technology of grafting where it can be used under the situations it can be used. It's well known that small molecules travel through the graft junction into the scion. So if you have RNAi suppressed rootstock and non-transgenic scion, the RNAi signal will travel into the scion and would affect the phenotype of the scion, although the scion will have no DNA sequences from the other than of the species concerned. So it should be more acceptable to consumers than transgenic plants. Move on. As I said, mutations are low frequency random events, and as Professor Swaminathan said, that his dream was to have directed mutagenesis, and what he did not say was at high frequency. There are techniques now available that can achieve this. They are basically of two types. using site-directed nucleases, any one of these four, and then chimeric RNA, DNA, RNA, oligonucleotides that can also induce site-directed mutagenesis. All site-directed nucleases are designed by genetic engineering. They are artificial. Zinc finger nucleases were first to be used, but CRISPR and Cas9 system has emerged as the most versatile and easiest to use and most effective also of the site-directed nucleases. The CRISPR-Cas9 system has two genes. One of them encodes the Cas9 nuclease, and the other gene encodes the CRISPR RNA. And the RNA itself has two segments. This is the CRISPR RNA. This is the guide RNA scaffold. It provides the site where the Cas9 nuclease binds. This portion here is homologous to the site of the target gene where you wish to induce the mutation. So the CRISPR-Cas9 system RNA will pair with this sequence here. It will place the nuclease in the vicinity of the junction here, and there will be a double strand break induced by the nuclease here. This break will be repaired generally by non-homologous end joining, and this will lead to either insertion or mostly insertion or deletion of bases in most cases. So therefore the gene will be inactivated. So this is one sure way of inactivating gene at high frequency. If you provide a donor DNA that has end sequences homologous to the sequences flanking the cut site, then there would be homologous recombination, and this donor DNA will be integrated. And this is the technology that can be used for integrating the cisgene or transgene or intragene at a specified site in the genome, so that you know where you have put the gene, what could be activated, what could not be inactivated. So biosafety concerns may be reduced by this. Move on. Then breeders, those who are working with heterosis breeding, know that development of complementing inbreds that produce excellent hybrids, that is the most demanding task, that it involves production of a very large number of inbreds and testing them in field by making single and double crosses. What has been suggested is a technology of reverse breeding to isolate complementing inbreds with very little effort overall from any outstanding heterozygous plant genotype. What is done in this case, reverse breeding case, is that the recombination is totally suppressed either by chemical treatment or by a suitable transgene in the heterozygous plant. Then double haploids are produced from this plant, and these double haploid plants are genotyped for a small number of markers, ideally as many markers as the N number of the species. So 7 chromosomes, 7 markers to be genotyped, and these markers should be polymorphic for the 2 chromosomes of the hybrid. And on the basis of this, then you can match marker genotypes and find the complementing pairs of inbreds. If you produce let's say 128 inbreds out of this, then they will form 64 complementing pairs, and finding one complementing pair that works for you in hybrid seed production should not be a problem. Finally, the dream that any perennial, any breeder of a perennial crop would tell you that it takes generations of breeders to work with perennial trees to develop a variety. And the dream would be that you should be able to breed the perennial like an annual and then grow the end product like a perennial. This is done by using flowering time genes. A number of them have been isolated. This scheme can be used for back-cross program only. The recurrent parent is transformed with this gene, flowering time gene. The transformant at the transgenic plant would flower in 1 year. It can be crossed with the non-recurrent parent and then with the recurrent parent, the hybrid. And back-cross program could be carried out. At the end of when you have a back-cross product with the desired gene, you can have segregation to remove the transgene so that the back-cross product now turns to perennial, and this can be grown. The status report is that cisgenic and intragenic plants have been produced. They are being tested, should reach commercial scene soon. The directed mutagenesis technologies are in different stages. Products from them are different stages of evaluation. Reverse breeding and grafting, they are in experimental stages. And the early flowering of perennial scheme being used for back-cross breeding of fruit trees like pear, apple, and so on. We don't have any report on the success of this. To summarize this, then, a classical breeder works with the phenotype in the hope that he will change the genotype, and this genotype will lead to a changed phenotype of the product that he develops by phenotypic activities. Modern breeder, on the other hand, tinkers with the genotype in the hope that the modified genotype will produce the desired phenotype. In either of these cases, it is essential that the breeder has a clear understanding of the relationship between phenotype and genotype. And because of this, the disciplines of phenomics, genomics, and envirotyping are being vigorously pursued, and it is hoped that one day you would have all the information that would allow you to predict any of these three terms, provided you specify the value of the remaining two terms. If and when that happens, plant breeding will become a very precise activity indeed. Thank you very much. Questions, comments? Would you like to make an announcement, please?
Sir, all the students of agriculture are very familiar with the textbooks on genetics, plant breeding, biotechnology written by you. And today, listening to you lecture directly is a great privilege indeed. Thank you so much, sir, for the wonderful lecture. Now I would like to invite questions from the students. Kindly participate. You would have got many doubts while going through the text. Sir.
Yes, yes.
Sir, since you have crossed 150 years of my law, because I am also a teacher, sometimes I used to teach this laws. I ask that question to the student. How many Mendel's laws are there? Quite often we get the correct answer, they say 2. But there are still students. Deepak Pental sir is here, students even from Delhi University sir, there also says that there are 3. So 150 years cross, we could not say they make the difference that which is the correct answer. Thank you, sir.
You should trust a teacher to ask a teacher. If you ask a question about Mendel laws of inheritance, of course, two. If you ask question of Mendel's laws concerning transmission of characters from parent to progeny or correspondence of characters between parents and progeny, then three. Because the third one, law of dominance, that relates to character development, not transmission of the determining entity. But as I said, and I would like to say it again, that most of us, when we talk of Mendel, we think of qualitative inheritance only. I think that's a great injustice to Mendel. Mendel described what genes are and how they are transmitted, how they behave during inheritance. And I know of, I do not know of any character, quantitative or qualitative, that is produced without transmission of the genes governing it according to the laws of segregation. There are only two exceptions to my knowledge, plasma genes and paramutation, that defies the law of segregation. So Mendel not only was criticized by R.A. Fisher, he is also played down by everyone who teaches Mendel, almost everyone who teaches Mendel. Even the, as I said, even the TAG issue that commemorates his paper, 150th anniversary, has phrases that play down contribution of Mendel. This may be a little too much to say, but if the Mendelian principle of transmission was not clearly understood, molecular developments would never have taken place. Yes, please. Sir.
You show that one slide before the last one. Is it still on or?
Would you turn on the slides?
Last one before, yeah, this one. You say in both classical and modern, a clear understanding of correspondence between G and P is essential. I would argue that for modern breeder it is much more critical than it was for classical breeding, because a lot of classical breeding has been carried out without knowing the inheritance pattern of the trait which one was looking for. So it's absolutely vital for the modern breeder to make sure that genotype reflects the phenotype properly, otherwise you will be going on a wild goose chase.
Nothing could be said better than what you said, Dr. Pentel. Before Mendel's laws were known to the plant breeders, they were producing improved varieties without realizing that they were modifying phenotype, leading to genetic modification. But today, in this context, when you do this, at the back of mind you have that you are changing the genotype and this genotype will change the phenotype. And why I say that for classical breeder also it is important to realize the relationship between genotype and phenotype is this, that sometimes when you have a variety tested in a particular environmental setup, many of us get the impression that this is going to produce the same next year, year after next, and so on, because the genotype is the same. We don't realize that the environment will change. So that realization has to be, and for the students, I thought I should bring it home, that it is important for you to realize also that genotype. But as you said, it's more critical for modern plant breeder to keep that in mind, that phenotype does not necessarily result from only genotype.
Students, if I dare may say, don't appreciate or don't like to do that part of the work because it is backbreaking and you have to do it all phenotyping in the field.
Yes.
This point needs to be emphasized that if you don't relate phenotype to phenotype, then genotypic breeding will not be accurate. As you are teacher to many of the students, I
so the if a modern breeder does not work with the phenotype at the end of the day, he will have very nice publications, sir, but no product in the field. And that is not what the modern breeding should be for. One acknowledgement, like at the end of every paper, one says this contribution is because of this. This acknowledgement to my young students, this is about me. There was in the morning a very nice remark by A.K. Singh. Many of you also said many good things to me, but let me acknowledge this, that what I have been able to do, what I am today, is largely because of this, my mentors, my professional colleagues, and more particularly my students, which includes Ashok. They have contributed greatly to many things that I have done, and they deserve all the appreciation. And you young students are the source of inspiration, and often source of inspiration is the key to anything, so you deserve everything. That the light that you shine on me should be reflected to you totally. Thank you very much.
Yes, sir. Sir.
Please.
Just a concern. Sir, nowadays we talk BT crops, BT brinjal, BT mustard, whatever it is. On one hand is conventional breeding, on other hand is elite breeding or molecular breeding. मेरा कंसर्न सर ये है जिस तरह से अभी सोसाइटी में रिसर्च में डेवलपमेंट हो रहा है, देयर इज़ वन लॉबी फॉर फेवरिंग कन्वेंशनल ब्रीडिंग, अदर इज अगेंस्ट द कन्वेंशनल ब्रीडिंग बट प्रो ट्रांस या जेनेटिक मॉडिफाइड ऑर्गेनिज्म या उसका ब्रीडिंग का जो है. आई एम नॉट ए जेनेटिसिस्ट, सो आई मे बी पार्डन. मेरा कंसर्न ये है सर, हैड मेंडल बीन अलाइव, हाउ ही वुड हैव responded to the present development of molecular or BT crops, BT breeding.
Okay. Here, I am asked to be a sort of I don't know what. Soothsayer, yes, a psychologist and a futurist, whatever they are called, to be able to read the thoughts of Mendel that would have occurred to him on 150th anniversary of his paper. I would try to answer like this, that I appreciate Mendel. And for a very long time I have been doing classical genetics and breeding. So if I had discovered those laws, what I would feel today, that I am being outsmarted, I wouldn't feel like R.A. Fisher did, that he was outsmarted by Mendel. I'll feel very happy that I postulated a gene somebody has identified and proved me correct, and I'll feel elated that okay, this is there. Now, for what it is used, it is, I might have two thoughts. Being a priest, I would not like to tinker with the biological entities created by God. That is where possibly my
that would come from religious consideration or ethical consideration, not because of scientific consideration. So I'll feel happy, and I would let the rest of the community decide today who are working. Like I would like them to decide whether use of transgenics is good or bad for their country and for their society and for themselves. For me, sir, that brings me to a safety issue of transgenics. Many of you may not agree with me, but conventional breeding does not guarantee biosafety. There have been conventional varieties developed that have caused human deaths, particularly in celery and potato. Similarly, transgenic breeding cannot guarantee human deaths. It can minimize the risk of human death by, say, biosafety evaluation and stiff regulatory regulation of the evaluation. Conventional varieties are not tested for biosafety. So, transgenics, if they are evaluated on case-by-case basis, approved, then they should be given a fair chance, and they should be safe for environment, human health, etc. Other issues are not so critical in the sense that whether there will be pollen transmission or not to the wild flora. You can determine this simply by this, whether you have wild related species growing that are compatible with your transgenic crop. Ban it if they are growing, don't ban it if they are not. They are simpler. Human health is the other issue that is evaluated. So, for me they are safe. For anybody else, I don't know. That's personal choice. I couldn't get the question. Cis, you are talking about cisgenic plants? Okay, what is the criteria? What I was saying is that the cisgenics do not have any sequences other than tDNA borders except from the primary gene pool. Even the tDNA can be replaced by sequences from plant DNA, like some citrus DNA sequences work as tDNA. So they can be used in place of tDNA, so that you have only plant DNA now, no bacterial DNA, and the gene is from primary gene pool, so safety should be known to you. If the site of integration were by homologous recombination, then the site of integration is known to you. The native gene itself is replaced, and they should be as safe as backcross products, but much cleaner than backcross products. You read any paper on molecular backcross breeding, after even 11, 12 generations of backcrossing, you have substantial amounts of donor DNA, of the donor DNA that is not the target gene, target sequence being transferred. Here you will have only the gene that is being transferred. So that is why you want plant DNA there, because you want to remove the concern about use of tDNA border sequences.
Thank you very much, sir. I am sorry to intervene. With an eminent speaker like Professor B.D. Singh here, we have many doubts in mind. I request all the people who are having doubts to interact with him during the lunch session. So we have a next presentation. Bhuvneshwari, please carry on.
Thank you, sir, for the deliberation. Now we have the second speaker, Dr. Raman Babu. Dr. Raman Babu is an alumnus of IARI and is known for his profound interest in the nutritional improvement of tropical crop. He is associated with developing the first generation of quality protein maize hybrids in India through marker-assisted selection. He worked in the past with ICAR-VPKS, NIAS Japan, and CIMET Mexico. He is currently with DuPont Pioneer as Technology Deployment Lead for the South Asia region. He will be delivering on the talk, Emerging Technology Trends in Plant Breeding, a Needless Sophistication or Research Necessity. So please.
Good afternoon, friends. I realize we are running short of time, so I'll try to be really brief about this. So I titled this little provocatively. I know that for any given issue there are always more than one opinion, right? So there is always there is an opinion which says that yes, technology can bring in resource efficiency. There is also another opinion that there is needless sophistication, and mostly for these technologies or publications, and what is their use in product development, right? So what I am trying to do today is, I mean, we had an excellent presentation from Dr. B.D. Singh where he laid out all the theoretical principles behind these modern tools. So what I am going to do is I'm going to focus exclusively on the application part. So how do we employ these modern cutting edge tools for product delivery? So what kind of products can be developed? How are they developed? Is technology really helping to gain that efficiency? Okay, so just few words about the company that I represent, DuPont, a pioneer, more than 5,000 researchers in 25 countries, and DuPont Pioneer has always been at the forefront of employing science and technology tools. We have world-class infrastructure in the Asia-Pacific region, and you can see the DuPont Knowledge Center, which is very popular, located in Hyderabad, and Dr. Amitabh Mohanty is here, who represents DKC, and they have upstream research capabilities, both in terms of genome editing, transgenics in a variety of crops, and we have multi-crop research center, which is basically a breeding organization where all the product development teams are concentrated, and there is also a corn research center in Bangalore which produces doubled haploids in corn. Okay, so coming back to the topic, technology, right? So not even a single day passes nowadays without using, say, Google or cell phone or online transactions. I think online transactions have never been so important than in the past few days. I'm sure you all would have realized by now. So my father called me yesterday and he was asking me, I mean, he's never into online transactions, so he was asking me, how can I get a net banking password and username? So that was a very big surprise for me. So that's what technology can do if you are left behind. So the same thing is what is happening in the multiple spheres of agricultural research. So how is technology affecting the researchers? Let's say I just put that organized into 4 major spheres, right? One is germplasm. So it's very important what kind of technologies are affecting the way you keep your germplasm. Say for instance, simple pedigree history, documenting good pedigree information, organizing heterotic groups, or even double haploids. You know, the huge number of DH lines that are developed in the breeding programs, they have changed the way germplasm is handled in the breeding program. Similarly, genome, high density genotyping. I'm sure you all must be hearing a lot about this, right? SNP markers, high density genotyping, whole genome resequencing. So all these things have actually caused a lot of tremendous explosion of data, genomic explosion of data, and we are really wondering, you know, what we do with this data. We have more data than actually what we require. So how do we make use of this data in the product development programs? And also the other important accompaniment is phenotyping, right? So you have all sorts of phenotyping techniques which are helping you improve the precision. And similarly, informatics. So the moment you have generated a lot of genotype data, phenotype data, weather data, climate data, how do you handle this data in your breeding program? So you really need sophisticated tools which does real-time analysis of the data, and also it does all of them through in a centralized processing manner. So these are critical infrastructure for any successful breeding program. Okay, so what do you recognize of this picture? Stars in the sky. Okay, so these are nothing but multiple genetic entities in a breeding program are represented by each dot, right? So this is just to emphasize how you should maintain the pedigree relationship in your breeding program so as to gain advantage. So for instance, line 1 in Pioneer breeding program, what is the kind of relationship the line 1 shares with other lines. So it's very, very important to keep this information and document it well so that it can be exploited later. So one is pedigree information. The other important piece with respect to pedigree information is organizing all your germplasm into heterotic groups. I'm sure you would have studied about heterotic groups, right, in your textbooks. So what do you recognize here? So this is something with respect to the temperate germplasm. We have two heterotic groups here largely, SS and NSS, and look at the wonderful relationship, how is it depicted here. So you have heterotic group A and you have heterotic group B here, so that if you pick a line from here and here, you have a higher probability of ending up with a good hybrid, right? So it's very, very fundamental to have this kind of relationship in your breeding program. So in case of temperate material, you have in corn SS stiff stock and non-stiff stock. In case of tropical and subtropical material, you have Tuxpenio and non-Tuxpenio. If you go to Europe, you have flint and dent. So it's very important to identify a heterotic pattern and stick to it. It doesn't matter. Now all the heterotic groups are all man-made heterotic groups. So you can always end up choosing one or the other system, but it doesn't matter. But stick to a heterotic group and then put your focus on developing those heterotic groups further. So once you have developed the heterotic groups, what can you do next? See, this is that thing that I talked about. So you have in the red color represented the stiff stock, in the blue color NSS. So you have to keep your heterotic groups apart, and when you make pedigree breeding crosses, you don't make crosses within these groups. Sorry, you make crosses only within this group when you make pedigree breeding crosses, and you make hybrids across these groups, right? It's very important to have this. Another important piece is, as I told you, documenting the pedigree information and identifying these founder haplotypes. So a line is coming from which of the ancestor line. So once you have these two pieces of information, what you can do is you can develop identity by descent profiles, IBD profiles. So IBD profiles are nothing but, if you see this red color chromosome segment, where is it coming from? It's coming from founder 1. If it is yellow, it's coming from founder 2. It's blue, it's coming from founder 3. So establishing those founder relationships, that is IBD. So if you have done this, then the last thing you can do is you can associate those IBD profiles with your trait effects. So if you are able to do all these things in a systematic manner, your chances of ending up with a good hybrid product is fairly high. So that's what we like to stress here. And another aspect of germplasm is the number of recombinants that we can develop. I'm sure you all would have heard about a saying, breeding is all about numbers game. So the more number of inbred lines you create, the more number of genetic entities that you create, better successful you will be, right? So that's what this funnel describes. So you create large number of recombinants, and then you pass them through genotypic and phenotypic filters, and you end up with a commercial hybrid. But can you create unlimited number of recombinants? No, because the resources are limited. Theoretically, yes, you can create unlimited number of recombinants, but then the resources are limited, so you have to put a full stop somewhere. So that is exactly this picture says. And can you guys tell me what technology in corn has enabled creating large number of recombinants? Any idea? Just make a guess. Have you heard about double haploids? Right. So it's nothing but one way of deriving homozygous lines within a short period of time using a special type of genotype called inducers. So this is really revolutionizing the way maize breeding is done. So I think that's where, as I mentioned earlier, Professor Sarkar's work on haploidy, on stock 6 cultures is fundamental and phenomenal, and the way it is being exploited now by all the commercial breeding programs is something unprecedented. So this is the technology that I was talking about. So how can you derive inbred lines in a breeding program? You generally make crosses between two parents, and you pass the progenies through multiple filial generations to fix the line, right? So it takes 7 to 8 generations to fix a line. So with this new technology, what's happening is there is a special type of genotype called inducer genotype. You cross your source population with that inducer. So what it does is it participates in the pollination, but then it doesn't contribute the genome to the progenies, right? So all the progenies are derived solely from the female parent. So the female gametes leading to individual plants, and then haploids are identified and they are doubled and you get DH lines. So the important advantage with this technique, we have a talk exclusively on DH lines later from Dindo. He will be explaining to you later on this. But this has really revolutionized the breeding program. The annual output of some of the breeding programs can be as big as millions of genetic entities per year. So that is exactly why, after creating these many DH lines, what do we do then? Can you take all of them and evaluate in the field? It's very expensive. Phenotypic resources are now more expensive than the genotypic resources. So that is exactly why we are talking about genomics now. So you have created large number of recombinants. You don't have resources to evaluate all of them in the field. So then what can you do? So that's where genomics is coming into picture. So genomics, when I was a student way back in 97, 2000 IARI, these three, RFLP I have never worked, at least RAPD and SSRs were very popular, right, around that time. So the marker data points were in thousands. So we used to, a PhD student used to work for almost like 2 years, and we will develop say 3000 or 4000 marker data points running all the gels. But now, after the onset of SNP markers and full genome resequencing, the marker data points are actually running into many thousands, millions, and even billions, right? So there is tremendous explosion of genomic information, and what it has done is that it has dramatically reduced the cost per data point. And I'm sure this rat race is not going to end here. So we are already talking about epigenome, methylome, proteome, metabolome, all these things, it will keep continuing. But what is important is we have the ability to generate large number of genotypic data points, and that is coming at a cheaper cost. Now the question is, how do we make sense of this voluminous data? How do we make it easy for the plant breeder to select upon this data? So if you ask me how is it all being used currently in the industry or in some of the major public sector breeding programs, the high density genotypes as well as high precision phenotypes, they are all combined together using powerful statistics, high efficiency computing, and this all gets channelized into two things. One is mapping, where you can identify specific chromosomal segments associated with a trait, and this is done mostly for simple traits which do not interact much with the environment, and there are only one or two, one locus or two loci controlling the trait of interest. So if you put in millions of genotypic data and a good quality phenotype data, genome-wide association analysis can identify specifically the gene responsible for the trait, and that can be used in the breeding program. So that's one way, right? What is the other way of doing it? So you had generated a lot of genotypic data points, but then you could not identify a large effect locus. So then how is that information being used? Or say for instance, there are traits which are highly quantitative. For instance, whatever traits which breeders are interested, grain yield under stress, drought stress or low nitrogen stress, they are all highly quantitative. You cannot identify one or two markers for such traits. So how do you use the marker information then? So there is a way to use the marker information even in that situation. That is called genomic selection or whole genome prediction. So this is nothing but you are trying to predict the performance of the untested line. So you have created a line, but you didn't have resources to evaluate it in the field, but then you could genotype it because genotyping is cheap. So now I am genotyping the line and I am trying to predict the performance of that line in the field. So that's very much possible, and it's being done. So what are the examples? What are the examples that these marker technology can result in products, right? So some of the successful examples that I was working with Simit earlier, and we identified a gene through genome-wide association analysis for provitamin A content, right? I'm sure tomorrow Firoz will be talking to you more about this, and a lot of advances have been made after what was done on the QPM research earlier. So what it has done is, the average pro levels used to be 1 to 3 ppm, but currently it can be driven to 15 to 20 ppm just with one gene. So 10 to 15 fold improvement in the trait just using one gene. So that's one of the very successful examples, and such products are now deployed in Zambia by Simit. And similarly, another important product is quality protein maize, and I had the opportunity to work during my PhD as well as during my ICAR days when I was working at VPKS with Dr. H.S. Gupta. So we developed this first generation quality protein hybrids called Vivek UPM9, and we transferred this opaque 2 gene by obtaining germplasm from CIMMYT. So this is again one of the stellar examples of how specific marker-based applications can be used in product development. Okay, so you can always ask, whenever I present that, people ask me, okay, the nutritional traits are simple traits, right? So you can always identify 1 or 2 loci and then you can go after it. But what about complex traits which breeders are interested? Say for instance, if I talk about nutritional traits in the private sector, there is no big takers, right? Because these are traits which are sort of invisible traits. So a farmer doesn't appreciate the value of a nutritional trait unless there is a premium price guaranteed for him. So what about yield? Highly complex traits. How can we make use of marker information for such traits? So that is exactly when we are talking about whole genome prediction. So this is like, not understanding the underlying genetics of a trait, but still making use of the molecular marker information to make genetic gains on that particular trait. This is like asking, if you don't understand the entire process of digestion, how can you eat food? You can eat, right? You don't have to understand the entire process of digestion to be able to eat food. That is exactly what is there. So breeders have never understood in the past or even now the complete genetics of grain yield, but that does not stop the breeder from making improvement for yield gains, right? So this is very close to the breeding concept. So wherein you genotype and phenotype a population which is used as a training population, and then the selection populations are just genotyped and their value is predicted. So that's what is all about genomic predictions. So in case if genome-based predictions are completely implemented, how can this redefine plant breeding? So I think I have given just one simple example, especially students, please go through this and just think about it. I'm sure you may not understand now, but if you start reading more about it, you will realize it. So for instance, in the past, if you were evaluating 10 entries in 5 locations, this is how you lay out the trial, right? You have 5 locations, so the 10 entries are replicated in all these locations. So same set of 10 lines in 5 locations, right? That's how we used to do multi-location trials. But now with the advent of genome selection, assuming that you have done all the research work to make the genomic selection possible in your breeding program, you have to do certain things. And if you have done all that, and if you assume that your genomic selection programs are operational, now the situation can change. With the same amount of resources, you can evaluate 50 entries in place of 10 entries, not by replicating them. So you put one first set of 10 lines in location 1, second set of 10 lines in location 2, yet another third set of lines in location 3. So now you can ask me, you are not replicating the entries, how can we understand the G by E effects, right? So if you consider what is the unit of selection here, you are selecting the lines as such, right? So there is another way of looking at it. Instead of selecting the lines, can you break a line into multiple chromosomal segments, and can you evaluate the chromosomal segments and not the lines, right? So you can regress the phenotype onto multiple chromosomal segments, and then you can identify marker effects for a chromosomal segments, and then for a given individual, the genomic estimated breeding value is nothing but the summation of their marker effects. So what I'm saying is, without replicating a line, but by replicating a chromosomal segment, you should be able to get the same answer like how we were getting this one. So basically here you were evaluating 10, but here you are evaluating 50. But for this to happen, you have to do certain things to establish good training population. You have to understand your breeding germplasm well, and this is all being done. And genomic prediction is used widely in the context of DH line selection as well as hybrid evaluation. So gone are those days where we used to make all possible hybrid combinations. So now you can virtually create a lot of hybrid combinations, evaluate them in silico, and make only good hybrid combinations using molecular markers. So I think this technology has tremendously progressed, and I'm sure if you read more about this, you will stand to gain. Okay, so the third aspect is about the phenotyping, right? I think just along with genotyping, the phenotyping has also been making a lot of advances, right? And phenotyping has in fact come a long way from crude balances. Now we are talking about very automated planters, huge mobile wind machines, as well as some of the combines, right? So these things are really revolutionizing the way agriculture is done. Not even agriculture, agricultural research is being carried out. See, one simple thing. How many of you have done QTL mapping or genome-wide association analysis in your PhD or master's thesis? Show of hands. No? Okay, I thought this is the most popular research topic nowadays. So, I was trying to impress upon you that before you do any modern analysis, identifying genetic factors behind a trait, the first thing we need to do is, have we conducted the trial in a good manner? So, are there variations in the field, unexpected noise kind of variations in the field that we need to correct before we proceed to our mainstream analysis? So, this is really, really important. As you can see here, there is one row here because of some problem with the drip tape overwatering, and that has resulted in stunted plant. So, if you have not visited the trial and if you are only looking at the data, you will consider this as a low yielding line, but it has no problem with the genetic potential. It is just the problem of that particular, the error with the drip tape. Similarly, there are many occasions in which you can encounter such variations, and it's very, very important to understand what these variations are. And there are very, very sophisticated tools nowadays, modern tools, statistical tools, to correct all these spatial analysis, spatial variations that we see. See, for example, you see here there is no spatial analysis done here. Signal and noise is confounded. You can't see clearly the signals. Whereas once you do a spatial analysis, the signal becomes much clearer than noise. So it's very, very important that spatial analysis to be done. What tool can do spatial analysis? Any of you have any experience? You heard about ASRML? I think this is one of the important tools that can perform mixed model analysis. Okay, so some of the other technologies. So this is one technology which is called Boreas. This is like a huge mobile wind machine. It can actually blow air at very high velocity to study the lodging tolerance of a corn plant. So lodging tolerance, stock lodging, is a business critical trait. Without that, no farmer is accepting these genotypes. So this can be blown on the plants, and you can select plants that are tolerant to lodging. So it's one of the very proprietary technology employed by Pioneer. Similarly, there is another trait called ear photometry, multi-dimensional imaging. We have Justin Gerke here, who is an expert. If you have any questions, he can answer all those questions. So it's nothing but multi-dimensional imaging of those corn ears, and so that you can very precisely select upon. You can clearly distinguish a susceptible line from tolerant line. Similarly, there is drone-based phenotyping. So drones are used now from pizza delivery to phenotyping, right? So everywhere we are talking about drones. So drone can be used to measure some of the key agronomic traits all over the field in a very high throughput manner as well as in a precise manner. And then another important phenotyping thing is a visual grain quality analysis, which is again proprietary to Pioneer, where single kernels can be imaged, and you can identify the opaque ones from translucent ones in a very high throughput in an automated manner. This is also another proprietary technology which results in market differentiating products for Pioneer. So I just want to impress upon you that phenotyping is still the king. So though you may be tempted with all the genotype information, the technologies, the phenotyping is still the king. I am not saying that you should do more and more phenotyping, do less and less phenotyping, but do with more and more precision. So that is exactly the key. And the fourth sphere that I talked about is information revolution. So you need good tools to be able to process all this information, the genotype data, phenotype data, and then throw all this visualization so that breeders can select upon. So the way it is handled is all this is centrally handled in a high performance cluster. The moment breeder takes the data from field, it all gets uploaded in a central server, all gets processed, breeding values come out, and then whoever is sitting in Hyderabad, in any other country, regionally, they all have that processed data they can select upon. So I think this is another very, very important component in a successful breeding program. And two simple examples of all these technologies being used in products. So I don't know if you have heard about this line of products from Pioneer called Aquamax. So this is a water use efficient maize hybrids. A series of maize hybrids have been developed through whole genome prediction, developed through managed stress screening, and also precision phenotyping. So all this have fed into developing a product called Aquamax line of products, which is designed to produce more crop per drop, efficient root system, better stay green, as well as it has reproductive resilience. So this is one of the very successful examples of DuPont Pioneer applying cutting edge technology. It's very popular in the United States, and now if you ask Anand, who is here, who is a research director for Corn, in Asia Pacific, this is currently the focus, the Aquamax kind of products, drought tolerant products. Another very successful example, which Amitabh will later talk about it, I purposely ignored the genome editing in the genome component because we have Amitabh who will be talking about it. So very recently Pioneer developed genome edited corn, Vaxicorn. So this has been accepted by USDA as a native trait product. So USDA decided that it will not be subjected to any regulatory approval. So we do not know what happens in India when such products come, but then this is a very, very positive development, and genome editing has already resulted in products. So just a concluding thought, technology is definitely, as I told you in the beginning, an undeniable and indispensable component in our lives. And most technologies are actually scale neutral. It is not that only big breeding programs can make use of technology. So it is just a question of organizing your breeding program in such a way that you can take advantage of the modern tools and weigh the merits and demerits of each technology. And for all the students, I would suggest reading this paper. I think it is the same paper that Dr. B.D. Singh showed it. So it is from Rex Bernardo, University of Minnesota. He wrote that bandwagons I too aware of, right? Something like that. So I think it's an excellent paper, and what it says is don't be intimidated by technology. Of course, there are going to be number of technologies, number of bandwagons, but some of those bandwagons may become tomorrow's mainstream technology. So don't be intimidated by the technology components. Objectively evaluate the merit and decide on which technology to adopt and which technology to reject. So I am very sincerely thankful to all my colleagues from DuPont Pioneer. So we have Dr. Anand here, Dr. Mahala is a very experienced breeder from Pearl Millet. We have Dr. Amitabh, we have Shreshti, we have Dr. Emerson, Dr. Justin, Dr. Dindo. So we have a big team here. I would encourage all of you to interact with all our directors and breeders and molecular breeding scientists. And I'll be really happy if we can make use of this opportunity. And I'm also thankful to my colleagues from CIMMYT, ex-colleagues from CIMMYT as well as ICAR. So thank you very much.
Thank you, sir, for the wonderful deliberation, and that too very timely deliberation on different emerging technology trends in plant breeding. To keep up the timings, I request the audience to ask very crisp questions in 10 minutes so that we will not be late for lunch. Thank you.
Sir, please.
Excuse me, sir.
Can a mic be given? Excuse me. Yeah, students have the first opportunity to ask questions. That's what Dr. Deepak said.
Hello, sir. Yeah, my name is Navdeep. Sir, you have explained this whole technology on corn, but still now on wheat, these work like for this quantitative trades for drought is not. We have not seen so much application in wheat. Why it is so? Because of the large genome size, or?
Well, I think different set of technologies are being employed in different crops depending on whether it's a self-pollinated crop, whether it's a cross-pollinated crop, whether hybrids are the final products or whether the varieties are the final products. So accordingly, there are technologies which are being employed. So for even in wheat, I don't have first-hand experience, but I used to work for CIMMYT in the global maize program. I used to hear from the global wheat program colleagues. Genomic selection is in fact a mainstream breeding tool currently. But thousands of genotypes are being evaluated used to be evaluated in the field, whereas now with the whole genome information, you can just evaluate 1/10 of what you evaluate in the field, and still you will be able to make a good prediction of the rest. So genomic selection is one of the mainstream breeding tools, even in wheat.
But as you said that final product, we haven't yet heard about the as such varieties or even GMOs we are talking about in wheat, especially in case of wheat. Means, is these technologies are still in the nascent phase in terms of particularly in case of GMOs? Is it working or I just want to know the scenario of genetically modified in case of wheat, whether DuPont is working on wheat or not.
No, wheat, currently we are not working on wheat, but we have a small research program in the United States. Maybe I'll ask Anand to comment if further.
I think, as Babu pointed out, every crop has its own small nuisance and advantages and disadvantages. So I think given that maize was mostly a hybrid crop, you could strive the two stiff stalk and non-stiff stalk. A lot of this technology that was driven helped the genetic gain to progress at a much faster level, and that's what has been depicted here. Having said that, in wheat, obviously, as Babu pointed out, there are several references to people using this technology, and DuPont Pioneer is working on bringing in much better heterosis in wheat through a initiation of its breeding program as well, and several other companies as well as public institutes.
Thanks, Anand.
Sir, I have a doubt. Actually, in double diploid, maximally we can have, after crossing with inducer line, as per I know, maximally 8% we can get, that means the haploid product. So how you are improving that one? Means that we can increase the percentage. How means you are doing in your areas? How you are doing that one?
That's right. So in general, the induction rates are governed by certain genetic components, right? So I think the usual induction rates that people have reported encountering is 7 to 8%. But there is an opportunity to improve this induction rate. And when I was in CIMIT, we have done some genetic studies on the genetics of induction rate trait as such, and it's a quantitative trait. So there is one major large effect QTL on chromosome 1, but that alone is not sufficient. So that has to be there. Along with that, there has to be few more additional QTL. So it is behaving more like a quantitative trait. So you need to run a population improvement program to improve the induction rates. So that's the only way. And Professor Sarkar should be able to tell you the stock 6 in the beginning when it was identified, it was only 3 or 4% maybe. Oh, yeah, so around 5%. Whereas now if you see all the modern inducers used by public sector and private sector, you have tremendous rates of induction.
Thank you, sir.
It's not anymore a constraint. Just wanted to say.
Hello, sir. Sir, you are talking about the heterotic group making. In case of a cross-pollinated crops, majorly where the end product is a hybrid, the heterotic groups are made. And in case of a self-pollinated crops also, where the better hybrid, assumption that a better hybrid will be if the hybrid is good, and then the end final homozygous line will be, we can able to get the better one. Is it in case of a self-pers also? Can we make a heterotic groups not for the hybrid purpose, for the development of a pure line itself?
So the heterotic group concept is relevant when the final product is a hybrid, right? So this is basically why it is done, because to enhance your probability of success. So if you pick a line randomly from one group and randomly from another group, it will give you decent amount of heterosis.
So in case of a self-pollinated also,
right,
for predicting the performance of the final product based on the best hybrids. If you have a best hybrid, then if you go on selfing, and then at the end you will get the best product, pure line. So by taking this concept, is it necessary to go for the heterotic grouping?
You can predict the F1s, but then there is no guarantee that you are going to successfully fix that F1 into fixed homozygous lines at the end of the filial generation. So you can certainly predict the F1 performance. You can predict the superior F1s, but then if your final product is a variety again, you have to self it sequentially, right, in 5 or 6 generations. So during that generation, there is no guarantee that the heterosis that you observed in F1 will get eventually fixed. So it's more relevant in a hybrid crop. It's not so. You may have to use the mic. I'm sorry, we can't hear from you. Fixing heterosis through double haploids? I don't know what you mean by fixing heterosis. So fixing heterosis is possible through apomixis, for instance, theoretically speaking. But the double haploids are not for fixing heterosis, but that is to create more.
Is it approach to whether we can fix the trait or those combination in F1 through double haploids or means using this technology?
Of course, double haploids can be used. Suppose if you want to fix 3 or 4 different loci, right? All of them are in heterozygous condition in F1 plant. If you derive DH lines out of this heterozygous plant, you have much better chances of ending up with a homozygous line, having all the favorable alleles in that line. So DH is definitely useful. Yes. So I think I would suggest you to read a paper by Thomas Leberstedt from Iowa State University. So he has written an excellent paper on this one, and where he argues that through DH technology you will be able to fix the favorable alleles in a faster manner than other way.
Yeah, because in my PhD I am also we are also focused on this aspect, whether we can fix the F1s or high performing lines through double haploid in wheat or no. That's why.
Sure.
Thank you.
I have 2 points to make, and I would appreciate your response to those. One is that if you take from 1940 onwards, Pioneer's own breeding program in breeding for temperate maize for the Great Plains, what is the percent increase you are getting in 10-year window, 40 to 50, 50 to 60, 60 to 70, 70? Is it becoming a game of doing very sophisticated things and much lower returns in terms of percentage increase? So that's my one question, and if that is so, then we should be talking about broadening the genetic base rather than making more recombinants out of the 2 pools and crossing them and fixing them. So what is your opinion about that, and what strategy Pioneer is following? Second, in India, as far as I understand, I am no expert on maize, is there anything you are doing for resistance to waterlogging? Because for rainy season maize, that is a very major issue. And with all your germplasm, it would be very useful if you do very strong phenotyping in the country for some resistance to waterlogging situation. These are my 2.
Thank you, sir. Actually, let me take the first tab, and then maybe my colleagues from Pioneer can also pitch in. So regarding the first question, so I think we will also see a presentation from Emerson, where I think in his first slide itself he shows how adopting multiple technology has resulted in incremental genetic gains. So I think he has mapped out very nicely each technology intervention and how much yield jump we have been getting. So that's one part of it, and then the second part of it is whether we should be creating more elite stuff, elite material. So they don't want to they don't want to introduce all diverse germplasm into their breeding program, and that's something has been the main stay of the commercial breeding industry. That's basically because to improve the predictability of the performance, and also whenever you bring in diverse germplasm, there is no guarantee that that's going to result in yield gains. And regarding the second question, sorry, I didn't get the second question. Oh, sorry, waterlogging. So waterlogging, I don't think there is a specific focus on waterlogging, but then I think one way of ensuring this is evaluating the hybrids in very diverse set of environments. I think some of our advanced material, they are screened across very large number of locations, representing all the farmer conditions. So if there are certain conditions wherein waterlogging has been observed, certain hybrids can be selected for that, but then there is no targeted effort for waterlogging. But I know that CIMMYT has been working on waterlogging tolerance for some time. Any of my colleagues who want to pitch in for Dr. Deepak's question? Okay, fine. Sir, please.
Raman, I would like to refer to the question of Professor Pentel to Professor B.D. Singh in the previous session, where he said that for the modern day plant breeding, phenotyping is much more crucial. and you have said so by several advances in phenotyping. But looking at the different models, genomic selection models in particular, the impression is generated that if your genomic selection model is in place, once it is established, probably the breeding would become phenotype independent without even so you could grow it in the glass house contained conditions, and then you can run your genomic selection model and identify the best productive genotypes without going to the field, and to save the plant breeders from the drudgery of standing in the sun, in the water, and the back breaking exercise and so on. So there is a contradiction that I would like to have your response.
Sir, nothing is further from truth than that statement that phenotyping has become irrelevant and you can evaluate material in greenhouse and then you can predict. No. So I think genomic selection, a very integral component of genomic selection is phenotyping. So the only thing is we don't have to phenotype more and more, phenotype less and less with more and more precision, and what you phenotype actually it should be evaluated in very representative conditions. So the germplasm has to be very representative. So I cannot train a population based on tropical germplasm and predict my temperate germplasm. So both your training and prediction data, the germplasm should be representative of your breeding program. And the other thing is the same way how the genotypes were evaluated in the past in multiple locations, that is still needed. Still needed here. Instead of evaluating say 5,000 entries in the field, I can evaluate 1,000 entries and then predict the rest. But that 1,000 entries do not escape the phenotypic filter. For sure, we need to do that. So I think it's a very good point that you made.
Thank you, sir. Now we'll break for lunch. All the students, research scholars, every one of you present in the auditorium are requested to have lunch, please. Thank you. And we'll assemble back at 2:30. There is a frameless specs found in the food court. So somebody, yeah, please come and take it.
Very good afternoon. I, Alok Sev, once again welcome you in this student symposium entitled Genomics-based Next Generation Crop Improvement Approaches, which is organized by Division of Genetics, ICAR-IRI, New Delhi and DuPont Pioneer. In the continuation with the session 1, where we are having various eminent personalities with expertise in different research areas will be interacting to us. In this first, I'm quite fortunate to invite our next speaker of this session, Dr. Emerson Limburger. Dr. Emerson Limburger is from Brazil. He has obtained his master from UFRGS in Brazil and a Fulbright Fellow at Texas A&M. Dr. Emerson specializes in quantitative and biometrical genetics and integrated molecular breeding tools into product development across the corn, rice, canola, and sunflower. Dr. Emerson is currently the molecular breeding deployment lead for the Africa and Europe at the DuPont Pioneer, and today he is going to address us about the deploying molecular breeding to enhance the crop selection. Please, Dr. Emerson Limburger.
Well, thank you very much for the introduction. I do not have a doctoral degree, I have only master's. I just wanted to clarify that. It's an honor to be here talking to such a nice and selected audience. So I just would like to share a little bit of what we're doing at Pioneer. And I think that the symposium, one of the greatest things that it brings is the opportunity for students to interact with the industry. It's important for us to learn what is coming because this is going to be brought by you students to us, and also to give a little bit of the taste of the things that we are doing right now. So if you could focus in different areas based on this interaction. So we already went through this slide. I just wanted to say that Pioneer is probably one of the oldest organization in terms of exploring international opportunities in research. So with that, we have developed a very extensive germplasm, and Pioneer is very well known for all of the diversity that it has in different germplasm pools across the world. India is a very good example of that. There's a lot of interaction between breeders. So when I started Pioneer, a lot of the hybrids produced in Brazil, they had somehow some Asia Pacific background as well because of this interaction that the breeders are always exploring the opportunities of gathering germplasm from one place to the other, as well as some of the materials here in India have a little bit of Brazilian blood as well. So a lot of you have probably seen this. This is the chart that shows how genetic gain in corn has evolved across the decades. Usually the way that this plot is represented is by the introduction of the double cross combinations with the first gain in genetics, then followed by the three-way crosses in corn, followed by the single crosses, which is the most current technology for selling hybrid commercial products. So this is just this very same plot, but we're just exploring a different set of information in the background that also contribute to this genetic gain. So starting off with the very first commercial hybrid from Pioneer, the copper cross hybrid, and that is just showing that sometimes, and the reason why we have this type of product is it's a response to what the market needs, and we're just exploring the best genetics, and the best genetics for this crop is in a hybrid combination. So heterosis has a lot to do with it. It's not a choice of a company or an institution. It's just a reflect of the biology behind the crop. So there are several different breakthroughs here. You can see more recent things like double haploids that are pointing out here, genomic predictions. We started working with marker selection in around 2003 with a very set of first markers. That is not everything, so I just wanted to focus that if you take the recent years, I would say that Pioneer has invested way more money into phenotypic data because the genomic information has no meaning without a good phenotype behind it. So we're just parameterizing what the genome is based on a phenotypic data. So if you don't have phenotypic data, there's no need to have any genotypes because you're just losing the foundation. So I just wanted to reinforce that if you want to do any good work in genetics, you have to start with a good genotype. So again, a little bit of the foundation in terms of how the company oversees its breeding program. So again, the germplasm is the foundation of everything, and Pioneer has been a key company in keeping worldwide germplasm. We're always open to introgression of new germplasm and exploring the combinations across the world. Conventional breeding is always going to be fundamental. I work with Europe, so there's no transgenics allowed in that continent, so transgenic is not an option. But we do have large markets for transgenics like North America and Latin America. And the molecular biology here in the middle, which is I'm going to focus a little bit more as my presentation flows. You have to consider all of those three parameters and the interaction that they give in a biological information. So one of the things that, for example, we neglected in the past, and we've learned with our errors is transgenic interactions with some of the backgrounds. So we have tested some of the transgenes in North America. We've made the assumptions that they were all perfect, and then we released some of the products with a few genes that didn't have a very good combination. So there was a huge drag in yield. We didn't understand anything about that. And now when we want to look at a new project, we're way more careful about looking at the biological information in terms of response before we generate a new product. So starting off with a little bit of philosophy here, Heraclitus said 535 before Christ that you can never swim the same river twice because that river is flowing and the water is never going to be the same. So in the same sense that everything flows, we have to think of breeding as well. So you can never select the same set twice. So all of the decisions that are made by the breeder in a given state, it's flowing. And it's very hard because it can never go back. So the other very important thing is that we can never test the same environment twice. And we have to manage between what is my germplasm response to this environment that it's always changing. So the breeder is always changing the germplasm to make it better, and the environment is always giving a different response. How do you manage that? So the question that I want to try to explore a little bit is, can we try to predict at least the same effects twice? So a few years ago, a colleague from North America, Sarah, she asked me, do we need to have estimation sets every year, or can we just skip 1 year and then do an estimation set every other year? So I was very curious trying to answer her question, and then I was trying to find a way of doing it. So what I did is I pulled the information, and this is from Europe continental, so it's a breeding area of Europe that is more Central Europe towards the east, and it has very stable environments. It's the most constant environment that I've worked with, especially coming from Brazil, where every single year there's a new challenge. Like 1 year you have northern leaf blight, the next year you have common rust, then the third year you have drought. So it's all of those components, they fall into confounding effect with the air, and it's very difficult to pull them apart. So I was trying to answer a very specific question, and I had to kind of filter. I don't want to have confounding effects into my questions. So I pulled continental Europe, and I went back and retrieved the information from the mean locus effect for the previous 5 years. So this is Bayes B analysis. So what you see here is you start off chromosome 1, position 0, and then you go all the way around until you get to the very last beam of chromosome 10 in corn. And here you're going to have the allele effect for each one of the loci. So you can see starting off at 2010, going all the way back to 2014. What you're going to see here is the allele substitution, and the allele substitution is going to always consider the highest frequency allele compared to the least frequency allele. So when you're seeing that there's a difference in the orientation of the signal, so you may have a positive signal and then it becomes a negative signal and vice versa, that is not contradicting information. This is just showing that somehow the breeder has impacted in the allele frequency in a given locus, and we have shifted from one allele that was previously a minor allele becoming a major allele, and vice versa. So we can see here, as Babu was showing before, we have very clear division of the heterotic groups. So we have the female pool and the male pool. And when we are estimating those effects, one of the way of doing it is we just parse the DNA contribution from the male and the female before estimating the effects. You can consider just the zygote with a complete genome where you have all of the three classes of homozygous parent 1, heterozygous, and homozygous parent 2, or you can use this approach. When you have very mature heterotic groups, you're more efficient if you parse those into female and male pool. It's going to give you a much better prediction accuracy. So it's very difficult to see a lot of things here. That was the plot that I thought that represented the best way of seeing this information. But then I went back and I filtered for the most representative alleles within each one of the years. So what I see here, and I wanted to share with you, is that there's a very significant component of the year effect. So you have different responses to different environments, and we are capturing different signals. So in my humble opinion, we should continue generating those estimation sets year after year. And if you want to account for stability, for example, you can just use all of the 5 years to generate your predictions. So you're going to account only for the loci that are very stable across the years. And it doesn't matter if it's the minor or if it's the major allele, those things are associated back to the parent in the correct allele form in which the allele has been identified. So I want to prove that we're actually doing some performance over those germplasm, and then you can think of some of the possible selections that you can have. So you can have a directional, stabilizing, and disruptive selection. So in most of the cases, we're probably going to stay between the disruptive and directional. Stabilizing probably would be if we weren't having any impact within any of the regions. So when you try to fit this hypothesis across all of the markers, you're going to have a lot of calculations to do. So I limited to the alleles that had the higher effect. And then you can see here how the allele frequency was extensively affected within some of those loci. So here you can see in the first option where your major, your effect was already at the major allele, and then you just increase the percent of that to almost eliminating the existence of the minor allele. Here is the same thing, so those two loci, they fit it into this hypothesis. And then here we have the opposite case where you start off with your desirable allele is in a very small percentage of the individuals within your populations. And then year after year, what you're going to do is increase the frequency of this desirable allele. In some cases, you're going to reach 50% of the total pool of alleles, and sometimes you're going, although you start with a minor allele, you're going to become a major allele in the future. Obviously, when we start fixing those loci, we lose the ability to estimate their effect, and then we can move forward and start estimating what is going to be the next set of loci that I should be looking at. So then we come into a new paradigm, and this is something that the breeders have to be keeping their mind on. If you're going to do a very good job predicting, you want to start off in a population, and previously we were looking at yield for grain corn, and now we're looking at yield for silage, dry matter, and this data is from maritime Europe. So what you can see here is that when you start off, you have a given average for the entire population, and what you want to do is you want to increase, and your selected pool is going to have a higher average than the starting population, and you want to have a smaller standard deviation. So if you fail in doing that, you're going to end up having a very similar average, probably the same statistically comparing, and you're actually going to increase your standard deviation. So it's very important that you know all of the information behind your predictions to make sure that you're going to make the correct selection. And then the question is going to be now, am I able to drift, and what is the vector that it's going to drive this difference? So now moving again back to yield for grain corn. This is Mediterranean Europe, and then you can see here, when I look at my female pool, you can see that the majority of the loci within my pool is not going to change. And what I had to do is I had to take the 5-year interval, which is the longest distance that I had, because it's very difficult to see any kind of trend if you go 1 year after the other. And one of the reasons for that is if you think of a breeding pipeline, you cannot impact 1 year after the other, because the populations that you are predicting in year 1 are the populations that were generated in year 0. And in year 0, you didn't have the information you had in year 1. So to be able to actually effectively measure the effect that you're putting into your predictions, you have to take a longer term where you have the full breeding cycle accomplished. So when I look at the, and then for some of those loci, you're going to see that there's a huge trend. You can see here the effects that they have and how much they have changed in frequency. So like from very small frequencies moving into very high frequencies from 2010 all the way back to 2014. Now when I looked at the male pool, it was quite surprising because it really seemed random. And there's no trend. You can see that there are a lot of loci that are changing frequency, but they don't follow any type of information regardless to the effect that they have. So you really have to go back then and start thinking, so what's wrong here? And the communication between the people doing molecular breeding deployment and the breeding community is very important, because sometimes they need to make introgressions to fix some issues that they're having. So in Italy, they had a viral problem, so they had to introgress material that was resistant to that virus, and that changed the entire game. So every time that you bring something new, you're going to have a real big impact in making this type of measurements. So then the other thing that we have seen is that usually you get a much better prediction accuracy when you're looking at prediction sets that belong to the estimation set. So it would be this scenario here. And when we have an independent prediction set from the estimation set, that means that there are 0 individuals that are simultaneously in the estimation set and in the prediction set. Our accuracy may drop, but it also may not drop. So when it comes to the breeder, they have to really put in an account here how much time they have. So if they have a very strong signal from the market saying, I need to have this set of agronomics to be able to come up with a product, I may not be able to use this scheme here, because I'm not going to have to afford keeping my new set of families waiting to be predicted after I generate those estimation sets. So you're going to have to go into a risk game, and then you're going to have to trade with the accuracy that you don't know how good it's going to be. So you can see here that you can either use your predictions as a discarding tool. So you say, I'm going to drop 50% of all of the individuals based on prediction. Or if you want to be more aggressive, and again, this is going to be a risk that it's on the side of the breeder to take, you're going to advance, and then it's going to be whatever selection pressure that the breeder wants to use. So it's just represented here with a 1/3 selection. So here on this side, what you're going to see is you have 4 populations, population 1, 2, 3, and 4, And then for each one of those sets, of a total of 11 populations, so I didn't represent the distribution of accuracy of all of the 11, I just chose the first 4. This is the accuracy for all of the estimation sets when I remove one of the populations from that estimation set. So I just want to highlight here that you have 2 different scenarios, the poorly predictable pops. So every time that I remove them out of the estimation set, the prediction accuracy collapses. So either I overestimate them or I underestimate them, and that becomes a problem. And then you have the highly predictable populations that if I didn't have the star here and the highlight, you would not be able to identify which was the drop population within those charts. So one of the goals that we have in the molecular group team is to try to understand which are the parameters that influence a population from being predictable or not being predictable, so we can help the breeder make a decision in terms of like how aggressive he can be in using a technology or not. But one thing that we can know for sure, and we can already advise the breeders to do so, so if you look here in the straight line, I'm going to have the average of the phenotypic result, and then in the dotted line, you're going to have the average of the predicted phenotype. So you can see that within a poorly predicted population, you can have very small difference between the predicted average of the population and the real performance of the population. But here, you can have a huge difference, and that tells us one thing. If the breeder is going to make predictions, they should never exclude the entire population, because although we might not be influencing the ranking and the order that the prediction is going to happen, we definitely don't know how well this population is going to predict. So here is another thing that I wanted to highlight that it's important in the group that I work with, and I really emphasize. So I call it handcrafting the estimation sets. And handcrafting is looking at all of the possibilities that may influencing the estimation set before they deliver that to the breeding community. So I have an example here where you have yield, and the mean locus effect was finding a locus with minus 5.4 bushels per acre effect. I mean, if we got that, I mean, we have to throw the information into the garbage, because if it's real, we all lost our job, because they're going to clone this gene for yield, and we're all out of the market. So obviously, that has to be something wrong. So we go back and we start to look what could be causing this issue, and then we try to remediate that. So there are different ways of doing that. So one alternative would be to use Bayes-C instead, so you don't have the ability of creating those overestimated effects. It's not the ideal one, because the shrinkage impact of Bayes-C is not always ideal for prediction. So we go back and see if there's drift, if there's genotypic error, if there's issue with populations, and we try to resolve them. So you can see that after the handcrafted was done, this is the final result. So there's no spurious effect detected, and I can see that the prediction accuracy, and those are all independent individuals. They did not belong to the estimation set. They're 100% hybrid or pre-commercial hybrids that don't belong to the estimation set. So my ability to predict those went from 0.14 to 0.48. And then I just wanted to show a little bit of ways that we use IBD as a molecular breeding deployment tool. So Babu already mentioned about that. So you start off with 2 parents, and then you're going to have inbreds that may derive from those 2 parents. So we know the genotypic profile of parent 1, and we know the genotypic profile of parent 2. And then we're going to eventually learn the genotypic profile of all of the inbreds derived from those. So if we assign that those parents are founders, then we can track that within the population. So for the very first inbred, it matched 100% of parent 1. So the profile for the founder is going to be equals parent 1. The second inbred matched 100% the profile of parent 2, so that individual is going to match the founder profile of parent 2. And there's going to be recombination, and we're happy to have recombination because that's what we're going to explore, new recombination events that are going to bring benefit to the future generations. And then you will see that in this case, within this interval, which is a contiguous interval, you know that A comes from parent 1, and then from this A over here to the end, you know that comes from parent 2. But then in the middle, you have an identical profile between parent 1 and parent 2. So how do you solve that? And then you just add a probability that somewhere in the middle, there has been a recombination event. We don't know where it is. We're only going to know what it is where we increase the genotyping and the density, and we could potentially have a straight line between that recombination event. So how do we use this information? So if you go back to the prediction and the estimation sets that I was showing before, we have to have the same genotypic profile between the alleles with the information in the estimation set and the prediction set. What happens is that eventually, and actually very often, we are changing the markers that we are genotyping with the company for different reasons. One of the major reasons for that is a new technology that is going to come up with a lower cost. So once we change, we have to remake all of the markers into the new technology, and not all of the loci, they are transferable into the new technology, so they may fail. And if they fail, they don't become in production, and we don't have information anymore. So then we start to have an unbalanced map, and we have to solve this before we run predictions. So if you remember the individuals in which we had the founder information, here represented in blue and in yellow, and let's say that now in this generation I lost the markers for those 3 markers that I'm showing right there. Because I know the profile of the founders, I can use that to make an imputation. So now, instead of having those empty spaces here, those gaps, and I could have a very high effect or important effect in some of those markers, I wouldn't be using those to predict those inbreds, and now with the imputed information, I do have the ability to use the complete match. Then the second option is when you have disease trait. So imagine a huge collection of inbreds that the breeders are dealing with every day. It's very difficult to know the name of all of the inbreds, although breeders know a lot of inbred names. They cannot memorize all of them. So imagine if you have a mapping for gray leaf spot, for example, and you know that parent 1 is resistant, and then you map, and the position of the gene is right here. So parent 1 resistant, parent 2 susceptible. If you go 1 generation after that, you could be able to estimate which are the inbreds that derive from this cross that are actually resistant and which one are susceptible. So it doesn't matter how many inbreds you're dealing with, you're only dealing with 2 founder information, and you can easily screen for all of them. So the beauty of it is that if you're trying to map multiple populations and your donor is shared between those 2, you're going to be able to enhance the ability of detecting this QTL, and you're going to have the ability to predict the effect of the descents of, and now I'm representing the double haploid, so let's say that you had several thousand double haploids derived from parent 1, you would be able to define all of the ones that carry the resistance, regardless of the population and the combination that they're in. So here you have the resistant ones and then the susceptible ones. Okay, so how I am with the timing? We have 10 more minutes. Okay, so going back to a little bit of what has already been mentioned here, we have to think of genome, phenome, and environment. Another thing that we are investing a lot right now is trying to understand the environment. So those hybrids that have been created for drought resistant, the Aquamax, they're being explored here as well. They are based on environmental modeling. So instead of just using the information of locations, you use the results of those locations, and you also use other information that characterize, and let's say temperature, rainfall, etc., to cluster the environments that should belong together to estimate the phenotypic value. Because the phenotypic value is going to be the one used to parameterize the marker effects, and if they're wrong, my estimation of the effects is going to be totally wrong. Having an idea of the environment, we come back to the phenome, and then we know that we have to split that in observations that are going to be more stable, and I may have, instead of one estimation set, two estimation sets, because my environments, they were parsed. And then with the phenome information, I go back to the genome, and I start to estimate their effect. So collecting a lot of data, it's very important. So you need to have pedigree information, and here you have the two het groups. I could be working in developing populations, so I will be only targeting one heterotic group here. It doesn't matter in which stage of the breeding process you are. You can use predictions at any stage. You don't need to have heterotic pools to use prediction. So I could use predictions to define which are going to be my best F1 combinations, as Babu mentioned during his talk. And out of those populations, it's not going to assure that you're going to end up with the superior individuals if you don't do a good job at the end. But at least you can say which are the best combinations out of all of the possible combinations that you can have. Because obviously, if you have 1,000 inbreds, you're not going to be able to make combination across all of them. So you have to make a decision, and prediction is a way of doing that. You can run predictions at the F2 level. You have to remember that we are not accounting for dominance. There's going to be a lot of segregation at the F2 level, and we could have the very similar GEBV for two individual plants in the F2 level that have totally different marker profiles. So you could have one that it's fixed, two good alleles and are showing the same GBV as others that are hat, but their hat are showing other loci with important effects as well, and that's delivering the exact same GBV. So there's going to be a lot of variation that you should expect out of decisions made in an earlier generation, so you have to be a little bit careful on that. But it's definitely something that can be done, and then if you have a DH, you know that there's not going to be any segregation later, so the decision that you made in the prediction for a given DH line is going to be capped after time. So here you have collection of populations. Now you have to try to understand what are the relations between the traits, what is going together, and so on. Here you have prediction accuracy, so observed versus predicted. You can see that a very nice correlation between both. Here you have the ability of looking at the breeding values. A good thing with and the molecular marker is never going to replace the phenotype, but here I'm showing the phenotypic data. So those breeding values, where you don't see information, it's because there is no data collected for those inbreds in combination. But we have genotyped all of them, so we could potentially predict. And predicting is going to enable us to look at a larger pool. And that's where it replaces a little bit of the field information, because you start to see pseudo phenotypes, which are those predictions of things that you haven't tested yet. Here you're looking at the genetic correlation between a set of individuals, and here a set of different important loci across the entire genome. But the important message across all of that has to be that we're managing very, very large amount of data. And the way that we have to make sure that we're going to deal with this is not going to be an Excel sheet. So when you think of your career, I think that one thing you should be looking at are opportunities of like, how do I deal with big data, and which are the skills that I need to have to be able to manage with those. So right now, in whatever area that we are hiring, we expect people to have some development skills and look in abilities of like managing this huge amount of information and transforming that, managing with a large amount of data and transforming that into useful information that can be delivered to the clients as a product. So here again, similar chart, just showing how we have evolved in terms of technology. Technology has to be always looked in a broad scale with any opportunity that we're going to have. So it's not only about going from RFLPs into GBS, but we have to look at phenotyping, planting, harvesting, all of the possible ways to collect the data. And then the question, what is next? And I think that some of the components of this question has to be answered by the part of the audience here that is sitting at the upper part, the students, because I think that this is going to be what is going to make the difference. So what is it going to be the next breakthrough? Now when we look at someone that is working with SNP and say, oh, SNP was a breakthrough from RFLP, it's very easy because it already happened, right? But the challenge that you have here is, what is it going to be that you're going to bring as a differential that's possibly going to be the next breakthrough in technology, and what are we going to be doing differently in the future that we're not doing right now, that it's going to bring some sort of benefit? Is it going to reduce the cost? Is it going to increase the efficiency? Is it going to reduce the time that we spend into doing things? Or is it going to be something else that I cannot think of? So I just wanted to bring this message. And I'd like to bring acknowledgements to all the colleagues back in the company that helped somehow with creation of this presentation. There's several different people working in different areas that contribute to the things that we do in molecular breeding. And then just leave this message here, you can read it later. It's from Wallace, the founder of the company, and the guy who discovered that making a hybrid would be something that could be turned into a better product to farmers. And then he founded a company that it's where some of you and us are working right now. So thank you very much, and can answer questions now.
Thank you, Limburger sir, for enlightening us about the different aspects of molecular breeding and how to enhance the crop selection. The session is open for the discussion, please.
most of your emphasis is on the genome to select the better individuals. But how much your breeding component is emphasizing on trait breeding?
So in terms of disease trait, any type of trait?
No, what I mean to say is focusing onto the trait, that this trait I want to introduce, something like that. How much your breeding program is tackling this issue, or they are not doing anything on that aspect? Trait breeding.
And could you give an example of trait?
Like suppose you have a good line there, you want to introduce a particular trait.
Yeah, so we do that a lot. So let's say that I use a gray leaf spot, for example. I can go back to Italy to the virus case. We had a founder, B73, that carried resistance to that, so we could use that information that we had to increase the frequency, and we actually had to fix that into the Mayo pool because it was bringing sterility into the production. So in those cases, what we're going to do, there are different approaches. Usually linkage is enough, so you don't have to know exactly where your gene is, as long as you have a set of markers that are going to respond to that, are going to identify that haplotype. You just use that in different ways. So one way that we use is doing backcross. So let's say that you have a commercial hybrid that is susceptible to that virus. We replace that locus with a favorable allele, and we replace the commercial hybrid with a resistance to that trait. So we do have a lot of efforts in native traits. In some of the tropical lines, it's a little bit more complex to be able to define haplotypes because there's so much variety, diversity, that sometimes you could have the markers that you have in production in Pioneer may not be able to identify correctly. So when you map those, we go into a new marker set development until we find something closer that can characterize that specific gene, and then we go into either forward breeding, back cross breeding, any type of approach where you have a few markers that represent one trait that you want to introgress. There's more reluctance in meat corn to do that because they're very fond of their closed germplasm. They don't want to break all of that linkage that they have created through the heterotic groups. But they are doing it as well. They have been forced to do that. But
how much here this molecular breeding you are applying to the canola breeding?
To canola?
Yeah.
So this year we used the whole genome prediction for the first time in canola to predict their new testing combination, so all of the new hybrids. The idea is that we're going to deploy that for all of the crops. Obviously corn was the first one and kind of was used as a role model, but we're doing the same thing for sunflowers, for rice. I think that Dindo just created predictions for rice this year, so we are in the process of leveraging this information for all of the crops.
Just quickly, I wanted to respond to your question. So when he talked about the estimation sets, so they are not just genotyped. So estimation sets are also well phenotyped. So phenotyping is very much an integral component of what he talked about. So it's not selecting based solely upon genotype. Yeah.
Thank you, sir.
Okay, one small quick announcement. The students who are going to present their poster, because we have a poster session in the evening, so somebody will collect from please pass it to them because they are going to paste it. And during the poster session, you all have to be there in front of your posters because committee will evaluate and others will be there. So somebody, some of us will collect. Yeah, we have to collect from here because the conduct is good. Everybody has to go. Session will. Yeah, please.
Now I call upon the stage our last speaker of this session, Dr. Justin Gerke. Dr. Justin Gerke is from United States, where he received his PhD degree in genetics and genomics from Washington University School of Medicine in 2008. He studied population genomics as a postdoctoral fellow at Princeton University and University of Missouri before joining DuPont 5 years ago. He is currently a research scientist in global breeding and marker technologies, specializing in population genomics and data science as applied to plant breeding. And today he is going to address us about the sequence-based breeding concepts and application. Please welcome Dr. Justin Gerke.
Well, first of all, I want to say thank you. It's a real privilege to be here at this institute with a rich history, a lot of expertise, and the talent that I see out in the students. So thank you for having me here. I'm excited to talk here today. And I'm going to talk about, as Babu titled it, sequence-based breeding. And another way I think about it is high-throughput genome science and how we integrate that within the specific context of commercial breeding. So this is an outline of my talk. So first I'll give an introduction about my background and the group I work in at DuPont Pioneer, and then I'll give a background of molecular breeding. And this is something that both Emerson and Babu have talked about. I'm going to go through it again in my own way, both to lay the foundation so I can then build high throughput genomics on top of it, but also because it's just something that's simply fundamental to what we do in molecular, with markers in molecular breeding. And then when I have gone through that background, I'll then put advanced genomics on top of that. And what I'm talking about there is primarily genome sequencing and what we get out of genome sequencing. And then I want to end by putting that back into the larger context of what we're trying to do with plant breeding at DuPont Pioneer. So first, the introduction portion. And I wanted to go through my background because it's not necessarily the traditional agriculture background. I didn't go to an agricultural university such as this one to get my degree. I have an interest in farming from my family background, but I got my PhD at a medical school, and I got my PhD work studying yeast actually. And what we were trying to do was identify the specific quantitative trait nucleotides, the DNA sequences in the genes, giving rise to variation in a complex quantitative trait. And at the time, in the early 2000s, yeast was really the only organism you could do this in. That's the way the technology was. But the technology shifted about 3 years into my PhD. I had been doing a back cross with selection to try to isolate these loci for about 2 years when something called next generation sequencing or 454 sequencing came out and completely changed the technological landscape. I abandoned over 2 years of work on my PhD thesis, reformulated, redesigned the entire project, and did the whole thing and that portion of it within 6 months, starting over from scratch using this new technology. And so that rapid shift in technology and genomics is something that's shaped my career. After studying in yeast, I then went on to look at C. elegans, which is another model organism, a nematode, and some additional increases in genomic technology allowed us to rewrite the natural history of that organism. So that's something that I've seen throughout my career is the rapid pace of change in genomic technologies. And what I saw in my postdoctoral work is that the technologies were getting to the point where it was cost effective to do this within applied systems, such as plant breeding. And that's what led me back into agriculture to DuPont Pioneer. So the group that I work in DuPont Pioneer is called Applied Breeding Technology and Genomics. And our role is to evaluate new technologies for use in plant breeding. And genomic technologies is one of the types of technologies we look at, but there are others as well. And we use a variety of approaches, including quantitative and population genomics, which is what my background is in. So a background on molecular breeding. There are many ways we use molecular breeding in Pioneer. And Babu and Emerson talked about quantitative trait locus mapping versus genomic selection. I'm going to focus on the molecular side and on the idea of a haplotype and what a haplotype means. So I'll start, because that's kind of where genomics, I think, gives us a real advantage. So I'll start just to define what I mean when I say a haplotype. Really, all I'm talking about is a segment of the genome. It contains many bases of DNA. It can be 100,000 base pairs. It can be 1,000,000 base pairs. It just depends on the context. And when I say that two individuals share a haplotype, it means that they have an identical DNA sequence in the region. And something that's been touched on is that we use identity by descent to track haplotypes. And this simply means that if a segment of the genome was inherited from a parent, then the offspring and the parent are identical by descent in that region. And they therefore also have the same DNA sequence and share a haplotype. But IBD is only one way to track haplotype sharing. We can do it by direct DNA sequencing as well, and that's what we can do with genomics. But this concept of haplotype sharing and tracking these haplotypes through a germplasm is critical to understanding how we use molecular breeding to relate the phenotype to the genotype. So here's a diagram of using parent progeny IBD to track haplotypes. So what I'm showing here is are in these rectangles, these colored rectangles are haplotypes within the genome. And I have two parents in this cross, parent 1 and parent 2. They have different underlying DNA sequences, different haplotypes, so they're colored differently. And those haplotypes are tagged with SNP markers. And then I also have shown here a single progeny from this cross. And when I show diagrams like this, I'm showing fully inbred individuals such that you would get from a doubled haploid, from a DH that will be talked about later. And in this case, we've genotyped the progeny at only a subset of the available markers. But those markers we can see inherit unambiguously from parent 1 and not from parent 2. So we know that at those markers by IBD, the haplotype is the same as that parent. Now, if we know there's not a lot of genetic recombination within this region of the genome, we can use an imputation algorithm to then impute the genotypes at the rest of those markers. And so now we've used IBD and imputation to fully project the sequence of parent 1 into the offspring. If we've used genomic selection to assign phenotypic effects to the marker alleles in those parents, as Emerson talked about, we can then project those down to that offspring as well. So this is an example of a figure for using molecular breeding and IBD to track haplotypes, and how we use haplotype sharing to then project phenotypic effects as well. And this is something that Babu talked about when he mentioned that one way to think about genomic selection is that we're not looking just at the effect of the individual, but the effect of the haplotypes as well. But we do this in more complex pedigrees, not just parent 1 and parent 2. Here I'm showing a case where we have 4 distinct haplotypes and four founder inbreds that are each marked by 2 SNPs. And if these are crossed together at generation 1, we can genotype that individual and then match it to the parental haplotype and follow that down through the pedigree. And we can do the same thing in another cross. And then when those 2 individuals are crossed together, we can just repeat this process and track haplotypes down through the pedigree. And we do this not just in one region of the genome, but throughout the entire genome. And we can do it in complex pedigrees. So this figure shows a complex pedigree where I've highlighted the specific regions of the genome, the specific paths through the pedigree that are relevant to the individual region of the genome that we're looking at here. And using markers, we can track that haplotype from founders down through multiple generations, and we can identify recombination breakpoints as well. If we move to a different region of the genome, we might find there's a completely different genomic history. That region of the genome inherits from a completely different founder, and so at that region there's a different haplotype involved. When we do this throughout the entire genome, for many individuals, we end up with these haplotype profiles, where individuals that have the same color within a genomic region are assumed to have the same DNA sequence underneath. And we can then use field evaluation, phenotypic measurements to assign effects to specific regions. Here I've isolated a specific region of the genome where the haplotype effects show a difference in the ability of the inbreds carrying them to withstand high winds. So the effect here is on the percent of inbreds snapped in a plot subjected to Boreas testing. And if we sum up the effects throughout multiple regions of the genome, we can then end up with a prediction of the effect of each of those inbreds. And that's one of the things we do in genomic selection. So that's another go through our background on what molecular breeding is. And so now I can talk about how genomics augments that picture of how we view our germplasm from a molecular basis and then relate that to the phenotype. I talked a lot at the beginning about how my career has been shaped by change in technology, but genomics is only one place where there's been technological change. Technological change is all around us. What I'm showing here is a figure of a hybrid breeding program. Most of the work I've done has been in hybrid breeding in maize. And in hybrid breeding, especially in maize, you have well-defined heterotic groups in North America. They're the stiff stock and the non-stiff stock, as Babu mentioned. And new inbreds out of these heterotic groups are advanced based on their performance in test crosses with the other heterotic group. And another thing Babu mentioned earlier today is that one of the things that's changed is technology. He talked about online banking, I think was one thing he mentioned. Just as technology advances, the word I wrote here is breeding becomes more global. Perhaps another way to say that is breeding and life in general is becoming more interconnected. We don't think about breeding in terms of an individual breeding program, but we think about many breeding programs that all feed information into each other. And they not only contribute that information to each other, but they pull information out of that as well. So that's, I think, the broader trend of what's happening in plant breeding, especially at least in commercial plant breeding, and I think in the public sector as well. And then change in genomics is happening in the background, and the main change in genomics is cost. This is a plot showing the cost of sequencing a million base pairs of DNA over time, and that cost has just plummeted within recent years, and it keeps going down, and it's not leveling off. It seems to be going down even faster. And so one of the reasons we can apply genomics within plant breeding, which is a numbers game, the reason we can use genomics on such large numbers is because it has become cost effective. The technology has enabled that. One of the most basic things we get from this is an increase in marker density. So what I'm showing here is what I would call low density genotyping, where there's one SNP marker every few centimorgans across the genome. And so each of these pixels represents a SNP. And they're color-coded based on parental inheritance. So this is an individual that shows a recombination breakpoint somewhere right in here. But with low marker density, we don't really know where exactly that recombination happened. It's uncertain what the inheritance in that region is. One of the first increases in marker technology we all saw were SNPplexes. In maize, there was the 56K SNPplex. And that allowed much higher resolution of these types of recombination breakpoints. However, we're still using markerplexes here. We don't know exactly where the recombination occurred. And if you see a region like this, where it's white, where there's no information on differences between the parents, we don't know if it's really the parents have the same sequence there, or we just were unlucky and didn't pick the right marker to distinguish them. So we're still using markers to represent the underlying DNA sequence. With genome sequencing, we actually know the exact or near exact sequence that underlies our markers. So our markers are no longer just symbols to say, well, one thing is different from another, but we can actually look at how different they are. So if I go back to this previous example and I go from a 56K view to a sequence view, just the amount of data we're dealing with explodes, and some of that information that we get out of that is useful. We can identify exactly where the recombination breakpoint happened, and we can identify clear regions where there's long stretches where there's no sequence difference between the parents of this individual. You can also see some errors, some little dark blue spots in a region that we know inherited from the other parent. And this is common in high throughput genomics, but the error, the noise is swamped out by the amount of signal we get. So we've moved from just saying with markers that in a given region, parent A is different from parent B, but we can now quantify exactly how different. So this is a software view that I collaborated on with a software developer to help us visualize this genomic sequence data, because there's so much data to look at. How do we synthesize that? So this is a view where every small pixel in this view is a single SNP marker obtained from whole genome sequence. And each of these columns labeled A through H is an inbred. And these inbreds are being compared to a reference inbred. And so the way I've coded this is that any SNP that matches that reference inbred is coded blue, and any mismatch to that reference inbred is coded orange. And so we're now, like I said, we're seeing more than just are they different or the same, but we're seeing how different. We can identify long haplotype matches between one of our test inbreds here and the reference inbred. We can also identify microhaplotype matches. So these are small regions of identity that may not happen through pedigree relationship, but through something else that we haven't captured before. We can also see rare double recombinants that sometimes are quite small. We'd have to be very lucky to capture these types of recombination events with traditional molecular markers. And then we can also see regions of extremely high diversity and regions of extremely low diversity where they're different. If we use molecular markers, we'd see that they're different, but that the level of difference, the level of genetic relatedness between the inbreds at these particular regions can be vastly different. This is a practical example showing that principle. This is a QTL region where there's an important a QTL in maize. It's an important economic QTL. And that's why we looked at this region. And what I've plotted out here is based on genome sequence, a diversity measure in 10 kilobase windows throughout this region. So the higher the point on this graph, that region of the genome shows higher diversity between these two haplotypes that differ for the QTL effect. And so what you can see is that there's this region in the middle where there's very low diversity within this QTL region. And in fact, if we do the math on this, the common ancestor that gave rise to these differences probably existed no more than 1,400 years ago. However, these are two distinct founders in our germplasm, and which has only been around for 100 years, let's say. The breakpoint that generated this piece probably happened, the recombination probably happened about 300 to 400 years ago, and the surrounding regions are over 200 times more diverse. So if we're searching for where the causative gene for the QTL might lie in this region, it's much more likely to lie in these highly diverse regions than in this region here. So we're getting extra information from the DNA sequence that we wouldn't get from other tools. And so what's happening here is that we're no longer just relying on our pedigree information to track haplotypes through our breeding, but we can now see uncharacterized ancestors. We can use the sequence data to go back in time and see events, recombination events, and hidden ancestry that we can't capture in our pedigrees because it happened long ago or it just simply wasn't recorded. So if I take a view like I showed you before, which is based on founder IBD, pedigree information, and I then shift that to a view that's based only on DNA sequencing and matching haplotypes based on DNA sequencing, you can see regions like this region right here where there's a lot more variation in the top picture than the bottom. And what that means is that there are founders that were different founders in our breeding pools, but that have the exact same underlying sequence. So we're adding information and getting a better view of what our germplasm looks like. And we expect this to happen in maize because of its rich history. Maize was domesticated in Central America and then spread through North and South America. And as Emerson said, there's been exchange back and forth between Asia and the New World since then. But these types of tools, this way of looking at the data can be used in other crops as well. I was going to go through this, but Babu showed these slides, so I'm going to skip this part. But just as a reminder that I showed those cartoon pedigrees, but in reality what we're dealing with is a situation that looks like this, with really a pedigree universe of, in maize, our two heterotic groups. So we can make these types of views for other crops as well. here's an example for a different crop where we don't have 2 different sections of the universe, but we actually have 6 if you look at it this way. And so what I want to end on is talking about how we got not just global or more connected germplasm knowledge and high density data, but we also get biological insight. And when I talk about biological insight, what I mean is getting down to the gene level. So one of the things we can do is not just make broad, megabase, millions of base pair level haplotypes, but we can make haplotypes at the gene level through the same process of aligning short reads to the genome, calling variants, and then building the models for what the alleles of those genes look like. Okay, so this is an example of a gene where we've identified a particular set of SNPs and small insertions and deletions that distinguish the different haplotypes that we're breeding with in our germplasm. We can also model these through haplotype networks where the size of the node represents the number of individuals carrying that haplotype, and the distance between these nodes represents the number of genetic changes between them. So we can synthesize this information quickly into a view that it makes this vast amount of data more easy to interpret within the right context. We can also look not just at SNP variation, but at gene presence-absence variation. This is something that's extremely common across multiple crops, especially for disease resistance loci. So what I'm showing here is a graph of sequence coverage in a set of inbred lines. And these 3 inbreds at the top have no sequence coverage within this particular region of the genome. I collaborated with some of our laboratory scientists, and we confirmed that this is in fact a deletion of this gene. This gene is completely absent from the genomes of the 3 inbred lines at the top relative to the 2 inbred lines at the bottom. We can also do this quantitatively, where we measure not just is a gene present or absent, but what's the copy number level of that gene relative to other inbreds. And just like we build haplotypes with SNPs, we can build haplotypes with these other sources of information as well. So this is an example where we built a haplotype network for copy number variants, where there's a set of inbreds that have what we call wild type or the same copy number as the reference genome. Some inbreds have 1 or 2 more copies than we see in the reference, and some have 2 less, which is probably a deletion of that gene. And just like we relate SNP haplotypes to phenotypic effects, we can do the same thing with copy number variants. Another type of technology we're looking at is different ways of using sequencing to assess what parts of the genome are active and being transcribed or being involved in gene regulation. This is work from Eli Rogers-Melnick when he was a postdoc, but he's now been hired at Pioneer and works in our group. This was work to identify regions of open chromatin in the genome through an assay called MNA sensitivity. And I suggest you read this paper if you want to learn more about this, but in general what it identifies is promoters, enhancers, and active gene regions. And what they showed is that if you combine this with phenotypic data, you can determine what portions of the genome are active and giving rise to trait variants within a specific context in a specific tissue. So those are some of the things that we're getting from genomic data that just scratches the surface, but hopefully gives you some ideas for questions to talk about later. And so I just want to end to reiterate what Emerson and Babu said. And that's that, what I talked a lot about was the genome. How do we understand the genome? But that genome doesn't give us any value unless it's put into the right context. It's put into the context of the environment that we're trying to breed in, because that's what our end customer sees. We sell the genetics, but they see this whole package. And so a continuing challenge will always be the fundamentals of breeding and getting out in the field and understanding the plots. What gave rise to the phenotypic signal you see in your estimation set? This is the most important part, regardless of the genomic technology, whether it's SNPs or RFLPs, SSRs, all of which I've used. The sophistication, the most important part of the experiment is always going to be the phenotype, and often nowadays it's the most expensive as well. Another challenge I want to leave you with is just the fact that these features we're looking at are locked up within the genome. This is a plot of physical on the x-axis versus genetic position on a maize chromosome. And this type of trend exists in other crops as well, not just maize. But there are regions of the genome where there's vast physical stretches of DNA, tens of millions of base pairs with very little recombination in any one generation. So a challenge for future research is when you have copy number variants, open chromatin, SNPs, indels, all locked up together, all affecting traits differently. How do you utilize that information within a genome that has this type of pattern? With that, I just want to end by saying that it takes a lot of people and a lot of different disciplines to use all this data together. And so I think it's an exciting time to be in agriculture and to be working at a place like Pioneer, where I get to interact with software developers, population geneticists like myself, quantitative geneticists, and lots of different people to try to make these data actionable and useful. And with that, I just want to thank the major groups that I work with and a long list of people. This is really a team effort. There's more people than I can fit on the slide and more than I have time to name. So with that, I'll end and take any questions you have. Thank you.
Thank you, sir. Now the questions are invited from audience, and students are highly appreciated if they will ask the questions.
No questions?
Sir, while doing haplotyping analysis, so how actually the corn genome is having so much of transposon, so how that will affect that one?
Yeah, so that's a real challenge. So, like if you sequence a maize genome, you're going to throw away about 80% of the reads because they map to transposons, and you know they map to a transposon, but you don't know which one. And so really, when you look at the maize genome with short read sequencing, what you're looking at are islands of genes separated by material that you just can't penetrate. And so you have to rely on the genetic map in the physical reference genome to help you order those gene islands. That's really what we're doing is making haplotypes based on those gene islands where we can get good sequence alignment. That's a great point.
Thank you, sir.
I think there's one more.
Hello. Thanks for the wonderful presentation.
Thank you.
Just wondering, I have seen from the morning that most of the approaches are mostly on SNP based association or understanding the trait genetics through SNPs or reading the nucleotides. Just wondering how much importance would you like to give, from your company perspective on other approaches such as transcriptomes, proteomes, or microRNAs? Do you think that these approaches are still relevant to understand the genetics of the traits, or are you using these technologies to fill up the gaps in understanding the complex traits?
Yes, we are. So the question was about other types of features such as RNA sequencing, metabolomics, and I did talk about copy number variation a bit, and although he pointed out that most of what I talked about was SNPs, and I think that's partly because SNPs they're easy to detect and they're easy to work with because it's binary. It tends to be one allele or the other. That makes it very easy to use in the computational framework. But I absolutely think that the other types of variation are important. We are doing active work in those areas, especially with RNA-seq. And really, I think a trick with transcriptome RNA data is to treat it like a phenotype, because you get a quantitative range of values. And then I think one thing that's very useful is when we identify, for example, a copy number variant that we know is useful, because they tend to have large phenotypic effects, can we use a SNP to track that, simply because they're easier to detect? But yes, we are actively looking in those areas as well.
Mm-hmm. Closely look into sequence level variation, which I fully agree with you is a great new development. But how are we fitting it into with trait specific improvement? After all, there is a lot of importance of old fashioned breeding methods, not breeding methods, breeding objectives, like objective based breeding.
Mm-hmm.
Is it uh, resistance to some disease? Is it resistance to... So how do you feel these techniques help us there, where you have some major trait that you want to bring in and you have to maintain that minor level variation to keep the yield up?
Mm-hmm. So I think that one way I like to think about it is that using genomics and gene sequence level brings the promise of bringing some biology or biological modeling back into that trait. Genomic selection is primarily a statistical operation where you're using statistical transformations to relate the SNPs to the phenotype. If we have gene sequence for a particular set of genes we know are crucial to a disease, for example, we can explicitly model those as factors in that analysis, separate from the background SNPs in the genome. I think that's the type of analysis we
basically
can do.
genomic selection is to pick up all possibilities for better yields as we go along.
Mm-hmm.
Is that where you think it's going to help us the most in terms of making sure that everything that is of importance gets stacked into the hybrids through these multiple markers available all through the genome?
Yeah, I think that genomic selection will always be important as a way to track that genomic background variation. But that, yeah, if you have specific knowledge of a gene you know is important, that can be added explicitly to the model as well. It's not an either-or situation, in my opinion. Thank you very much.
Thank you, sir. It was really a wonderful talk. Now we will break for the coffee, and after 10 minutes we will reassemble here. Thank you. All the registered participants from IARI, please submit your posters at the registration counter. Welcome back once again. Now we are going to start our second session, second technical session, which is about enabling technologies. I take this opportunity to call upon the stage our first speaker of this session, Dr. Anand Pandravada. Dr. Anand Pandravada is a research director for CON for the Asia Pacific region at DuPont Pioneer. Dr. Anand obtained his PhD from Kansas State University and is an accomplished corn breeder, known for modernizing the corn breeding programs in the major private sectors, organizations such as Monsanto and Pioneer. And today, he is going to deliver a presentation on the next crop breeding frontier, precision phenotyping, the next crop breeding frontier. Please welcome Dr. Anand Pandravada.
Thanks for the introduction, and very good afternoon, everybody. It's a pleasure to be back here at IARI. I did work at IARI as a research associate. You know, a lot of people here were my mentors, including Vishwanathan, who's sitting here, and spent some time, started my graduate school here, but then went on to Kansas State to get a PhD and finish my degree there. So it's a pleasure to be back at IARI, and this is like second home because there's a long association with this place. So, you know, I think there's a lot of, you've heard a lot about genomics, and I'll relate it back to the first talker, Dr. B.D. Singh, and you know, one of the things that he said about, and I think there was a very relevant question on whether all of this will supplement or take away phenotyping and phenomics. So we have a very eminent breeder in our company who has been breeding corn for 35 years in Brazil. His name is Delmar Brenner, and he's one of the most successful corn breeders. He has hybrids that have been put up in 30 million hectares if you take year on year in terms of what was sold in Brazil. So he initially commented, well, great, technology is great, but I do have two very powerful markers, my right eye and my left eye. And so that's, but the breeder, at the end, in his career, he came back and said, well, but I think there is a significant enhancement in my productivity when I'm using these tools, and this is helping me make better selections as I go forward. So that's the power of these tools. So they're not standalone, but they are ways and they are supplements to what breeder does with his field phenotyping and what is going to happen. So I'll spend a few, I think the next 30 minutes or so talking about phenotyping, and so I just wanted to acknowledge a few people because conceptually a lot of this has been worked out in corn, not just for the genomic selection but also for the trait specific breeding. There was a question earlier on, do you do this with traits? And I think the concepts apply equally to both trait as well as whole genome prediction. And this group here are very much involved in developing some of these concepts. It may have taken 15 years to do this in corn, but now we are realizing that the concepts are in place, the framework is in place in a crop like rice, in a crop like mustard, in a crop like canola, in a crop like wheat or sunflower. We are able to deploy some of the same strategies within a couple of years. So all the hard work that was done in corn sort of set up the framework to then very quickly deploy not just the genotyping but also the phenotyping methodologies across crops. So if you look at the spatial variogram, you know, let's say you're selecting for two different traits, and you have this peak where you're selecting for high yield and this where you're selecting for drought. Well, if you're here and you're selecting within this population that you have, you do not have the variation to go further than where you're here. And if you're here, you do not have the variation to go further here. But if you're in this valley, you obviously have a chance to both go up for yield under, let's say, well-watered condition and yield under stress. If you extrapolate this to multiple environments and multiple geographies, your phenotyping is then a factor of your genotype and the environment, and each of these may represent one of these environments, and also the genotype by environment interaction which is coming into play that you see here. And then there's this error component that you can never get a good handle on. So what does this mean? That at any given time, the phenome is more a theoretical construct than a realistic target for characterization. I can never get the perfect phenotyping. I can never say I can achieve heritabilities and accuracies in my phenotypic selection that will be 90% year on year, because that's the effect of environment, right? But what you're trying to do is you interpret it within the context of the dynamic temporal and spatial conditions. And these dynamic temporal and spatial conditions could be your locations, it could be the soil, it could be the temperature, it could be whatever else that is causing these variations that are there. If a breeder knows that this is the source of the variation, then he will be able to address by selecting for the variation once he knows the source. And unless you know what this source is, it will be very difficult for you to breed for it. So interpreting the phenotype is also in the dynamic germplasm context. So if I don't have variation for antisilking interval in my germplasm, I mean, I may put the most intense drought pressure, I'm not going to make any genetic gain for it. So everything is in the context of the germplasm and the variation that you have within this germplasm, and integration of phenotypic and genotypic data through the prediction framework thus helps a breeder to put these two things together and then understand the germplasm much better to make good advancement decisions. And rapid precision in phenotyping provides a great mass of data. So Justin just showed the power of sequencing, right? So we were discussing yesterday, so at a 3x coverage, you can take a maize inbred and get a sequence done at about $300 or less. That could be 10x coverage for rice. So the cost of doing that for genotyping has come down significantly, but the cost of phenotyping is still not come down. So it is much more expensive in today's world to get a good phenotype than to get a good genotype data. So while we are going through a breeding process, because you have untested materials, and you see these numbers, and so it's like 1 million double haploids that are possible, right? You cannot test 1 million double haploids. You can genotype 1 million haploids, but you do not have the resources to test 1 million double haploids. You would probably then take about 10,000 of them, and then, well, I can phenotype them at a couple of locations. But then if I need the precision in terms of my G by E and trying to understand the stability of a hybrid across multiple locations and multiple years, I don't have, even being in the private sector, we don't have the infrastructure to do this at such large levels. So the challenge is to come down from this 10 to the power of 6, which is 1 million double haploids which are untested, to that the 10 commercial hybrids that will make it to the end of the cycle, and which go into the farmer's field, and the farmer then says, well, I have a hybrid that I'm going to come back and plant next year. I think that's the challenge that we have to face. So in doing this, we are creating recombination between all these untested lines, and the other additional power that comes in the breeding program is the way we leverage the breeding across programs. So for example, if there are 10 breeders and each one of them is having a breeding program, if you have, this is very much similar to the AICRP testing, but it's much more at the pre-code level and at the basic TC1, TC2 level. If you are able to combine all of these together, enhance their data and utility, it's like one big breeding program, and that funnels the phenotype data that you need for generating good quality estimation sets, good quality advancements. So, and again, if I were phenotyping with one breeder at 4 locations, I may not have the needed resources. But if I have 4 breeders and each of them are doing 4 different locations, and all of these breeders are able to send their material to all of these places, that's 16 data points instead of 4. A breeder will now get a lot more opportunities to select on G by E than he would have otherwise, right? So now, putting into context some of the things that were talked about, Babu showed this pedigree diagram. So you have your stiff stocks and non-stiff stocks. This is in the case of hybrids. In the case of varieties, you still have your varieties that you know are doing well, and you have them still separated. You have the IBD, which Justin so elegantly explained, and the haplotype that was explained, and then you're selecting on the QTLs that are coming from these particular founders. And then what I was just wanting to show here is, very quickly, you can, if you're using markers and you're saying I have 300 markers, or my entire, let's say I have a 300 centimorgan chromosome, a large portion of the variance is explained by the first few markers. And then as you go on to the top, in this area you have very few, you do not have a lot of the variation explained by your genetic models, and this is where you're operating in this sphere which is not completely additive. It has a little bit of additive and epistasis mixed into it. So how do you make selections in this sphere? And so I'm sorry. So one of the things that then we started to go in was then take these estimation sets that we have and then identify, let's say this is the correlation coefficient between estimated performance of a trait and the predicted performance of a trait. Let's say it's at 0.8. We then went in and said, what has been tested is about 400 different lines in a line by line combination. If you go in and say, I have the tested by tested subsample versus a new test of tested by untested and untested by untested, what would your correlation coefficients be? And you can see here that as your trait heritability is going down, your correlation coefficients are significantly going down, and especially in untested by untested, you do not have as much predictability, or you do not have as much correlation. So this still means that when you create new germplasm variation, to create an estimation set for this new source of variation, you do have to phenotype some of these untested by untested, so that then you have a better estimation set going forward. So the other aspect of this whole prediction is traits, and coming back to trait-based selection. So in drought, for example, shedding, GDUs to shed and GDU to silk are two key traits for corn. And what you see here is the correlation between observed and predicted for GD to shed in stiff stalks, the ASI, which is the anthesis silking interval, and the GDU to shed and the ASI in the non-stiff stalks. You can see that it significantly varies, and so you probably have a very high correlation coefficient R square between your observed and predicted for shed GDU in the stiff stalks, but then you do not have that correlation in your non-stiff stalks. And this is maybe because that you're selecting for female producibility, you're selecting for silking vigor, and all of those traits that happen in the females in the stiff stalk pool, maybe. So this again points out that you do have to make substantial selections in the field to maximize this potential genetic gain. The other aspect of this whole phenotyping is spatial variability, and we did see a slide from Babu today earlier about spatial variability, but I just wanted to bring this up again. And so if I had a field and I drew a heat map of yield, my green would be the low yielding plots. Each of the square here, by the way, represents a 4 meter by 2 row yield trial plot. And what you're looking at is a red area would be a location which is low yielding, a green area is a location which is high yielding, and orange is somewhere in the middle. So if my experiment was in this particular block, then I would have lost a lot of data. So once I knew the variability of the field, and this is where phenotyping becomes accurate. So if I go back to the same field next year and I redid this experiment, and this is exactly what is being depicted here, I've avoided these spots where it's red. I've avoided these spots where there's a lot of variability and tried to map my experiments into regions where you do not have as much variability for yield. And so what does this do? It brings down the spatial variation in your experiment. So this depicts the adjusted yield versus unadjusted yield. And so what you're seeing is in unadjusted yield, you're seeing that you have a much higher variation than in the adjusted yield, and you're seeing that within the reps. So if you had 3 reps and you had 1 rep here and 1 rep somewhere in this area, you would see that this rep 3, which falls into this low yield, would probably have a significant difference versus rep 1, which probably falls into a higher yielding location. So the point I'm trying to make is, as a breeder, I need to know where I am phenotyping. And if I know where I'm phenotyping and can adjust my experiments so that it does not fall into regions with variability, I may be able to increase my heritability or my prediction accuracy as we go forward. So having locations which are very uniform is very important, especially during your early stage selection. The other aspect of this of phenotyping is environmental classification. So what you're seeing here is days after sowing, and on the y-axis you're seeing supply-demand ratio. So let's say it's about 60 days after planting. This curve, this line is showing you the supply-demand ratio in the case of non-stress. And what it is indicating is there's a relatively high supply, especially for the demand in this particular window. And so when we look at this data, and this would be one of the 130 locations or 20 locations, we would classify this location as being a non-stress location. So a similar exercise can be done now. Now that was at a location level, but then you extrapolate the data to all of North America, for example, and then you can say in 2012, for example, in the US, a lot of this geography that is to southern US was considered subtropical, and you had a lot of temperate dry weather in 2012. In 2011, you had a different distribution compared to 2012, where you had much more of a milder weather with higher rainfall activity that is given by this dark green, which is temperate. So 2012 was a comparatively drier year compared to 2011, and then 2010 was also, you had a lot more rain and humidity compared to 2010. So once I know this, the next step is to go back and say, okay, I layer this information to where I am exactly testing. So what I'm showing here is some of the locations that we have used for testing corn in India, right? And this would be spread out between all the states, all the way from Punjab, UP, Rajasthan, parts of Gujarat, Madhya Pradesh, then you have Maharashtra, major Karnataka and Andhra Pradesh. And using this classification, what we can then now go back and say is at what stage of the crop's development you had incidence of stress. So if I was breeding for drought, and as all of us very well know, drought at flowering is very much different than drought at grainful, and drought in vegetative is different than optimal conditions. So at one stage you have an effect on ASI, at the other stage you have an effect on barrenness. So this helps me then go back and say, well, out of all these locations, I had 5 locations where I have flowering and grain-fill stress. Now, if I didn't have this, can't I do this? Of course I can. So if the person who is growing these plots has the knowledge, he'll go back and say these are stressed but you don't include that in your data analysis. You don't make that part of your decision tree. You don't use the data to create breakouts of stress versus non-stress and look and identify hybrids that are stable and can be performing across locations. So the weather data, the location data, and the uniformity data helps us then pick locations where we can get significantly higher heritabilities for the trait of interest than if you were doing this on random. So I think this is something that is very, very important, especially now that, I mean, you can even do jivas with random data, but so as many of the speakers in the morning pointed out, the quality of your phenotypic data is affecting the quality of your estimation sets and thereby the quality of your predictions. So with this will help you create the right breakout. So for example, if I was selecting for drought, I would probably focus on these 5 locations to create the right estimation set versus not selecting the low stress locations going forward. So what does that mean? It means that we are trying to develop climate resilient crops. It means that we are understanding how the climate is changing, and then we are using this to translate climate to yield and then adjust our breeding programs so that we are then increasing our genetic gain for a given change of climate. So this also helps us to react very rapidly to changing climate. So if I knew what I was characterizing and what I'm breeding for, I'll be able to do a better job breeding for the future. So just moving on from the environmental and location classification, I think one of the other revolutionary phenotyping methods that's come on board is the satellites, UAS, and rovers. And for example, in North America, many of the commercial as well as public institutes are routinely using drones for phenotyping their yield trial locations. And they get data for things like population, stand count, plant height, and we'll talk about this in the future. But there's also high resolution satellite data which was earlier restricted that is now available publicly. For example, Digital Globe will be permitted to sell imagery up to 25 centimeter panchromatic and 1 meter multispectral GST. So imagine this, 1 meter is, so I get for my 10 square meter plot, I get 10 pixels of data. Earlier, we can't get 1 pixel for a 25 square meter data. So 10 pixels versus 1 pixel is a huge difference in terms of what can your ability be in terms of interpreting the data. It's like having 1,000 SNPs versus having GBS data on a particular inbred. And that's the difference that we are seeing in terms of ability to use some of this data, provided we know what are the useful spectrum that we are after, right? So the other aspect of this is the rapid explosion in drone technologies. So there's on-ground phenotyping equipment like this. For example, this has a high boy and which can go over either soybean, corn, or you name whichever crop. It has multiple sensors on it, multiple cameras on it, infrared, hyperspectral, UV, you name it, and this is all GPS reference. So for every plot that you'll get a score for not just the phenotype but also the different spectral characters and every other sensor that is on this particular plot. The drones obviously are much more high throughput because they will be able to do a lot more geography, but this gives you the resolution that you want on terms of ground. I was talking to a professor at Purdue and he said they are now looking with IBM at a collaboration where on a single tassel of corn in a plot, they're going to have close to 1,000 pixel resolution, right? And imagine that, and so we're talking about 25 pixels if it's a 1 square meter resolution, 10 pixels, and so now we are looking at each plant getting 1,000 pixels of data, and so that's the kind of big data explosion that is happening, and I mean this is where the future opportunities in terms of really understanding the phenotype is. So quickly moving on, The other aspects which could come in are using the spectral signatures, either satellite, airborne, through thermal radiometry for different kinds of measurements. So, and the physiologists will be very aware of this. The photoinhibition, photooxidation, sun exposure, water stress, each of these are defined by a certain spectral signature. So now you have drones that are fitted with cameras, or you have high buoys that are fitted with sensors that just go over the field, and for every plot you get a different measure of all of these things. So now this doesn't supplement the yield and the breeders walking the field. It's adding an additional layer of data for him to make selections. So this is another way of, for example, here you have a sensor that is looking at soil moisture level in the soil, and it's transmitting that to the weather database. All of this data is being collected at a central server, and this central server then says, well, for these 1,000 locations, this is the rainfall that you have, this is the cumulative rainfall that you have, this is the soil water content that is available, this is the evapotranspiration. You have other images and other stuff that is happening, and so this is just a pictorial view of that. So image analysis, and Amitabh's group does a lot of this more than we do at in breeding, but it also helps you define growth and parameterize growth. So so here you have some plans of either soybean or Arabidopsis, where you're looking at different images and then using the pixel counts to sort of see what is the different growth parameters that are present. And you can then look at the spectral data and then sort of parameterize some of these things to see with time how are the different genotypes growing and morphing themselves in different. So if I say I'm interested in a genotype that has high leaf area accumulation, I'm probably then very quickly looking for 1, 2, 3, and then if I'm saying I want something that has a late stage explosion in leaf area growth, I'm probably looking at this guy in the red right here. So that's the capability that is added, that is sort of in place, and guys like Justin, Emerson, Dindo, they help us do a lot of this this analysis. The other aspect of what we are specifically doing in terms of trait selection in corn is drought, and Babu referred to a slide on Acromax, that the product that we have developed in US, and we are also in the process of trying to develop similar products in the rest of the world. So what we see in corn is there's a significant difference in terms of tip fill, and this is one of the things that corn does. As it senses drought, especially after flowering, different genotypes behave differently. Some of them, they leave the tip, the others, they become barren, and some others become scattered. So if you were able to get an image of each of these kernels and then convert this image into something called the individual ear traits, which is the number of kernels per ear length, proportion of filled kernels, then you can go back into each of these plots, and if this was a single plot, you would say that you could get an image and say how many of those have scattered, and then you take it through an analysis where you have the raw image, you filter for the background, you separate the kernels, you mask the entire ear, you count and measure kernels and measure them, and then find the tip kernel abortion, and then identify how many of these kernels are tipped. So now each one of these is a different trait that is available. So for a breeder, he has a plot level yield, he has the days to flower, he has the days to silk, and we were talking about phenotype being a limitation with thousands of genotypic data points. Now I have tip kernel abortion, I have area where I have barrenness, I have scatteredness, I have basal skip. All of these are traits that are additional enablers for the breeder, and the breeder is selecting on these additional traits along with the yield that he's getting from well managed versus droughted locations. So that's what I was trying to convey here. And then the other, now how do you integrate all of this together? So in today, you're having field temperatures, and tomorrow you'll be do predictive analysis, driver and scale with UA scouting. So I think this is a reality in the US now. So farmers have on their combines on their planters sensors that are sort of saying, this is the level of moisture I have, this is the level of salinity I have, and in some cases they're saying, okay, based on what I know about my soil from last year's yield, in this particular part of my field I'm going to grow this hybrid, in this particular part of the field I'm going to grow soybean, in this particular part it's much more of a low-lying area, so there is a chance for water logging, and I'll try to find a hybrid that is much more reactive, and then I have a slope where I don't have water, so it's on the height, so I'm going to put a drought tolerant hybrid. That's where you're trying to go with this in terms of offering solution to farmers. Now, in a context like India, where you have farmers with 1 hectares and 2 hectares, the level of service we can provide on this can be completely on a different scale, trying to individualize the farmer selections in terms of areas or blocks or districts where each of the hybrids or varieties are being grown. So, and that sort of depicts what is happening for some of these large growers. So they're looking at a time lapse on this is on 27th of July, and then on September 12th, and they're looking at the variation in the field spectral signature, and then using this to make recommendations on which part of the field the variability is there and what recommended hybrids have to go. So I mentioned about using drones for phenotyping. This is a real life example of how we do it. So if each one of these here is a plot, each one of these is a 4-meter plot, we can fly a drone quickly on the canopy and then get a spectral signature of this plot, and then you take out the ground signal out, you take out and then create a model, and you'll very soon be able to get an estimate of plant height. Now, earlier plant height is taking a stick, walking a field, so, and then if I have 10 acres, I'm going to have 10 acres that every person walks each one of those plots. Now, we are able to do this in 5 minutes, but it's a lot more accurate, because now you have thousands of plants, and every plot you're measuring every plant instead of one average plant being the measure for the plot. So you're increasing the precision of your phenotyping, thereby giving the breeders the ability to use genomic selection with much better ability to create good estimation sets. So this is another example where you could use the same technology for canopy health. So a trained machine learning algorithm calculates the probability that a plant pixel is green, brown, or soil, and then utilizes that to basically give you canopy health. So if my trait of interest is northern leaf blight or anthracnose or I don't know, stem rust. So what this does is it goes back and uses the pixel data to say this plot has a green foliage cover, this has a lot more brown spots, and then gives you a relative measure of NLB, northern leaf blight, or anthracnose of any other foliar disease, for example. You could also use this for nitrogen sensing and use it very accurately. So I think this is where the technology is growing, and the reason I want to point this out is for students who are here in the cusp of entering into their careers, it's a completely new ball game in terms of what you could do with breeding, genetic gain, plant selection, and doing accurate phenotyping. So putting the whole thing together, you've seen the germplasm, you've seen some of the genotyping, phenotyping, and IM systems that Emerson, Justin, and Babu talked about. And at the end of the day, if you are not able to phenotype well, you will have very low prediction accuracy. So once you do that, once you create your break routes, once you identify the traits you want to focus on, you can create very good estimation sets based on your phenotype, increase the response and increase the ability for a breeder to select on them, and then skew so that your early generation testing is mostly genetics based, but then you generate all the needed data during your relay testing. So pre-commercial, long strip trials, you cannot not do that. You still need it. You need to do multi-location testing at your advanced stage hybrid so that when it's released, you have 60 to 70 locations of data based on which you have confidence. So, and then the new component that is being added here, which again makes this multifunctional, is the importance of physiology, because crop growth models can do a lot of stuff. You have seen that with the plant height illustration that I have given, the Arabidopsis growth that we have given, that then can be used to train the data sets appropriately. So in this example, for example, you can use, you have the different models that then affect every leaf's initiation, and then when the flower initiation will happen, when will the flowering happen, and then use this in terms of the date of planting, the type of weather, to model the stress that a hybrid would experience. So a quick example is the maturity, where in a linear model, and we were talking about Mendel's laws, and this is a 2-gene model that we have here. A double homozygous dominant will have 10 kg yield advantage, but when you have a QTL, the QTL, this is for maturity, I'm sorry, so this will be 10 days later compared to the AABB. But if the same region you have a QTL that has an effect on yield, you will drive down the maturity, but you will also be affecting the yield. So understanding the relation between these two traits that are not synergistic but are antagonistic is very, very important, and you're seeing that in some of this here. So if your leaf number is between 12 to 14, that's where you're peaking in terms of grain yield. And if you say I'm going to breed for a phenotype that has 16 leaves, the modeling data says that you're not going to gain more yield, but you're going to lose yield. So all of this then can affect your predictions and the gain that you can make for each of the traits that you have. And this is a model this is a slide that shows the crop growth model included prediction model compared to the genomic blobs, and you're getting the same level of accuracies with the ABC CGM compared to the genomic blobs. And so for this to work, you have to have a suitable model. You need to identify the traits suitable for a particular model. So in the case of drought, for example, your leaf exertion, your silking, all of these are traits that you're important. In the case of heat, you're interested in tassel blasting. So in the case of diseases, it could be something else. And define suitable prior for the traits, characterize the soil and weather management, and then implement advanced agronomic management so that you can hit the precision in terms of phenotyping and perform high quality phenotyping. And so with this, that sort of significantly increases your genetic gain, but also increases your response to selection, right? So, and I'm going to skip this in this interest of time, but what you're seeing is that when you are doing a CGM model, your heritabilities are quite high, and they are comparable to what you were doing with just based on yield. So what else can be done? I think you can broaden the range of environmental conditions included in the MET. You can extend the ABC algorithm to have multiple environments, and then refine the crop growth models appropriately for each of these crops. And I think this is time that needs to be invested. So we did this with corn, and maybe that's something that we can do with rice, with wheat, and depending on the importance and the value that the breeders see for these particular traits, and then increasing the prediction accuracy. So all of this boils down to increasing our success at this particular point, and then making sure we are bringing only what we think is worth testing into this pipeline here, and then using genetic selection to take the untested, and then I narrowed the funnel from the million that you have to 10,000 here, and the 10 products that you want here. So with that, I think that's the last slide that I have. Thanks for this opportunity, and again, a pleasure to be back at IARI.
Thank you, sir. Your lecture was really very informative. You made us clear about how to go for the precision phenotyping. Now the session is open for discussion.
Either you understood everything I said, or I spoke garbage. Students, any questions from you guys?
So when we are breeding for the yield or complex trait like drought or heat stress, so we have many component traits we are phenotyping for with the help of a phenomics platform and all those things, hundreds of traits we can take. So what is the selection criteria that your company is following for the many traits in order to select for the best plant? Right.
So that's a great question, and the question is, out of a plethora of traits that you can collect, what are the traits that you would want to focus on? And again, so at the end of the day, there should be a strong correlation between the trait that you are phenotyping and the yield that you would be able to demonstrate under stress, under farmer's condition. The trait should be scalable, meaning that I should be able to phenotype. If I'm not putting it in the field, I should be able to phenotype hundreds and thousands of them very efficiently. If I take 10 minutes or 1 hour for a particular X phenotype on a trait, it's not worth the time in terms of trying to do it. So scalability, repeatability, and accuracy in terms of yield under farmers' conditions will drive the number of the traits that we are focusing on. And so, for example, ASI is one of those traits. So out of you could look at water deficit, you could look at stromatal conductance, you could look at stay green, you could look at whatever else you want from a physiological point that can affect yield. But after looking at all of those things, this is something that is scalable, quickly measurable. You can take large phenotypic data and has high relevance to yield under flowering stress.
Sir, for example, if you selected the 10 traits that are very important, after the 10, within the 10 important traits which are correlated and regression is also very high, then with the 10 traits, how you are going to further select?
So then if all of them are equally scalable, equally correlated, then the scalability and the ease of trying to measure that becomes one of the most important factors. Because remember, I'm not looking at one breeding population and 200 lines. I'm looking at 10,000 lines in 10 or 20 different breeding populations, and how to screen it. So scaling up the phenotyping becomes, and doing that accurately becomes one of the ways that we are selecting the trait.
Thank you.
So I want to know whether the controlled environment phenotyping you do, or all the phenotyping you do it in the field condition.
So Professor Vishwanathan, I think controlled environment phenotyping is one of the center stage of our phenotyping. Because it allows you to make sure that you know what you are looking at, and you get a measure of your genetic reaction and potential to that condition. So, for example, we do have a drought controlled environment here in India. We have a heat controlled environment in the north. We have such environments that have been created, and that's where those fields that we remember, that we know exactly which part of the field is good, which part of the is bad, so that we don't map a drought experiment into low yielding location and create variability. So those are all controlled locations where we'll be able to do it. But now you could also do this with the new technology in other locations and sort of mimic the controlled environments very efficiently.
I wanted to know what is the phenotyping cost in terms of developing the infrastructure and facilities for individual location for the kind of operations that you are talking about.
So, so if you were trying to say in a location where I have about 4,000 test crosses that I'm going to screen for, let's say, drought, I would need about 20 acres of land. I would need to make sure I have a good source of water. I would need to make sure I have uniform pressure so that there is no pressure differential and the slope of those land. So doing all of this, I think it is very much within the realm of trying to do it. Now, the question is, if you have to do this in many locations, it becomes an issue. But within the breeding programs, we do have 1 or 2 such locations where we scale up and create a capacity for about 20 or 30,000 plots, then take them to multi-environmental testing and do the environmental classification, then to make sure that this hybrid performance is translatable to the performance in the field. But the cost is not something that is a huge factor in this. This is, and in some cases you could just do this in contract farms where you have taken the land on lease from the farmer, and then all you're investing is precision in soil leveling, irrigation, and then trying to put in drip infrastructure and things where you could do liquid fertilizer and other to minimize variation.
I'm just curious to know, how do you map that plot that this is high yielding, this is low yielding, this is moderate?
So that's a good question. So, because these are different genetics, and so one of the things that we do when we do the spatial analysis, and we do this over all the locations, whether it is controlled or non-controlled, is, there's a spatial analysis you could do when you have the spatial checks that are embedded into all of these locations. So the spatial checks gives you a spatial semivariogram for each of those locations, and that's where you generate blues that then you are used to calculate multi-location blobs.
So thanks for a wonderful and very comprehensive presentation on phenotyping. Just as a follow-up of what Dr. Vishnathan was asked. Sometimes back, I had a lecture or presentation from Peter Langridge, who was the leader of this Adelaide Functional Genomic Facility in Australia. And in fact, they were one of the pioneers, establishing this kind of facility in the world. He made a presentation but the conclusion was they were not able to find any correlation between the field-based phenotyping and control phenotyping analysis. Incidentally, I have also seen that there was not much about control phenotyping data from your slides, but of course you said that you are also looking to that kind of things. My curiosity or question is that do you have any real-time data using field-based and highly controlled facility, real-time data, or what would be your opinion about that?
So Napoleon, that's a great question. And so I think for corn, one way it's fortunate because you can generate very good controlled phenotyping data for a trait like drought, for example, and you would have very high correlation with what is happening in your target market. So the question is, if I was doing my controlled environment in US for, let's say, drought, and my target market is in India, and I know that there's no high night temperature in the US, and therefore the plants get enough time to recoup and regenerate, versus here where your differential between night temperature and high temperature is very high. It does not reflect the performance of the product. So I mean, US to India is a very wide extrapolation, but it is possible that people can do things like that where the controlled environment targets the trait, but then there are other things that are happening. For example, the differential between night temperature and day temperature is such an important thing, especially in the tropical world for heat, that if you didn't have that variance that is captured in your controlled environment, it will never give you the field performance or correlation or accuracy in target environment. So I guess I go back to setting up the controlled environment right, and if you did that right, then you would have a high level of correlation.
Have you tried this approach for phenotyping against any of the biotic stresses?
So that's a great point. So especially the satellite imaging data and some of these drones are being right now calibrated for doing diseases. Emerson had an example of GLS, but wherever you can you can find detectable differences between phenotype, you'll be able to scale it up using some of these technologies. But for diseases, we have some of the best heritabilities when we are trying to do a managed environment. So in the case of downy mildew in Indonesia, if you are able to ensure that you have a 80% humidity, you don't get high daytime temperature, and you are fogging it whenever the humidity drops to a certain point and create a managed condition, you will get the disease that is very highly heritable and expressible. I'm not so sure how we are doing on time. So.
Yes, sir. Very nice presentation. I would like to, I have a different type of query, like after all these precision phenotyping and the product going into the market, have you got any situation wherein the predictions have said like this is going to be your yield, and in the real farmers field it has not performed, and then you have gone back and trying to find out the reasoning for why it has not performed? Any such specific cases in your experience?
So not specifically, and I think well, in all of this, we have to keep in mind there are other things that we are not factoring in. For example, we may have situations where a hybrid is high yielding, but the female has some issues with producibility, and hence you cannot take it and scale it up in the farmer's field. Or the male has severe tassel blasting under heat, and you didn't predict for that, and then but in general, I mean, there will be a few. I mean, if you're having 100, the chance of having a false positive is always there, right? This is because this is a type of error, and it's not that it is error proof, but you're trying to minimize that as much as possible. So for major hybrids, we still didn't have any of the disasters we are talking about.
Thank you, sir. Thank you for answering the queries. Now I would like to call upon the stage the next speaker, Dr. Amitabh Mohanty, Senior Research Manager responsible for trade discovery and optimization in the Asia Pacific region for DuPont Pioneer. He obtained his PhD from DU and postdoc from Cold Spring Harbor. He leads a team of highly talented researchers engaged in the state-of-art basic and strategic research in corn, rice and Arabidopsis at the DuPont Knowledge Center, Hyderabad. And today, he is going to talk to us about a very popular technique nowadays, genome editing a carton-based genetics. Please welcome.
First of all, good evening everyone, and it's great to be back to IARI, this great place which has got such a great legacy. It seems like yesterday, but I used to be working in the genetics division rice field with Dr. VP Singh, who was here till few minutes back. I owe a great deal to him for my career. So when I started my PhD in DU, I came to him that, sir, I want to do transgenic work. And please remember, I'm talking about 93, 94. There was not much transgenic known anywhere. Rice was not transformed till that time. He said, you know what, we have a really great variety, PV1, but it has got so and so problem. If you can fix this, that would be great. And that started a long association with Division of Genetics. Although I was not a student here, I was a student of Delhi University, but that relationship continues. And it's great to see like several of my teachers, Professor Pentel is here, my colleague Dr. T.R. Sharma, Dr. Singh, Dr. Tilachal Mahapatra, with whom I interacted in the rice genome sequencing days. Again, it's great to be back. At this outset, I would also like to thank my pioneer colleagues. Many of them are here, and some of them who are online right now for their passion and what they do every day. So I'm representing everyone here. So since it's a student symposium, I thought that I'm going to talk not only about genome editing. I know everybody is excited about genome editing, but I thought I'm going to talk about trade discovery challenges and how genome editing has the opportunity to change the landscape completely. So that's what I'm going to focus. So those of you who thought that it's going to be all genome editing, please don't be disappointed. I hope there is enough information in their presentation to make you happy. With that, let's begin with the first slide. This is what I'm going to talk, like, who we are, what do we do, what are our aspirations. And as I said, I'm going to focus mostly on the trait discovery. There were some nice questions about control environment. Hopefully some of those would be addressed in the talk. And as I said, towards the middle of my talk, I'm going to focus on the CRISPR-Cas. So we are facing this big challenge, right? We need to have 70% increase in food by 2050. We're going to have 9 billion people, right? Energy demand is going up. Pollution is going down, right? Farmers are burning straws in the neighboring state, and Delhi is getting foggy, right? All of us are like, the environment is deteriorating. Our land usage, the availability of land is going down, right? We are losing huge amount of land for urbanization. At the same time, one of the stark contrasts, like I was talking to Professor Pentel a couple of years back, maybe he was writing an article, and he mentioned to me that during discussion that the land holding of Indian farmers are getting even smaller than what it was a decade or two back. So that poses challenge. Anand and others, all the speakers before me, they alluded about all the great thing that technology can do, but when you have such a small farm holding, how much of this can be applied in that scenario, right? That's another challenge. So we have huge challenges. So what do we do next? So what are the main challenges for agriculture productivity? Now this is basically two of them. Drought is one of the main one, and how do you use nitrogen better, right? If you can solve these two issues, it's going to take care of it. I'm going to talk mostly about the corn context, but many of the lessons can be applied for rice. And again, without going too much detail, depending on which stage the stress is, you can get up to 40% to 50% yield loss, right? If you are having at this stage, you pretty much lose huge amount of yield. Also, another point to be taken into consideration is that nitrogen represents a major input cost all across, right? Along with seed, nitrogen is one of the main cost. And there are two factors here, right? One is that we want to use the nitrogen better, and we also know that the nitrogen is getting leached into the environment, right? So how do you use nitrogen better? So that's the need for a sustainable use of fresh water. There is a need for sustainable use of fresh water. And how do you leverage conventional, native, transgenic and management practices to improve plant performance? We spoke about G by T by E, but management also play a huge role in getting good yield. So you heard about Acumax. This is one of our flagship product. We are very proud of it. And it's a work of a great many colleagues from Pioneer that we have this great product. It's under huge acreage. And one of the things that I want to highlight here, it has got lots of great qualities. Better root system, better silking system, lots of them. But the major part is that under water limited condition, it gives you high yield, right? Lot of times we ignore that, under stress condition yield is good, but what happens in the normal condition? This is one case where under normal condition also is good, right? So we tend to ignore that part. It's a very, very critical, crucial aspect of any commercial organization is that, the performance has to be good under stress condition, but at the same time under normal condition it should be at least neutral, right? And that is a filter, so when you have this control environment, when you go from control environment to field, this is what needs to be taken care. So it shows that genes do work, right? So because these are combination, this is a native traits product, but of course this has got important genes. So what do you do? So there is a big translational challenge, right? So we talk about gene or molecular expression, right? But then what we need is yield. Now, how many layers are there in between? There is cell level, tissue, organ, plant, crop, community, cropping system. This is where the yield gets reflected, right? Now, why it is so challenging to we had many questions about traits. What about including trait, right? So why it is so challenging to implement trait-based crop improvement, right? Because diversity of germplasm and complexity. A trait may work in one germplasm, may not work in other germplasm. Component trait phenotype may not be penetrant or relevant broadly. That's another challenge. Environments vary widely over the years. We saw many of the my colleagues very elegantly showed that the variation that is there, and significant interaction between transgene by genotype or gene by genotype plus environment. And I'm ignoring management part right now, right? So this is a huge translational challenge, right? How do you deal with it? We deal this part of the problem with multitude of options. So first of all, we do a lot of control environment discovery, right? So for example, we use model system, Rhabdopsis. We have a multitude of different assays for like nitrogen as well as drought. We use cutting edge high throughput phenotyping imaging system to get data. So we can observe plants growing faster. We can observe plants growing or dying slower, you name a thing, we could do it, with the imaging system. But again, those are tools, right? End of the day, we want to see plants giving yield to the farmer in a farmer's field, right? That's the goal, right? So all of these are nothing but tools. We do have component trait-based surrogate assays, and then of course ultimately the taste of the cake is in the pudding, so that has to be in the large scale field testing, right? Now, how do you do? We do scale and diversity of our discovery engine, trying to get to unique and diverse mode of action of various genes using model system. We do a lot of testing in the elite germplasm, precision phenotyping, many speakers including Anand talked about this. We have lots of different assays, control environment assays, field managed stress testing, crop modeling. So there is a matrix approach to optimize G by T by E interactions. So it's a very complicated universe, but we are trying to manage the translational challenge by using all these platforms. So what do? We use forward as well as reverse genetics in model system, and rice fortunately for us is both model as well as a crop of interest, so that really helps. We do that in Arabidopsis as well. We mine a lot of our knowledge, so as you know that Pioneer has a big pedigree, really huge pedigree information, right? So we use, we have many leads, so which pathways they are in, we expand leads from there. We use computational approaches a lot, looking at literature, in-licensing, all sorts of, so knowledge-based, forward genetic as well as reverse genetics-based, all these discovery platforms are employed for discovering particular trait. It could be drought, it could be nitrogen, all of these, right? So if you can see that, what the power it gives, you have diverse high throughput platform, you have diverse gene sources, and I alluded earlier that, we are looking for different mode of action. When you have genes, we don't want to have similar gene, we want to have different types of genes, and diverse functional assays. Now this is a depiction of our pipeline. These are in the phase 2 which are advanced, but look at the things which are coming next generation, right? These are even better than this, which is really exciting. We have already seen, there was a question about traits. We have in transgenic approaches, we have seen significant level of efficacy, reproducible efficacy across years and locations, unique mode of action, and of course all these are proprietary. Right now we have invented, we have discovered, we have tested them. So this puts us in a really good shape. This is one example of lead optimization. So once you get once you have a lead from your discovery pipeline, it's not the one that's going to show up finally in the product, right? There's a lot of work goes on, and we use a plethora of options to improve these leads, including expression optimization, combinatorial design matrix, diverse enabling elements, expression profiling, gene stacks, and all these come into picture. How do you manage the G by T by E interaction? We test lots of constructs, then advance only small number, then finally only when smaller number goes down, right? We have precision phenotyping. You talk Anand talked about. This is one location in California. We use double haploid, which I guess my next speaker is going to talk about, and ultimately this leads to some very few number of events, which is going to be commercial. This is an example of yield and yield stability for improved productivity, across environment. These are kind of qualities that we are looking for top end yield. This is a control hybrid. This is a hybrid which has got transgene into it. And I want to also emphasize, if you can achieve something with native traits, the genes that are naturally present in corn, we do not want to unnecessarily, go to transgenic. It's not that. We want to use all the approaches. Whatever approach is there, we are looking for improvement. If the gene is there for a particular trait in our corn genome, yeah, then we will exploit it. If it's not there, we will use transgenic. Same way for genome editing, we are trying to look for an opportunity which was probably not there previously. Now, what kind of yield advantage? Like this is one example of one particular transgene. These are two different events, and you saw the numbers that I showed in the earlier one. In order to get to these two, it takes several years and hundreds of testing to reach here. But you could see the efficacy levels. They are really looking good. So I hope that I have answered that question. That whether you can use model system or model approach or control environment phenotyping to get leads or genes, which will show up in the product pipeline. So I think that question is answered. So what is we have several elegant talks, all across. So as you could see, I hope that with these talks we are not scaring the students. These are, please remember, these are all tools at our disposal. These are nothing more than tools. You remember, think about India a decade back, right? Having a landline was a prized possession, right? So what we did as a country, we moved to cell phone. Everybody has cell phone, and India is the largest market for cell phone. So that barrier is gone, right? But that's a tool, right? Landline was difficult to get. It was difficult to maintain, manage. Only limited number of people had landline. We went to cell phone. So all these technologies that we saw there, these are tools that we could use for improvement, right? So let's see what could we apply, right? So in the plant breeding context, molecular breeding, all these great tools that we have at our disposal. How do you enhance the traits? Phenotypic evaluation is one of the key criteria. And how do you use biological information, math, statistics, modeling? Anand talked about modeling, breeding, bioinformatics, all these together with the aim of increasing rate of genetic gain for the desired trait. You could pick and choose your trait, what you want to work on, and look for these areas, right? With that, I'll stop the first part of the talk. So I wanted to give you a quick glimpse of the trait discovery effort, and I'll move to the CRISPR-Cas, right? So what is CRISPR-Cas? And I must thank Professor B.D. Singh for giving an introduction. So I got rid of some of the slides because I didn't want to repeat. But let's look into what is there, right? CRISPR-Cas is nothing but a bacterial adaptive immunity against viruses, and it works by RNA-guided nuclease system, right? The full form of CRISPR-Cas is clustered regularly interspaced short palindromic repeats, and Cas9 is nothing but CRISPR-associated protein. So there is a guide RNA, which is a small synthetic RNA, so which guides Cas9 to target DNA, right? And how it came into being, you know that there are bacteria and there are bacteriophages, right? Bacteriophages would go and kill the bacteria. So if you have useful bacteria, for example, lactobacillus, right? If the bacteriophage goes and infects lactobacillus, it's going to spoil that one. So the lactobacillus which produces yogurt, you will not get the right kind of it will spoil things, right? So the bacteria has evolved an immunity system that when these bacteriophages infects them, once they get infected initially, they developed an immunity. So what they do, they incorporate part of these viral sequences in their genome. So whenever the invading virus comes second time, right, they're able to attack and chew those genomes of the viruses. This is how this system evolved, right? Now this was a 3-component system. Now it has been made into 2-component. But very simply, why there is so much hype about this particular technology? There are other genome editing technologies that has come to picture, that has been fairly got fair decent publication, right? For example, few of the earlier technologies, right? Now the big difference between the CRISPR-Cas and any other technology, I would give again a cell phone analogy, right? So with the earlier technologies, what we need to engineer was protein, and all of us know engineering protein is much difficult than engineering DNA or engineering RNA for that matter, right? So what CRISPR-Cas does is that what you need is a Cas9 protein and a guide RNA. This guide precisely takes that Cas9 to a particular location and does the edit. Okay, and I'll get into the more detail. just coming back to the analogy, right? So this can be like an analogous to a cell phone. So for example, you have a cell phone which is nothing but the Cas9 protein, and you have a directory of people listed there. So with that Cas9 protein, your directory, you are guiding to, like, you are able to call 2 people, whosoever you want. You may have a list of 300 people, like, so you call them. Whereas with the earlier generation technologies was like for each person that you want to call, you want to have one cell phone, right? Because you have to engineer each protein, right? I think that is the biggest difference, and that's why this is such a revolutionary technology. Let's try to understand what is it. Now I want to emphasize here is that genome editing is nothing but a precision mutagenesis tool, okay, or precision breeding tool or advanced breeding tool. Why I am saying this? As you know that mutagenesis is fundamental to evolution, and crop breeding has a long history of safe use of it. And there are eminent geneticists and professors sitting here. I hope all of us will agree that agriculture today is where it is because of lot of it is because of chemical mutagenesis that was used 20, 30, 40 years back, to create variation. Genome editing is nothing but a tool to create precise variation, in a very targeted manner, right? So genome editing continues the history of improving crop through modern targeted mutational application, right? It allows mutation to be targeted to a very specific and desired location, which is why this is so precise, right? This is where the power comes from. For multiple I think please remember this point. I think if you get this part and from the talk, I think I would have been make a good point. For multiple applications, the final plant lines are indistinguishable from those that could be obtained through conventional breeding. And here is an example what the mutagenesis has done for mankind, right? These are all the crops or species that, this is how the corn used to look like, teosinte, this is how it looks like today, and all these species, right? Just take for example, tomato, like there are so many variations there, and this has, a lot of it has come from chemical mutagenesis, a lot of it has come from genes, so this is where we are today, right? Now let's compare it with conventional breeding, like, and please remember I'm talking about conventional breeding, right? With the tools, things have improved quite a bit now. So if you have a plant line which is tolerant to drought, but it has got lower quality other plant criteria, and you have another one which is sensitive to drought but higher quality. Now you want to bring these A plant to B plant, these two qualities together. What you got to do? You got to cross them multiple times, select them, keep on selecting, right? At this stage, you have after how many generations? 5 generations, still 3.4% of your genome is of the undesirable character, right? And we know a small small chunk of DNA even in kbs can have a yield drag, right? Now what happens next? In the advanced breeding or precision breeding, what we can do? Going from incorporating desired characters in multiple cycles of common breeding practices, we can incorporate in as little as 1 to 2 cycles by CRISPR-Cas. So you have a plant A which is lower quality and tolerant to drought. You have a plant B which is high quality and sensitive to drought. With couple of generations, you can get both these traits together. So this is one example. This is a drought tolerant hybrid that we have created. This is a drought sensitive one, right? Now what has made it possible? So please remember, CRISPR-Cas is only one component of it, right? There are other important players in this role. So who are they? You heard from many of the earlier speakers from Pioneer that understanding the elite genetics, high quality DNA sequencing, cost effective DNA sequencing, informatic tools and infrastructure is absolutely critical, the ability to deliver the elite genetics, I think this is a significant gain that we have made, and there is I would urge all of you, please, there is a plant cell paper that has come recently from us. Please go and read that paper. You will see a lot of advancement that has happened, And then the third part is that, the CRISPR-Cas technology itself, which are the tool with superior activity and targeting specificity. I think this is very, very important. The activity has to be really good and the specificity has to be really, really high. Then only will make good progress. And incorporating in-house as well as collaborators expertise. So we have been very open to collaborate in this area, and we have many, many collaborations that we already have in this area. So CRISPR-Cas furthers innovation for pioneer brand products, and you can have potential, like the potential products are listed in this slide. We could have output traits, hybridization, increased nutritional value, drought tolerance, disease yield, and our hope is that we could have the products as early as 5 years. Of course, this is subject to regulatory approval. There is still some uncertainties there, and we have to see how it is dealt in the country. I really commend Professor Pentel for really pushing the technology, transgenic technology for many years. Probably I remember when I was a PhD student, he was doing this work, and it's 2 decades since then. But hats off to him that he is still pushing really hard the system to make it happen, and which is the need for us as a whole, So now let's go to the next slide. I promised Dr. A.K. Singh that I'll finish early so that I can give back some time to the next speaker, and I hope I'm able to do that. Now how the technology works. The technology is very simple. So what happens is you have this protein which is a double-strand binding protein. It goes and binds to the DNA, which is guided by the guide RNA, and makes a cut, right? Now plant has got its own system of repairing those cuts. These nicks can happen. So the predominant way of repair is non-homologous end joining. So you create a cut, plant by itself, they fix this cut. So a lot of times you will have deletions, leading to gene off or disruption. In the other way, you can have a homology directed repair. So for example, this is done by homologous recombination. You may have a template. The difference between this one and these two, this is nothing but SDN1, site directed nuclease 1, and this is 2 and 3. In this case, in the first case, there is no template. You go, make a nick, let the plant fix it, then look for the mutation that you created. In this case, you provide a template, which is a difference. So for example, you want to change a single amino acid, you provide a template, or any changes they want to do. That template is there. During the homology directed repair, that template gets incorporated in your DNA, in your genome. So that's nothing but the edit. And in the third cases, you might want to bring a big chunk of DNA, which we call a search and replace as a DNA insertion. So which is nothing but the SDN3. So I will give you a couple of examples of this technology. So the first one is the one I'm going to talk about is the high amylopectin starch corn. This is also popularly known as Vaxicon. So let's look into the commercial corn. Yellow dent is nothing but the commercial corn. So what is there? This appears as translucent. It is used in feed, food, ethanol quite a bit. And if you look into the difference between this and the next one is that in the yellow dent or commercial corn, 75% of the starch is amylopectin and 25 is amylose, and this is what it gives the appearance. Whereas in a Vaxicon, which already is there in the market, and Pioneer is one of the leading supplier of Vaxicon, this gives a appearance of candle wax like, and the application is mostly in food and industrial. The difference here is more than 97% of the content of the starch is amylopectin. Okay, and this has got lots of food as well as industrial applications. Now what is available right now? I'm sorry. So in the current Vaxicon product, the change in Vaxigene is responsible for amylose production. This particular mutation originated more than 100-year-old. It has got its own challenges. So this lags our elite line by at least 2 generations. So we can never have elite in the Vaxi background. So it is always lagging behind. That's one of the challenges. It also has some yield drag, right? So these Vaxi lines that we have right now can never be as good as our elite lines. So we want to fix that. And the other thing is that this is a partial deletion of the Vaxigene. The one that we currently sell is a partial deletion. So with Genomating, what we have been able to do, which is the next generation Vaxicon product is going to come to market, is we have changed the Vaxigene responsible for the amylose production, accomplished directly in the elite background. So please remember, so there is no back crossing, no moving from non-elite to elite. We are doing this in the elite background. So basically the parents of commercial hybrid, we are doing this one. And there is a very efficient solution to current Vaxi product challenges because these two are gone, right? And the difference between this and this is a full deletion of the Vaxigene, So what happens is Vaxigene is non-functional and you get this phenotype. So what is the current status of this particular technology, right? So we have regulatory policies for CRISPR-Cas are in the development in many countries, right? So United States Department of Agriculture recently confirmed that it does not consider next generation Vaxicon as created by genome editing as regulated. So it is not regulated by USDA, USDA Biotechnology Regulatory Services, and we continue to consult with the regulatory authorities in corn cultivation and import markets, regarding the regulatory status of next generation corn. Please remember that corn is exported to many countries, so it's not only United States. We need to have approval for many of those areas. But this is a really good example. So the example that I showed here for genome editing is the first version, where you go and create a deletion, a very precise deletion, right? The next example I am going to give is the second kind, and this is again very exciting one. So in this case, and there is a paper that we have published, so please feel free to go and consult this one. So we know this particular gene, when in a transgenic background, when you change the promoter of this particular gene, we know with multiple field trial data that we get good level of expression and good drought tolerance. So under drought the yield is good, under normal condition it is also good. We know this from our background. We have done a lot of work with this particular gene. This is what happens. So what we have done, and we know that happens because of a change of promoter, we use a different promoter. Then this gene is there, this is a native gene in corn. So in this genome editing approach, what we have done, we have inserted a native promoter again to modulate expression of this gene. So you can see these red histograms. This is the expression of this particular gene under different conditions. This is V3, V6, different condition. You can see the expression is pretty low in general in the wild type conditions, right? This is not a transgenic context, wild type condition. So we have created two different variations of this particular gene by genome editing, and which one of it is depicted in blue color, the other one is green color. And as you can see, in all these, in both these cases, in multiple different time point, we get very high expression of this particular gene. Okay, in both the variation, version 1 as well as version 2. Now we know from our previous work that a higher expression of this particular gene leads to good yield in drought conditions. So what happens in this genome edited line? So this is a 2015 field trial data. We have fresh data now again. It got confirmed. So you can see under drought condition, we get more than 5 bushel per acre more increase in yield. Under optimal condition, about 2 bushel, with the version 1. With the version 2, similar one, under optimal, it is even better than the first version. So I hope I am able to confirm you that, the efficacy of candidate gene to improve salt, stress tolerance. We have demonstrated the ability to use CRISPR-Cas advanced breeding technology to modulate expression of native gene, So this provides enormous opportunities, right? What was not possible earlier in a one of the big problem of the transgene world was that, once you put a gene, it goes to a random place, right? You had no way of modulating. So you had to do lots of screening to find the right one. This is a very precise way, right? The other point I want to highlight here is that, unlike the transgenic cases, in this case, you are creating the mutation, and that mutated line, you do not have any footprint that is coming from transgenic, because what you are selecting, unlike in the transgenic case, you are selecting for a transgene, right? In the genome edited line, you are selecting for the mutation. You are not selecting for the transgene. So there is a huge difference between the way these two are generated, right? So in the genome edited line, there is no transgene footprint. The only thing that is there is a very precise mutation that you have created in the background that you want to use, right? So that's why I used one of the phrase earlier that, for many of the applications, the final product is indistinguishable from the original product, right? So if you are creating, there is a very precise mutation. For example, if you just create a one single base for SNP, which leads to disruption of a protein, which gives a different phenotype. That is not much different from the the wild type plant that we have used for doing this. So with this, I would like to end my talk, and summarize the points that I have. So I hope I showed you that, we are using diversified platform and assays and gene sources feeding to our pipeline. We have identified several unique genes and unique modes of action that impact yield and yield stability. We have already seen positive translation in corn, so I hope some of the question about whether the control environment, whether the model system works or not, that is answered. We have devised strategies to optimize efficacy broadly across genetic background and environments, and we are leveraging CRISPR-Cas enabled advanced breeding technology for accelerated trait discovery and deployment, development and deployment. End of the day, we want to improve yield in farmers field, that's what we all want. So I hope you agree with me that there are multitude of tools that is available to us. It could be a transgenic platform, it could be a gene discovery platform using like a lot of model systems, positional cloning, gene optimization, genetic variation coming from the germplasm, breeding, and enabling technology as CRISPR-Cas advanced breeding. These together, we can get the product that we are looking forward. So what we are doing is we are gaining genetic knowledge and we are trying to make new varieties out of it. With that, I'd like to end and thank you for your patience. And I'd like to end with the famous phrase used by our former Prime Minister Lal Bahadur Shastri, Jai Jawan, Jai Kisan. Thank you so much for your patience. Yes, sir. It has got glyphosate, sir. Yes, and the first generation, yeah. Yes.
Released in India?
No. Actually, since we do not have any transgenic corn growing right now in India, so it is not. But Anand can reply is better.
Max minus glyphosate can be used.
Yes, it can be, sure. Yeah.
Yeah, you have to put it for the American.
Yeah, all the other traits that you need, insect trait and. Yes, Dr. Sharma.
Dr. Mohanty, how much off-target effects you got in your experiments doing CRISPR-Cas?
That's a great question. We do not see much off-target, and I think the key lies to the tools that we use. If your guide design is good, you will not see off-target. We have not seen. Actually, we have several, and I can give you example of rice. We have multiple edits that we have seen in in various stages. We do not see any off agronomics. It's too early to say how they behave in the field, but so far we have not seen off agronomics, or any other bad phenotype.
No, if you are targeting a trait which has more than one copy of genes, so
see,
how do you deal with that situation?
Yeah, so there's a great example, as you can see, that the Kaikse Gao paper from China, where she targeted 3 homologous chromosome in wheat, very precisely, and those are related. You got the disease tolerance. I think it all depends on how precise your targeting is. If your targeting is precise, if your guide design is good, you will not see off targets. Having said that, there are some reports in literature which says that there is off targets. So, but I think we have enough data to show that we do not see. Yes, please.
Yeah, my query is like Argos 8, the target is for drought tolerance, right? But in the version 2, the data says under optimal environments the yield has increased. How do we see that? Like the actual target is for drought tolerance, and the promoter and the editing has been done for tolerance to drought.
Actually that's a good position to be in, right? So we know, see, we have done a lot of work on this particular gene for many years, so we have multiple years data. So in general, lot of the transgene fail because they would give under normal condition, they would give efficacy. I'm sorry, under stress condition, they would give really good efficacy, right? The challenge happens is that in the normal condition, you will see yield penalty or priotypy, right? This is one of the cases where we see positive phenotype under drought condition as well as normal condition. So which is a good one to farmer is getting benefit, like you get 3 bushel per acre even in normal condition.
No,
so
my question is not the better performance under optimum condition. the question is like what was the wild type yield, and after editing under optimal condition, what was the wild type yield, and under
you saw in this case it was 3 bushel more, like even like see, please remember, in the wild type condition the expression of this gene is very low. I showed you the red histogram was very low, right? When we are putting with a different promoter, what it does is that in under all conditions it elevates the expression. It's not a huge elevation in expression, but there is elevation in expression, and that is what drives the yield, right? So if you look into the expression data that I showed you, both the variants under normal condition as well as under drought condition, there is a higher expression. Whereas the wild type gene, when it is only wild type is there, the expression under different condition V3, V6, under 24-day DAP and other condition is still very low. That is what is the key.
Is it because of pleiotropic effect of the gene itself, or due to off-target?
I would not say pleiotropy, I would not say off-target, and I think this is a beneficial effect of the gene, and we are able to do this because this is very targeted. And the promoter that we chose also is is a key. It's not a very strong constitutive promoter that we have there.
Wonderful. You have rightly mentioned that off-target is relatively less in this system compared to JFN or TALEN. I also agree to that point. But my concern is that suppose when you are targeting that particular gene and you have got your target, okay, you have disrupted the particular gene, put it off. Now, gene never works independently. They work in network mode. So, what is the effect of disrupting one gene on the other?
Yeah, and that's a great question. And of course, yeah, the genes work in a combination. So you really need to have good biological information for the editing that you are doing, right? Unless you know really well about the gene, I don't think you can do successfully editing. So end of the day, whatever we do, we do genome editing, end of the day it has to show good performance under farmer's field, right? If they don't, that's not going to show that in the light of the day, right? And we do I hope you saw from the earlier speakers, we do lots of different trials before anything goes down to make it a product. So if there is any in that interaction, if there is any bad interaction, which will obviously show in the phenotype, that will not go up, go downstream, I mean.
Good evening, sir. Sir,
Yes.
my question is, in one event, how many genes can be targeted through this particular technology?
Actually, this offers you multiple options, right? Even in one experiment, you can you can target multiple one. How many number? I can't give you exact number because I have not tried too many. But multiple one is possible. Because I can't give you a number of like 5 or 10 because we have not done those experiments. But with the precision that we have seen so far, you should be able to target multiple genes in the same experiment.
So we can do something kind of pyramiding kind of thing.
Yes, that's a possibility, definitely.
Thank you, sir.
Dr. Singh.
I had a query as quite general in nature. As we know that there are 3 approaches in CRISPR-Cas9, SDN1, 2 and 3. Which one is deregulated as non-transgenic technology in US, and what is the understanding or status for the most advanced one like SDN3 and 2? And the product that you have, high amylopectin maize, comes from which approach?
So yeah, one clarification I'll say that this is a expectation of a product. We do not have a product as of now in the market, but we have gone. So the example that I showed you was the Vaxicon. That's the SDN1 approach. Basically, we have deleted the Vaxi gene, and that is what USDA has given us the verdict that this is not regulated. Now, how SDN2 and 3 is going to be regulated? The consultations are still going on, and I don't think we have as of now very good clarity, how they are going to be treated in different countries. So I think things are evolving. The regulatory paradigm is still evolving, and it is going to take some time before it is settled down. But the expectation is that SDN1, as you saw, that this is basically nothing but creating a very precise mutation. You do not have any footprint. So scientifically, I do not see any reason whatsoever why they should be regulated, because there is no footprint. You are creating small mutations, or deletions in that case. I hope I answered your question. Yes, Vishwanath.
And the recombinant protein. So, whether your company is also exploring that option, or you think that the conventional transformation, agrovector mediated transformation or bialistic is good when it comes to final regulation of the
Yeah,
product?
so again, that's a great question. I think if one is able to establish those kind of system, that would be really great. So if you are able to, maybe use a protoplast-based system where the delivery is transient, and then regenerate plant, But all of us know that it's really difficult to regenerate plants from protoplast. Uh, those of us who did PhD in 90s, that was the first experiment we did, and believe me, it's not easy. Okay, and even today it's not easy. Okay, so we can get transgenic plants much easier with a callus based system than with protoplast. But yeah, that offers a great opportunity. If one is able to crack that, it allows you enormous opportunities, because then you can probably do transient, the delivery of the tools that is required, and get plant from there. So maybe it will make the things even simpler. But I think technically it is quite challenging to do that, Okay, I don't see any more questions, so I want to thank you once again. Thanks for your patience, and hopefully all of you who are here as students, you are going to take away something good from all the sessions are going on. And I would also request you, please do not hesitate and interact with the faculties, so this is a great point. One of the reason of this student symposium is to interact, please do feel free to ask question. There is nothing like a stupid question, this is a question, so please do feel free to ask question. Okay, thank you so much for your patience, and thank you Babu and Dr. A.K. Singh for giving me the opportunity. Thank you.
Thank you, sir. Thank you for elaborating the genome editing technique and its application in the easiest possible way. Now I pleased to invite our next and the last speaker of this session, Dr. Dindo Tabano. Dr. Dindo Tabano is from Philippines and obtained his PhD from University of Minnesota and worked on quantitative genetic aspects of corn with Rex Bernardo. Dr. Dindo leads the molecular breeding discovery as well as the deployment activities for the Southeast Asia region in corn as well as hybrid rice. And today he is going to talk to us about a game changer technique in corn breeding called double haploids. So please welcome Dr. Dindo Tabano.
Good evening, everyone. We're down. So after a long day, we're down to the last presentation. So I'm hoping that this will be a sort of light presentation and not very easy to, not very hard to digest. So this is all about doubled haploid, and so I'm going to talk about briefly some introduction and spend several slides in the steps on how to do how to employ DH technology, and then the last slides to talk about applications. Okay, so angiosperms such as corn, they do have a gametophyte portion of the life cycle, and that's the part of the life cycle where the plant, the organism, actually exists as a haploid. And so when we say haploid for a diploid organism, if you have two sets of chromosomes, so when you say haploid, you only have one set of chromosome. And when you have a chromosome and you copy exactly the same chromosome in the same cell, then you double that chromosome, and that's why we have the concept, which is a doubled haploid organism. Okay, so the context is diploid, that's why we call it haploid. But for hexaploid organisms, when you have a cell that only has one complement of the chromosomes, we would call it monoploid. Okay, so I hope to be able to show to you how we appreciate the DH technology because it benefits plant breeding to a very big extent. So the basic steps. So there are 4 basic steps. One is to be able to induce haploidy, and then next is to be able to identify the haploids, and then the third is to double the chromosome complement, and then to produce the DH plants. And then there are certain aspects that we want to consider when we want to employ DH technology in a commercial breeding program. So first is genotype independent. So if you've heard an illustration of this concern is of this matter is, for example, in rice agriculture, we know that indica varieties respond to a lesser extent than japonica ones to agriculture. So that's not genotype independent. So we don't want that. So if we want to employ a DH technology, DH procedure, DH induction procedure in a commercial breeding program, as much as possible, the technique should be genotype independent. And then it should not favor one parent over the other. So when we do the cross, when we do the breeding cross, both chromosome complements from both parents should be represented. And then once we have the DH lines, we want those DH lines to be normal and stable. So this technology was first put forward in 1958, and we call it Stock 6, and the authors call it Stock 6, and so the observation was that there was a certain type of corn genotype that when it cross pollinates another corn, some of the kernels have haploid embryos, and then the rest of those in the list are improved versions of the Stock 6, and RWS and UH400, they have an haploid induction rate that are at least 8%. Okay. So number one, how do we induce haploidy? So in plant breeding we cross good by good. So you have a parent inbred A, we cross it with a parent inbred B, and then you get the F1, and either you induce at F1 or F2, it's up to you. And then so the inducer serves as the pollen parent, and then this is when you induce the haploidy. So in this illustration, some of the F2 plants are successfully doubled in terms of their chromosome complement. The next step is how to identify the haploids. Okay, so in this DH induction technology, wherein we employ the inducer, so this is the kernel, this is the seed that we want, that we're after. So this inducer has a color marker, and so the haploid embryo does not have the pigment, but the endosperm has the pigment. So this is what we want, and depending on the inducer that we use, so for example, we get about 8%. We recover about 8% of this haploid embryo, and so we do embryo rescue, and we plate the embryos in a media, and the media contains the doubling chemical reagent. So it's in vivo because the haploid induction was done outside of the lab. So we do actual crossing, fertilization. But then after that, we do proceed to a tissue culture type procedure. So here, after we plate the embryos in this media, then we proceed to a normal tissue culture type procedure. So once the plant regenerates, we transfer them to hardening pots, and then finally grow them out in net houses, and then harvest the kernels, which are now double haploid lines. I'm gonna skip mustard. Just just a summary or an overview of that whole process. So you do your breeding cross, and then you grow your F1 seed and pollinate that. Use the inducer as the male. Okay, and then we generate the DH plants and grow them out and self-pollinate to produce to increase the seed, and then the breeders will come in and evaluate those double haploid populations. So this is the very reason we want to do we want to use double haploid techniques. So we do so whether it's a self-pollinated crop or a cross-pollinated crop, part of the breeding process is what we call line development. So we do breeding process, and we do generation advance to get homozygous pure breeding lines. And using the conventional way, it takes so many cycles. And here up at F7, at the 7th generation of self-pollination, you only get about 99.2% homozygosity. But with DH techniques, you get double haploids in 2 generations. Okay, so essentially, the very idea is to save time in getting pure breeding lines in your breeding cross population. Okay, on this slide is the message of this slide is that you get a higher probability of target gene of isolating the individual with a fixed gene. So in DH, you only get 2 types. So at one locus, you only get the 2 types, the homozygous dominant and the homozygous recessive, whereas in the conventional F2, you get the 3 phenotypes. And so you have twice the probability of being able to isolate the recombinant that you want. And in terms of field testing, phenotyping, so in a traditional F2, F3, F4 population, you have segregation within lines, and so that adds to the variation. Whereas when you have doubled haploids, when you have recombinant inbreds that are doubled haploids, the error is lesser. You reduce the error, and then it's the same. Basically, it's the same message here. The variation. So the variation that you observe is among lines, and the variation within lines is minimized. And so our phenotyping is more. We improve the precision of our phenotyping. So this is an example of the expression of disease of a disease trait among lines. So the uniformity. So you get the uniformity of trait expression within lines, and then the maximum separation of the disease resistance across or among lines. This example is for northern leaf blight. Okay, so again, still on selection efficiency. So for a trait that's for a dominant trait, if you have an F2 generation, for the dominant phenotype would be 3/4 of the population, whereas the recessive phenotype would be 1/4. But in doubled haploids, when you have a doubled haploid population, you only get the big A, and the small I homozygous lines. So it's there is no dominance variance, and you get more the additive genetic variance. More effective field evaluations. So when you have because there is less variation within lines, then your phenotypic evaluation is more effective and more precise. And the additional benefit is that since you no longer have you no longer have F2 I mean F3 F4 F5 populations in your breeding materials, so it saves a lot of resources. And then from the point of view of molecular breeding, so it helps us with estimation set design and also in genetic mapping. We want recombinant inbreds in genetic mapping, and so if we have DH, then we can do genetic mapping more quickly. And that's it. Thank you very much.
Thank you, sir. Thank you for explaining about the double haploids. Now the session is open for discussion.
Thank you, Dindo. I would like to have an idea, like you have two heterotic pools, and the improvement is going to be within the crosses within a pool, right?
Yes.
What would be the incremental gain you expect when you how much incremental gain do we expect in terms of improving the yield itself for the inbreds when we use only the germplasm within a heterotic pool?
Yeah, so we would expect a better gain in yield. I mean, we would expect a better genetic gain. One of our previous speakers, Emerson, he presented a graph earlier. So that would give us a very good view how using technologies, not only double haploids, give us a better genetic gain.
Yeah, so just to add to what Dindu said, so Gopal, if I am correct, what you are asking is what per se yield improvement in the inbred line is possible when you go through double haploids, right? Is that the question? So I think technically there is no difference between whether you go through the conventional selfing way or you develop the material through DH pipeline. So that is technically there is no difference. But then it's because of your ability to generate large number of DH lines. So the chances of ending up with a DH line with more per se yield is definitely better. So it is just the numbers game. Yeah.
What is the doubling technique and what is the percentage of success?
The doubling technique. So right now the DH inducer is commercially available, and the two of those types of DH inducers that I showed in the table, they give you about at least 8% induction rate.
No, but how do you double? What is the colchicine percentage?
Oh, so in terms of induction using colchicine, I'm gonna need some help from my colleagues.
Sir, actually, some of the protocol details we may not be able to reveal, so we will have to contact our legal department before we give out those information. So I hope it's
What is the percentage
yeah.
of success? You say that 100 haploids you are giving it
Anand, you want to comment?
So for, I mean, typically for every 100 kernels that we give, we probably end up between 50 to 100 double haploids. So it's two seeds resulting into one DH or one seed resulting into one DH in terms of high efficiency.
Before doubling, are you assuming that there is really haploids?
Yeah, when they do the markers, we clearly know if they're haploids or if they're coming from F1s. Yeah, but based on the color marker, that's our right. And again, we are presenting this data. We are not the technical people leading the science process there. So we may not have answers for some of the deep technical questions, but we can connect to the right people if you want to.
Yeah, getting back to the same question, if I can link up with the haplotype analysis also, if the cross is between two heterotic pools, and then identify a DH inbred which is having a different haplotype or possibly a complementary haplotype from the, since we have the haplotype information from the other hetero, each of the inbreds possibly it would be there, I suppose. So the getting a inbred through inter pool crosses with higher performance would be much better than the pool itself, and still be able to make a combination with the haplotypes available within the other pool. Would you think it would work at all, or it is not? It won't work at all.
And inbred across head pools. And inbred across head pools.
So that is clearly a banned practice. So we don't cross between the groups. No, it's basically because of your ability to predict the performance in future. So that the predictability comes down. So that's not a good considered a good practice. Emerson.
So when I think of the crosses that have been made between the head groups, when you have a recessive gene that it's bringing you advantage, you're going to have to have that in the hybrid as well. So there have been cases where we had recessive resistant genes and we had to cross, but double haploid wouldn't be the best technology for that. A back cross to introgress just that very narrow region, it would be best. Because as you have seen there, if you're just going to create a double haploid population, you're just going to shovel the entire genome. So like years that you have put an effort in selection and purifying those pools, you're just going to reshovel everything again and go back to the point zero. So if I think of any crosses between the head groups, it would be just for a single point, and you'd back cross back to the original head pool.
Dr. Professor Pentel was mentioning about introducing new germ plasm. Normally it is elite into elite crosses. So if at all have you experimented with any cases wherein new germ plasm is being put in and tested for increasing any of the traits, except those cases where you mentioned for disease resistance possibly you are doing a back cross grading and then reconstituting the genotype. Because most of the cases what we hear in all these agrobiodiversity conferences and most of the cases we are narrowing down the gene pool, and if at all if there is
Probably Anand has a better question to that, but it's a very good point. One of the strategies that we use is always introgress into one of the groups, so the other group is always going to be clean. The other strategy is to use a test cross. So you test against the selected number of testers on both group, and whichever respond better, you introgress to the opposite one. And now with the acquisition of Panar, as you've seen in the map there, we had this challenge of entire gigantic set of germplasm. So what we're doing is using molecular whole genome approach to align them, and then we're putting them in whichever group that they're more closely related. So either is one is a no-brainer. You pick one group and you always put it there. The second one tests cross-based on the phenotype, and the other one is the genetic data.
Yeah, when it comes to diversity, we want diversity, but we don't want just diversity as is. We want to be able to characterize diversity and organize this diversity into useful information. I think that's the message.
The one question to you and one to Justin or the other gentleman who I'm sure can answer. What is known about the genetics of induction phenomenon in terms of major and minor genes and the background effect? That's to you. And the second question is that, the molecular diversity in relation to heterosis. This has been one subject of investigation for very long. 1989 Lee paper in crop science started, and still it is an unresolved issue, how far we can rely. I would like to know, is there any SOP for formation of heterotic pool, standard operation practice for the crops where heterotic pools are not available? In maize, since we had a reference population available based on pedigree history and nicking ability, which is not the case in many other crops where we are struggling, for example, in case of cotton, also in case of rice, the efforts have been made, but there is no standard operation protocol. So how to combine the phenotypic data and molecular data to develop a heterotic pool, how to proceed about it? These are the two questions I wanted to answer.
I can take the first question. Genetics of induction ability. So CIMMYT did a few genetic experiments to identify QTLs for the induction ability. So what did those publications are available. So they were published in Genetics, the journal. So there was one large effect QTL on chromosome 1, so which was explaining close to around 15 to 20% of the genetic variance. So that was one of the major genes for the ability, but then that was not the only thing which is required for say 15 to 16% induction ability. So you still need more minor QTL. Those minor QTL information is not well detected. So we know that there are a few other QTL on the genome, but then they are not yet detected. But then the one which is definitely has to be there is on chromosome 1. So that's a large effect QTL, and University of Hohenheim, Professor Albert Melschinger, and in association with some Chinese team, they are almost very close to cloning that gene along with the CIMMYT. So I think that information will come out very soon. So that's very well known. But other thing is like it's sort of oligogenic, we can say. So it's not polygenic, it's oligogenic. The second question regarding heterotic group formation. So there is no one way in case, in crops other than corn. So in corn at least major heterotic patterns are established. So in tropical we have taxpenio, non-taxpenio, and SS, NSS. In Europe you have flint and dent. So you can stick to one of these things and proceed. But then with in crops other than corn, if such things have not yet been established, perhaps I think one thing is like use molecular markers and just identify the diverse groups. So it may not assure you any heterosis, but then it just helps you in separating those groups, and then you start breeding within those groups. So you are sort of artificially creating heterotic groups. To begin with, there is no assurance that heterosis will be guaranteed, but then when we do this over a number of years, then we start really seeing the benefit. Say if we have gone through some 20-25 years doing this in a breeding program, then we start seeing clear separation of the groups and clear separation of effects. So Anand, you want to add something?
So I think one other thing that we can definitely consider, and obviously the marker data helps quite a bit, but you know a lot of these crops are also defined by their sterility systems and how you could do this for example, a 3-line sterility system or a 2-line sterility system, the availability of genetic and cytoplasmic sterility, and the female producibility. At the end of the day, hybrid has to be producible so that it can be done. So that sort of defines what could be females, but then identifying, then using a line tester on what is compatible with these lines was some of the things that we have started doing for these other crops that are mostly self-pollinated and could be considered potential hybrid crops in future. I don't know if Justin, if there's something else you wanted to add.
Yeah, I don't have much to add to that. I think that that's a good point. They both made that the heterotic groups are somewhat arbitrary. The main point is to have two different pools of germplasm that never really touch each other, so that allele frequencies diverge. And so what is dominance variation in the starting population becomes fixed or additive when the two populations are crossed together. And so that can be done in a very arbitrary fashion. And so the way it was done in North American maize was, as Anand was suggesting, some individuals made good females because you had good female yield. And so the heterotic, that became one heterotic group, the stiff stalks, and then the non-stiff stalks were just the lines that happened to combine well with those. So it was an arbitrary distinction, which is how it starts. And as Babu said, you may not have a lot of heterosis to begin with, but you get it over time. So I'd echo both of what they said. And the other thing that markers can give you is if you're trying to set up heterotic pools and identify what combines well with your females, if that's what you're going to have to start with, is what makes you good females. I mean, ideally you'd cross everything by everything, which is just a giant experiment, right? But if you use molecular markers, you can identify clusters of inbreds that are related to each other, and maybe only test one or two in each of those clusters and use predictions to do the rest. So what markers can do is help you, rather than test the entire factorial array of possibilities, is to just test a sample and then use the rest, use that information to predict what might be happening with the rest. So I think there's a lot of, it's still arbitrary like it was in corn, but there's a lot of ways that marker data can help that happen more quickly and more cheaply.
In this context, another thing that bothers me is that with a large number of hybrids available in the market from private sector in different crops, more so in case of maize, the development of inbred from hybrid, commercial hybrid, has become a common practice by each companies. Now, you may try to see that the heterotic affinity is maintained when such line breeding is done. But somebody uses your hybrid and he does a line breeding program, and when he has to put those inbreds derived from a commercial hybrid from Fanier into a heterotic group, it amounts to each company has to maintain its own heterotic grouping, which goes contrary to the fact that we are talking about Flint and Dent and so on and so forth. They were pretty well established programs and there were never intermixing of the kind. But this is happening very frequently in case of rice. The line breeding is very common, and then once they are developed, people don't know where to put and only looking for some commercial gain. But ultimately there is a loss of identity which will threaten to the program in the long run.
Sir, excuse me, I have one question. We know this year is considered as a pulse year. There is a lot of scope for both for farmers and companies for pulses. Whatever you have presented, the techniques, whether you are these techniques are only restricted to maize or rice like that, or you are continuing research on pulses, or whether you have developed any good hybrids in pulses like chickpea and pigeon pea, like that. Last from 2 to 3 years, pulse per rate of pulse per quintal it is around more than 9,000, 10,000 like that. There's a lot of scope. Yes.
So I think pulses is currently not the mandate crop. I think we have soybean as mandate in the US. I'm sure these technologies are quite heavily used in soybean as well, right? The genomic selection, the whole genome prediction stuff, it's all being used in this one as well. So coming to pigeon pea, these are not clearly currently mandate crops within the organization, but we know that institutions like ICRISAT, they have been using this, and one of the primary impediment to using all these modern tools is basic genome information. So that wasn't available till recently, whereas ICRISAT has now championed it. So now ICRISAT, because of ICRISAT's efforts, I think we have many genome sequence data available. So I think it's going to be possible in the coming years. Once you have that framework, basic framework, we should be able to evolve those techniques.
Just coming back to the maze itself, just I wanted to know how much effort is involved in double applied breeding in terms of infrastructure as well as team involved, even if you can indicate a proportion of your effort with respect to other. Okay? And second is, how much of the lines which are already being used in the hybrid program are coming directly or indirectly from the DH program. And third and finally, when you talk about the temperate lines, B73 and MO17 are two of the inbred lines where even currently for molecular fine tuning they are being used. This is temperate. So similarly, are there any reference lines even from the tropical group, or else even if may not be your, if you don't want to diverge from your company, even like CML lines, that where the two or even group of lines within a maturity can be used as a reference line for the diversity, like even similar to B73 or MO17, that kind of thing. So what are just complement from your experience?
Sure, I'll take the last question based on my CIMIT experience. I'm free to disclose whatever I know based on my CIMIT experience. And the other one on DH, I'm sure Anand will comment. So in corn, as Dr. A.K. Singh also said, it's relatively easier to make a choice. So in case if we are in this region and we are worried about only the tropical germplasm or subtropical germplasm, I think you have very good choices in CIMIT lines. So there are two CMLs, one is CML451 and the other one is CL02450. So these two define two largest heterotic groups for the CIMIT tropical germplasm. I think these are good starting points in case if we have a program in which we haven't made any investment on heterotic group separation. I think these two lines are excellent lines and they work very well for the Latin American tropics as well as Asian tropics based on my CIMIT experience. So the proportion of DH lines, perhaps I will request Anand to comment. Yeah.
So just to add to what Babu said, I think CIMIT also has been contributing a lot of these tropical lines that have been used by private sector as well throughout the tropical world. So coming to the other question, in most geographies currently 80 to 100% of the breeding programs are through double haploids, starting with the first stage of testing. So some places it is completely double haploid, some places it is for a reason they have kept it at a slightly lower level because you still need some flexibility with back cross breeding and things like that, and want to bring in trait specific improvement, then that flexibility is what you get by having some of the non-double haploid approaches that are available.
Hello sir, I have a very basic doubt in double applied. Actually, through conventional breeding, when we are developing inbreds, we are having a very residual amount of heterozygosity also. But in double haploid, when we are processing it, so at that time we are not having any kind of heterozygosity. So that means when we are developing inbreds through our conventional breeding, so the maintenance is always okay for that. So in that case, we can say that the the inbreds will be performing good only. But in case of double haploid, is there any kind of, that means when the haploid lines are developed and they are making double haploid, so is there any kind of deterioration in the performance of inbred developed by double haploid?
Is there any kind of
compare, that means any kind of decrease in the performance of inbred developed by double haploid? Actually, the whole of loci in double haploid is completely homozygous.
So if I understand correctly, if you develop DH lines versus if you develop inbred lines through conventional means, is there a performance difference? Is that what you are asking, right?
When you're developing through conventional line, there
capacity and homeostasis would be inbuilt in those lines as compared to the DH lines where there will be 100% fixed for all the loci, and whether that leads to any performance difference in terms of adaptation, and whether someone has compared say 200 DH line from the same cross versus 200 embeds derived from the pedigree method from the same cross, and whether there is any difference.
I think it's a very good question. So I really do not know, but at least we haven't seen any obvious performance differences because of that residual heterozygosity. But if there is anyone else who would like to comment.
I think the other thing that we have to keep in mind is when we code in inbred, it's an F6 or an F7. It has 99 point something at that. But as the line gets used in hybrids, it gets to the next 5 or 6 generations. So, I mean, it would have very little left. But also on the DH, we have not seen any effects in terms of its ability to be produced differentially compared to a conventionally different line. So both lines for us in terms of maintaining the female pose the same level of challenges. Inbred is always an inbred.
Just one more additional point, I think one thing has been established. So several cycles of meiosis that the conventionally derived inbred lines go through, that is clearly an advantage. But then in the kind of elite by elite crosses that we nowadays make, so DH definitely is superior to conventionally derived lines.
Thank you, sir. Thank you, sir. Now, with this, we are completed with our second session. Once again, I would like to extend my heartfelt gratitude to all the eminent speakers of today's session and all the minds of science present over here who give their precious time. Now, our next program is poster viewing session, which is arranged in the division of genetics.
One more addition to the announcement. After the poster session, all of you are requested to attend for the dinner, which will be starting at 7:30. And tomorrow's program will be starting sharply at 10 a.m. All the students, SRFs and research associates are requested to be here by 9:45. Yeah. Huh? Faculty also, yeah.